Good morning, ladies and gentlemen. I am Gérard CREUZET, Fellow of the National Academy of Technologies of France and Chairman of the Steering and Organizing Committees of this conference, CAETS 2022. On behalf of the French Academy, I would like to send you a warm welcome to all of you here in person or remotely through the digital BlueJean link. Along with the committees, we decided 3 years ago to focus this conference on breakthrough technologies for healthcare. The COVID pandemic has shown us how relevant is this subject. For sure, technologies play a crucial role in solving the challenges we must face in the health sector. I wish you a very exciting conference. And without any delay, I invite Denis, Chairman of National Academy of France and Chairman of CAETS, to join me to give the opening address.
I am very much pleased on behalf of the French Academy of Technologies to welcome you for this CAETS 2022 conference here in Versailles. There are here many of us, and thank you for the participants for having made the trip, often from far away and even from the other side of the planet, down under. I think we are happy to gather again in person and pleased to come back to the formula of physical meetings after 2 years of interruption caused, of course, by COVID. Not everyone, sadly, could be here because the pandemic is not over yet and has prevented some of our member academies and many individual members from being present. Also, in the context of international tension, we also have great concern for our whole world, as we all know. So, I also want to greet those who participate virtually through the network and welcome to them, even if they are further away. I would like to continue by making some thanks. First, of course, many thanks to Minister Sylvie RETAILLEAU, the French Minister for Higher Education and Research. She will address us right after my short introduction. Sylvie, it's a privilege and an honor to have you here. And I welcome also the members of the French Parliament and Madame la Préfète. I would like also to thank our sponsors, without which we would not have been able to hold and to fund this event. And I will extend a particular thanks to Dassault Systèmes, our main partner. By the way, on Thursday morning, there will be a parallel event, parallel to the CAETS event, an event held by Dassault Systèmes, which will illustrate how digital technologies and virtual universe do trigger progresses in medicine and in sustainable development. But back to our CAETS conference here. It is the second time, in fact, that we host in Paris the annual conference of CAETS. But it is the first time we do it as an independent academy. In fact, we are just 22 years old, and the first event held in Paris was held by our mother, the Committee of Applied Sciences in the French Academy of Sciences. And in between, we became an independent academy. As Gérard said, the topic, of course, of this year's conference, disruptive technologies for health, no doubt that is an extremely important subject, which affects all mankind, and which therefore fits perfectly well with this assembly, bringing together many countries from all continents. In addition, the recent pandemic has shown how important technology is, not only to cure the disease, but to prevent it through the vaccine, but also to measure and control the spread of the pandemic through tests and data processing. This is not new. History of medicine is marked by major contributions of technology to diagnosis, to therapy, and to prevention, with constant improvement of each technology since the first inception. It would be out of purpose to detail all of them, because in my life in Thomson CSF, which I renamed Thales, I was used to work with image intensifier tubes. I will just say a word about X-ray. Just think that it's not far away, at the end of the 19th century, that Röntgen discovered X-rays. And by the way, he received Nobel Prize for physics for that, the first Nobel Prize ever attributed in physics. And the application to medical imaging went very through after, first with radioscopy, then with radiography, improved by my famous image tube amplifiers, which are now become solid-state amplifiers. And finally, invention of scanner at the end of the third quarter of the 20th century. Other imaging technologies, of course, have followed through the use of ultrasound, and we'll speak about ultrasound in that conference, or through nuclear magnetic resonance. More recently, genome sequences and new tools for modifying it open immense fields for new therapies and for vaccinations. Our Academy is very proud to count among its members, its fellows, Emmanuel Charpentier, who received the Nobel Prize for invention of CRISPR-Cas9, along with the American Jennifer Doudna. Finally, I will not forget the considerable contribution of digital technologies to medicine, whether it is artificial intelligence for diagnostic aid, the processing of massive data required by genome analysis, the use of simulation and modeling, the telemedicine, the storage and sharing of the patient's medical information, and so on. And also recently, the use of data and simulation for the pandemic management, for the epidemiological management of the pandemics. But, and it's important but the paradox of our time is that as the benefit of health technologies becomes more and more evident, and by the way, more and more rapid, an increasing number of our citizens, especially in developed countries, have started to express doubts about the reality of such contribution. This is just one particular case of wider skepticism towards science and technologies, and vis-à-vis experts of all sorts. These phenomena must challenge us, and we have to address it as academies. It is a great concern. Of course, these doubts, or even those oppositions, become particularly important and delicate when they affect health technologies and the human body. This is why our approach in our academic work must be marked not only by the greatest scientific rigor, but by the greatest possible sense of ethics. We must listen to this protest movement, not sweep them away with the back of our hand, but try as best as possible to listen to them, to understand the concerns, and then enlightening the public on the reality of progress allowed by technologies, but also in addressing the risks they may carry when not properly designed and properly implemented. This concern will be manifested in our conference by the great place given to ethics, which will mark the whole thread of the presentations and will serve as conclusions. Here in France, our Academy is particularly sensitive to the need of a good understanding of technologies by our citizens, for making sure they bring real progress, and by giving all the relevant elements to the general public for an informed and rational democratic debate. This is, no doubt, a difficult task, and probably the most difficult of all our missions, especially because the modesty of our means compared to the power of media, and even more this of social network and other digital means of influence. Nevertheless, we are working on it with determination, in particular by seeking partnership with the medias and the press, whom I thank to their presence today. Finally, I would like to say very short words about our Academy of Technology here in France. We have 350 fellows, all volunteers and working pro bono. They are all identified and elected by the peers through a rigorous process which guarantees their skills and their independence. All this being, of course, reinforced by our collective work processes. Independence of individual may be challenged, but independence of our academy, because we have cross looks at things, cannot be challenged. One important mission for us is, of course, to provide the government and parliament with the advice they need for conducting policies on technologies. In this regard, we are pleased and proud to have been recently entrusted with the task of advising the government in its plan called France 2030, which mobilizes 54 billion euros in funding the technologies for the future. It's the beginning of the task for us. It will be a long way, but we are very proud to have been put at the center of the French mechanism funding new technologies. In this context, we are happy and proud to host this CAETS 2022 edition dedicated to technologies for health. I hope you will agree that contributions to the conference and quality of the speakers are at exceptional level and will deserve to organize further outputs well beyond the distinguished audience we have today. For our part, in France, we will make the best use of it for the enlightening of our government and our citizens and our companies. And I have no doubt that our CAETS international community that we constitute here, which is quite unique in this field, will be able to do the same at international and national level. Thank you very much. I wish you an excellent conference. Ministers, the floor is yours.
I would like to offer you first a warm welcome to Versailles today. Many of you traveled a long way to be here today, and I am really very happy that we can finally meet each other in person today. Since COVID, this is indeed the first time the International Council of Academies of Engineering and Technological Sciences has been held. And Denis has just spoken about vaccines, and more specifically RNA vaccines that have largely contributed to the fact that we are able to be here together today. Speaking of vaccines, many of you know that France is the birthplace of the vaccine, discovered in 1884 by Louis Pasteur. And perhaps you also know that Pasteur lived 5 kilometers from the place where we are standing. However, Louis Pasteur was not alone in the process. He was able to create the vaccines thanks to Edward Jenner, a British researcher who worked on vaccination a few years before. It shows that science is a collective and international process. More than a century later, no one can ignore the role of international collaborations. Without them, we would not be here today, vaccinated only 2 years after the outbreak of a worldwide health crisis. Because I just talked about Louis Pasteur, I can't help but think of Marie Curie, as both of them engineered a better world through major breakthrough technologies. She left her lab in 1914 and created X-ray radiology mobile units, saving the lives of hundreds of thousands of soldiers during World War I. The discovery of radioactivity and its application to medicine is a technological breakthrough that still inspires us today and demonstrates the importance of science and technology in meeting the needs of our society. This is also why 2 of our prestigious institutes bear their names. I'm sure that many of you know the Pasteur and Curie institutes. Of course, This technological breakthrough also came with their share of errors. The X-rays overdoses in the 1920s, for example, which caused numerous leukemias. Because of the numerous accidents and deaths, the International Committee for Protection Against X-rays and Radium was created in 1928. This committee established protection rules for radiologists and technicians 3 years after X-rays were discovered. Innovation was clearly ahead of the associated issues, underlining the necessity to question innovation beyond its technological aspects. These two examples underline the relevance of today's Congress, the key role international exchanges and ethics plays a better world, especially in health technologies in the process of engineering. This is why I will follow today's discussions with great interest, and many things depend on them, such as our citizens' confidence in scientific progress, but also the right balance between progress and caution. Ethics is key in the appropriation of new technologies, so is a common international frame of reference. Finally, I am also very happy to welcome you today in Versailles in France, because what is happening in France regarding health technologies is truly exceptional. Let me give you a quick overview. Of course, COVID highlighted the fact that technological change is faster than ever, and in that regard, the RNA vaccine is a lesson to us all. We need to be more reactive and flexible, and we need to support more strongly research and development and technologies, as France has the ambition to be among the countries that inspire the speed of technological change. That is why we passed the research programming law, investing 25 billion euros of additional credits over 9 years, and also the France 2030 plan, which invests 54 billion euros on high-risk, high-gain projects in research and development. We are building a strong health tech ecosystem, which is a major focus for the government's economic policy. And our startup ecosystem is also flourishing, with more than 2,000 companies and also over 60 creations per year. And the sector is gaining in maturity. Leading companies are starting to emerge, and the excellence of French research also, with in particular INSERM, which is the second largest health research organization just after the NIH. But today, the big push came with the 2030 Health Innovation Plan presented by the President Emmanuel Macron in June 2021. And this plan was conceived in partnership with all the actors of our health tech ecosystem. In addition, we are investing 7 billion euros in health and research and innovation. And we will do everything we can to encourage the creation and also the growth of the French startups. All of this has one goal: to boost our future champions. Of course, investments are not everything. We also passed several reforms to shorten delays, and we are working on even more accelerated procedures for innovative and breakthrough products. I would also like to say that the Health Innovation 2030 plan package is something that has never been seen in the French health ecosystem before. With this plan, we can confidently expect that startup creations will boom and that a fast and steady growth in the field will take place within the next 5 years. More than ever, major breakthroughs will emerge. As you can see, I am really happy to welcome you today here for 3 main reasons. First, you represent how valuable international cooperation and exchanges are. Secondly, a real acceleration in the development of health tech is taking place in France, and it is really exciting. Thirdly, because I believe to these discussions to be crucial for the understanding and the acceptance of technological innovations by society. If we can build a strong ethical framework, innovation's acceleration will only lead to mistrust in technological breakthrough. So for me, it is a real honor for me to be with you today, as I know how important your work is. I now give way to Elias ZERHOUNI without further delay, and I wish you really an excellent conference today, and stay in France. Thank you very much.
It's a pleasure for me to address you. Today I was asked to give you my view of the grand challenges in life science research and development. And as I was thinking about the topic and I was thinking about the complexity that we're dealing with, I try to really give you a perspective that is more mine than generally accepted. But I think at the end I would like to also provoke some controversy, maybe a challenge for our community to address. So if you really wanted to remember something out of this lecture, I would just use the 2 words, complexity on one side and precision on the other. It's clear that as we learn more and more about living systems, their complexity has become daunting. And it's also true that for us to act in a way that is beneficial to society, we need to act with precision. And so the 2, precision and complexity, are the forces that are driving the challenges that we're talking about. And the challenges are made even more interesting because there is a scientific transition that is ongoing today. If you look at the natural history of any science, you find 3 phases. You find that there is a reductionist phase at the beginning, where small teams unravel the elements of a system, whether it be particle physics or atoms or the chemical elements, or in our field, whether it's the discovery of DNA or RNA or proteins or in their translation. It's really a phase of reductionist understanding. However, no living system works in a reductionist mode. It works in a complex mode and an interactive mode, which really is driving what I think is happening in front of our eyes, and that is a transition from a reductionist phase to a systems phase, where at the beginning of systems phase, we try to understand modules, the immunological module, for example, or the cancer module, and then we put all of that together into, hopefully, a comprehensive understanding of the system. And that's why I think the other word I would like you to remember is convergence. So complexity, precision requires convergence of sciences between physical sciences, biological sciences, data sciences, and other fields of engineering, because without that, we can't really make that happen. And the data tsunami that we're dealing with, based on our ability now to generate data at a scale that we never experienced, requires really innovation in data sciences, as you'll see. So when you look at it, you really wonder, why is it that it has become so complex? And the fact is that if you understand the reductionist phase, we've been building a world around the cell. And within the cell, we're building a world around DNA. So frankly, if you thought about what's the 21st century in life sciences, it's like the DNA century, just like it was the electronic century in the 20th century. And so as we build that, we started to understand that cells alone don't do the work, and they have to be organized in tissues and organs, and that leads to organisms. And the organisms have emergent properties related to our behaviors, and that behavior interferes and interacts with the environment in a bidirectional way. But all of that, think about the fact that that cell, at the beginning of your life, multiplies and replicates billions of times. And because of that, the replication errors make it impossible to have one individual, an exact clone, and another individual. So each one of us here is absolutely unique. There's no statistical possibility of you having a clone somewhere in the world. Your fingerprints are unique to you, your iris is unique to you, and that is the other side of the equation, that all of that complexity translates to unique individuals. So we can't really apply our understanding of systems to the individual without a translation of those complex findings. So when you want to understand the first challenge, think of it as two ways. One is to understand the biology, in other words, read the biology, and the other one is to transform the biology, to write the biology, to transform the biology for our needs. And if you look at it, what you see in the emerging technology landscape, which is evolving at a high speed, is that currently we can do high throughput experiments, but not at high resolution, or we can do high resolution experiments, but not at high throughput. The future is really emerging where we can see where we're going to be able to do both high throughput and high resolution experiments. And you see it. You see next-gen sequencing, you see spatial profiling, you see single-cell sequencing. We have better and better methods of separating the elements of a system, better and better methods of studying the function of these systems. And so that relates to then what I was telling you, which is the evolution of a reductionist view to a systems view is going to require a multiplicity of approaches, but those approaches need to be integrated. How? Because we will go from a modular biological system. Understanding the cell, for example, relates to understanding the genome, understanding the epigenome, understanding the proteome, the RNA world, and many other dimensions which require then data sciences to be able to really rebuild this in a coherent fashion. As you look at that, you wonder about exactly the structure of the whole system. So here I'd like to share with you my understanding of it. Think of it as nested networks. In other words, what we're dealing with is that each module is embedded in the other module, and another module, and another module. So you have layers of modules that are themselves complex, that interact also with other systems next to them, adjacent to them. And so when you look at that, you realize that we have a multiple dimensional system made up of multi-layered interacting networks. So I'd like to show you, it's clear that when you look at epigenetics, which control the DNA, and genetics and genomics, those are really controlling the cellular system and, by extension, tissues and organs through the ability for us to transcribe and translate that information. So that's another layer, transcription, translation. You want to understand the regulation of these interacting networks, and that really leads then to the expression of proteomics, the expression of metabolomics, and obviously we can't forget the environment, because we are in constant interaction, in complex interaction with the environment. And this is essentially what I think is the most difficult part, because when I mean an environment, it's not just the immediate environment of you as a person, but it's the environment of populations, the environment of the planet, and all the ethics that go with it, which we will hear about soon. One experiment that I like to show, which proves that the environment is very dominant right now, where 50% of our diseases are really environmentally related, is this one. This is the statue of David, and when I was the NIH director, I was able to bring the statue over to the United States for people to show and to visit for about a year. And this is basically a statue. It has no biological component. And this is what happened after 1 year in the United States. So, remember, the environment drives our systems. Now, what is the emerging translational paradigm that I would like to share with you? Under one hand, on the left, is what I call multi-layered biotyping. Using all of our technologies, we need to really understand the layer by layer by layer. Epigenomics, genomics, transcriptomics, and you see what I meant there, but I think at the end, we have to integrate that into a quantitative networks and pathway models. And this needs to be at the atomic level, not just at the tissue level or cell level. And this requires single cell analysis. It also requires the creation of new disease models, which are possible now with CRISPR technology. On the other hand, the phenotyping, the understanding of the individuality of each one of our patients, each one of our populations, requires us to study the extreme phenotypes. They're very important. When you see someone suffering from a disease that the population doesn't suffer from, that's information. That's fundamental information that we need to unravel. Longitudinal population studies are going to be critical. Aging, how does that really happen? And how does it go awry? Biomarkers are going to be important. We need to be able to generate biomarkers at a speed we are not able to do today. Solid and liquid biopsies, human genetics, real-time biosensing. In other words, follow the patient where they are in their environment rather than them coming from time to the healthcare system. Health records integration, a huge problem. How do we track the health records of an individual over time? And obviously imaging, as you've heard. But all of that cannot function without data sciences capabilities. And those data sciences are really fundamental and need to be supported and need to be integrated in what I call the convergence of sciences that is required to make progress. Let me just show you one example of a data science revolution in structural biology. You've heard about the DeepMind technology, the AI technology that was used to play games, the game of Go, but that was then developed at Google to also be used in biology. And the project that was started in 2006 was to address the fundamental challenge we have, and that is that we know how DNA creates RNA, creates proteins, and creates protein sequences, amino acid sequences, but we don't know how these amino acids then form and shape in the three-dimensional functional shape of the proteins. That problem has been plaguing biology for over 50 years. The team at AlphaFold, which is the company that broke the mystery, used 100,000 atomic level structures of known proteins which had been accumulated for decades in data banks that we had. And through that, this deep learning algorithm, which was based on deep teaching, they were able to solve over 80% of structures, and more actually today, by 2020. So in 4 years, they basically changed the face of structural biology. And recently, they've deposited 200 million new structures in the EMBL database this year. Imagine 200 million in a few months, as opposed to 100,000 over 50 years. That is not the entire solution. We still need to understand exactly how these proteins interact. We still need to understand what ligands can really attach to these proteins and change their function. Still a long road to travel, but this is a revolution due to data sciences. Nothing to do with biology. It had to do with the ability of humans today to accumulate large amounts of data and then integrate them into an understandable whole. Now, let me talk about the two challenges that are derived from this picture that I shared with you. The challenge of modifying biology is one that I think we need to talk about because the promises are great. There's genetic engineering now, from plasmid to gene editing tools that are available. The targeted gene editing with CRISPR technologies, which have revolutionized the field and earned a Nobel Prize for Emmanuel Charpentier and Jennifer Doudna, RNA-based control of translation, mRNAs, for example, for vaccines, but other forms of RNA controls. And obviously, the need for us to develop viral and non-viral vectors, because the biggest problems we face, the biggest challenge, is the in vivo delivery of all these very smart constructs that we are able to do now. Safety concerns, ethical concerns, expression controls. I mean, you don't want to really embed a gene that can essentially go out of control. You need to have controls over that expression. And the last problem, a huge bottleneck, is biomanufacturing, which I will tell you about. If you really think about what is the fundamental bottleneck to all of this, in my experience as a R&D leader and actually looking at biologics and antibodies, I'll tell you, the number one obstacle for us is the low yield and the high failure rate of biomanufacturing. It's one of the best kept secrets in the industry. I mean, you have essentially an efficiency that is too low. That's why we couldn't produce what we needed to produce for the pandemic on time for all peoples of the world. We couldn't because the yields are low. The error rate is about 15 to 30%. So imagine when you do a batch of an antibody or a vaccine, the batch is made in large vats and it costs about $10 million. So when you have a 30% failure rate, imagine what that does to the failure of delivering the vaccine or any other medication. In the United States, if you follow the news, there was a company called Emergent Biosolutions, which was used by J&J and used by AstraZeneca to produce millions of doses. At the end, they produced 60, 80 million doses that were just thrown away because they failed. So when you look at that, you say, there's no way that we can really progress. And you ask yourself, why is it that biomanufacturing is so inefficient? We use the same mammalian cells that we used 30, 40 years ago. All of our productions are basically related in mammalian cells to the Chinese hamster ovarian cells, which were discovered by Richard Axel at Columbia University over 40 years ago. And for a serendipitous reason, he was looking at the receptors for smell in the brain and wanted something to really express those receptors. And that's how he came up with the CHO cells, which were then patented and then used in the industry to produce all kinds of biologics. This is the technology we use today. And you know the yields? Grams per liter, if you're lucky. 2 grams, 3 grams per liter. It's just not sufficient. It's a tedious and long development with low yields. It's also very costly, except for high-priced medical applications where you can sell an antibody for thousands of dollars, then you can afford the cost of this manufacturing. But it will never serve the life sciences of today and the hope and promises of life sciences. And it's because we're attached to a very old technology of bioreactor technologies, which was used actually, you know why, you know how? In winemaking and beer making. And we're basically translating that into biomanufacturing today, despite all the progress we've made. There's no question that the industry will not take the chance, academia will not take the chance to transform this biomanufacturing because the regulatory pathway is very long. And nobody wants to change the regulatory pathway and be stopped. And so that is where I think governments have to come in and have to take the initiative. We have advocated this in the United States, and about a month ago, President Biden has announced that one of the priorities of the new funding is going to be discovery of new methods of biomanufacturing, and he dedicated billions of dollars to this effect. I think that in the $54 billion that France is planning to spend, and other countries, I know, I would hope that a significant percentage will be dedicated to discovery of better methods of biomanufacturing in the world. That is my message. On the top right here, you see essentially bioreactors from a biotechnology company. It's typical, very, very 10,000 liters, and then you have these batches that take 2 months to produce, and then if you're lucky and it has not gone out of control, it's a good batch. But 30% of the time, it will be no good. Look at this, which is you can see the barrels in the back. That's, I think, a wine or a beer factory. It's exactly the same thing. So do we deserve that in life sciences? My answer is no. We need to really break the mold, break the bottleneck of biomanufacturing. So that's what I wanted to share with you. If I could advance this one. It doesn't work. So well, the message is really we need a technological revolution. And if you ask where should that revolution be, as I said, complex systems, understanding them, I think we're on the way to do that. Biophenotyping at the individual level, I think we're on our way to do that. But no matter what we do, if we read and write the biology, we won't be able to produce it unless we realize the promise of life sciences through a revolution in biomanufacturing, bioprocessing. I think it's just as important as computer chip technology that everybody talks about. In agriculture, environmental, and energy technologies, it will be fundamental to have better ways of producing biologics. Let me give you an example. There's a company in the US that was created by a friend of mine to produce what they call molecular agriculture. So it's taking cells to produce proteins, to feed the world. It uses 99% less water than the typical agricultural methods. It produces no methane because it doesn't use animals, but it's impossible at current yields. We're talking about grams per liter. We need 100, 150 grams per liter. We need 10, 20 times more productivity to make that a reality in the world. The vaccine production that we've had, the world vaccine production is limited because the technology transfers are very prohibitive, and we need to have the ability to serve the entire world population equally. And the countries that master bioprocessing, in my view, will have a significant competitive advantage. So I'm really sending one action message to the Academy. Let's say something about the bottleneck of biomanufacturing. So, in conclusion, complexity, precision, requires the convergence of physical sciences, biological sciences, and data sciences. And last but not least, that convergence needs to be expressed in engineered solutions across many dimensions. And the one I would like you to remember is biomanufacturing. Thank you very much.
I'm very honored to be part of this conference and to contribute the ethical issues. So I invite you now to look through different glasses than you're accustomed to or than you heard before. So looking through the glasses of ethics means looking for the morally good and flourishing life. And life here is not as in life sciences, the biological life. Yeah, the material life, the molecular life, and so on, as life sciences has occupied life in a way. But that's the reason why I founded the Center for Life Ethics, because his life is much more than that. And health actually is much more than a biological endeavor, but I come to that later. So a flourishing life means that at the end of the day, or during the day, or at the end of the life, you say, this life had meaning, this day was a wonderful day, and I had lived a life, or I'm living a life that is really meaningful for me and for others. So ethics by design, what I'm talking about now, is not about theories. Yeah? So don't have the impression to be in a pro seminar in philosophical studies or so. Of course, there are a lot of theories, and an ethicist has to know them. But if you practice ethics by design in technologies, then it is about a shared experience of technology against the background of human rights as a common ethical framework all around the world. I don't say that it is practiced all around the world, but at least it's where the UN Declaration, European constitutions, and others are built upon throughout the entire process, from the idea of a health technology to the application. And Perhaps some of you say, well, technologies are value-free. But I don't think that this is true. The European Group on Ethics in Science and New Technologies, where I was chair until 2021, our last statement was on values for the future. And there's this wonderful sentence, I think it's wonderful: values are baked into everything. Because as a human being, you cannot live different to having shaped your actions according to values. You do something you find important. And there's a reason why you find that important. You cannot only behave like an animal or so, but you have to decide, you have to choose something. And that's always shaped according to values. And there are hundreds of values out there. There are individual and societal values. There's not necessarily one better than the other, but we have to be in dialogue which one is really valuable to be followed by societies. So if you choose a technology, if you design a technology, you choose to do that according to values. And another issue is very important for me. When I sit on a panel as an ethicist at the end of a conference, the first question I get is, where are the limits to what we are allowed to do? And I say, well, that's not the question for an ethicist. I want to talk about what we are asked to do, what is valuable to do, where we can harness opportunities and chances and make this world a better world. So not setting primarily limits, not being the responsible expert for the risks, but also for the chances, for the benefits, and where this world can become better. So values and ethics are no limit or obstacle to innovation and change, just the opposite. They are the gift of innovation and change, and they represent the compass indicating what responsible, inclusive, and sustainable ways of future making are. So another question mark I want to address regarding technologies is, how do we define technologies? There's this wonderful book, Why We Work, from Barry Schwartz, an American psychologist. And he defines it as the use of human intelligence to create objects or processes that change the conditions of daily life. And of course, we think mainly about thing technologies. Yeah? You can invent them, you can see them, you can touch them, you can repair them, you can destroy them. Those are thing technologies. But around those thing technologies, there are idea technologies. And you cannot see them immediately, but they are able to suffuse the whole culture before you even notice them. And one of those ideas is the image of the human being. There's another quote from Barry Schwartz that goes, "A theory of the universe doesn't change the course of the planets, but a theory about human nature changes human nature." So the way we think about who the human being is or what nature is changes the way we deal with it. And this is on a very situational basis in a doctor-patient relationship. If the doctor doesn't believe in the ability of the patient to overcome his addiction, this patient will not be successful in overcoming this addiction. But on a general level, societies build their institutions around what they think that health is, that a good life is, that regulation should be. And this is shaping the opportunities of our acting and thus shaping our ideas of ourselves and our self-perceptions. So these idea technologies were addressed from our president by saying we have to take account of what the societies think about what we are doing, about vaccines, for example. The minister addressed that. And it is not only convincing societies about that sciences and technologies are doing a very good job, what they actually do, but it's not only to tell them and to communicate what they do, and this society has to understand it, but it is to be in a vivid dialogue on eye level. So what do societies want to have? Yeah? And so the vaccines, I think, are a very illustrative example. We see here BioNTech eyes up to 17 billion euros in vaccine revenue in 2022. That's a great success. Absolutely. But on the other side, you see that not all people are taking vaccines because they're just not convinced that they are a good thing. Yeah? And we can think that vaccines are wonderful, but if the public doesn't take it up, there is no sense in this technology. So technology alone does not make the job. Yeah? You have to be in a dialogue with society, and you have the distributive justice questions like how do all people in this world for this global problem get the vaccines. So we have 67.9% at the beginning of September, I suppose, take up of the first dose in the whole world. In France it is 81% or so, but only roughly 22% of the people in low-income countries have gotten their first dose. So there's really a problem in distributive justice and in our regulatory governance framework to treat global problems as global problems. And this is also about these idea technologies around a think technology. So which stories do we tell about the pandemic and about technologies? What stories tell politicians, the society, about their pandemic management? And it's actually about stories. Yeah? Not stories are the opposite of science, but if science is not able to tell the right story to people about what the meaning of the sciences are, then you won't make a benefit for societies as far as it could be. And the policies for distribution of vaccines, I addressed that, is also part of the stories we tell about our understanding of the world. The European Group on Ethics, again, has made a statement on artificial intelligence, robotics, and so-called autonomous systems. And we had some principles and democratic prerequisites that we established to have a measure, a yardstick of how technologies can be assessed. Ideally not after the technology was developed, but from the beginning. This is not an encompassing list. It's not comprehensive. It's not the only thing you can think about that. But for this digital sphere, we thought that these 9 principles are mainly important. And I start just with one small example regarding human dignity. So there is this algorithm to predict the death or the probability of a patient dying in the next few days or weeks or years. And here was a group from Stanford University, and they made the experience that doctors usually overestimate the life expectancy. At least in these US hospitals, they assessed and examined. And they said, well, if we have an algorithm looking at the electronic health record of the patient, we can actually manage to be quite earlier aware that this patient needs palliative care. So this is a wonderful thing, to have an algorithm who tells the doctor, please have a look, this patient might need palliative care. Yeah, so the algorithm doesn't decide something, so algorithm can't decide anything anyhow, but only if we let them. But so this algorithm helps for a better healthcare. There was another algorithm with the same purpose, to see how likely it is that the patient will die soon. But it was developed by a company called Aspire Health. It has a lot of funding and investing from Google, from Alphabet, millions of dollars. And the aim was not to improve the quality of life of patients, but the aim was at least as well to cut costs for healthcare. So being applied in the domestic environment of the patient, and the patient suffered from, let's say, a heart attack or ischemic insult or so, then the algorithm said, well, it's not worthwhile to take him to the hospital for treatment, yeah, so you can really use your scarce resources in a more efficient way. I think there is a difference in these algorithms. The latter one was not transparent at all. Nobody knew what went in there, which data were used, and so on. The other one was transparent and explicitly dedicated to fostering the quality of life of the patients. So you see, the same algorithm with mainly the same purpose or the same object can be used in a completely different way. And this is where ethics by design, of course, comes in. When you then ask the question, which ideas are driving technologies for health? And we heard a lot already in these lectures and greeting words in this conference. It is about drug development, ICT and health, and so on and so on. You see all of that in the program. But what do patients and citizens and healthcare providers actually want? Yeah? Who is driving this technological development? There's a wonderful group, Patients Like Me, in the US, more than 800,000 patients that care for themselves and build technologies for themselves that really makes their everyday life easier and better, that they are more confident to be health literate, to be self-aware and self-efficacious. So bringing other stakeholders and players at the table to see where do we want this technological endeavor go would be a major advantage. And then you can ask what benefits individual and public health the most. And then there is this, and I'm a doctor myself, I was a doctor myself, there is this narcissistic problem for clinicians and so on, that healthcare is actually not doing the best or most important job for individual and public health. So what is done in healthcare only contributes up to 20 or 30% of the health state of the public. It's more about the social determinants of health, yeah? Like you see it in the bullet points on the right side: income and social protection, education, working life conditions, and so on. So a lot of things that you cannot address only with health technologies, but with idea technologies or other kind of policies. So I would wonder, if you have a health plan as a government, how many of these dozens of millions do you spend for education, for good teachers in schools, for having technologies to govern food or working life conditions? That would be a major issue for bettering health of the individual and societies. And then again, who is shaping technologies for health? They're engineers, technologists, software experts, and I very much appreciate your starting point from the complexities, yeah, and the convergence. But still there, where are the patients, the social sciences, and the ethicists? Yeah, it's more about biological and material and physical and natural sciences and so on, and it's great. Please don't get me wrong, that they work together, and it's really important that they do that. But it's not yet the systems level, because for the systems level, all these social sciences, humanities, societal issues have to come in as well. And here's an example why I would say that ethicists are not for limiting, but for harnessing opportunities. Because I, as an ethicist, would not say it is not allowed and it's a very problematic thing to use all the health data. I would say it is an obligation to use health data, because this can be so beneficial for improving the healthcare system and healthcare delivery, and bringing together health data from every area of life, because health data are not only medical data. It's all about your social life, about your work, about your mental health, like the WHO definition also stresses. And using health data means building up a learning healthcare system. You take all the everyday health data, of course they have to be qualitative, validated, and so on. I know, of course, of all these problems, but you have to take these data from everyday healthcare, using them in research, and bringing the research results immediately back to everyday practice. Let's say, for example, about clinical, via clinical decision support systems or something like that. All this is very tricky and very complicated, but this circle of learning healthcare systems actually is an ethical claim, yeah, to have it for the sake of individuals and societies. My Center of Life Ethics and my former center, CREUZET Cologne, we did an expertise on tech giants in healthcare. You immediately find it in the internet. It's available in English now as well. And we scrutinized 16 tech giants all over the world, the Asian ones as well, and listed what they do for healthcare, either for the patients, devices, processes, and so on, then for healthcare providers, for the systems level, so building insurance companies for health, for example, or building up hospitals for employers of their companies, and also about the economy, getting startups, building spin-offs, and so on. And of course, there is a very huge dynamic in there. And tech giants are those who have the money and the competences to do really a lot, where universities are, can difficult live up to that standard. So actually, there's a lot of cooperation with tech giants as well. But do we want tech giants to really define this landscape, or do we want to have a more diverse spectrum of those who are involved? Let's go to who is responsible when using technologies for health. Tech giants who build it, tech giants who use it, tech giants who deploy them and get a lot of money with them. Let's take the example of clinical decision support systems, and just to say that data is not yet enough. We have to come from a data view to a systems view as well by taking different steps. So starting by data, you have to put them into a context. The same data can mean something completely different in another context. And then you have a kind of information. But that's also not enough. This information has to get meaning. And this makes knowledge out of information. And if you then assess this knowledge, because if you know that you have an 80% risk to get Alzheimer's dementia in the next 5 years of your life, this is your knowledge, but you don't really know what that means for you. What does that mean for your insurance companies, for your family planning, for your employment, for you perceiving yourself? So if you know that and you forget your keys in the morning, now you say, well, this night was not so well, I forgot my keys. But then if you know that, you say, well, this is the beginning of my dementia. That's something completely different on your self-image. So you have to assess it to get insight, and then you have to make a purpose of it so that you can take a decision. So do you really want to know whether you have Alzheimer's dementia via biomarker assessment or so in the next 5 years? I'd prefer to think before the diagnosis about that and not to realize afterwards that this is impacting my life so much that I actually had preferred not to know it. But also it can the other way around. It can be that you say it's great to know that because then in my autonomy, I can shape the rest of my life according to that. I can plan beforehand and so on. So there is not this one ethical solution for this problem, but the ethical claim is have the insights and all the knowledge and the counseling before this diagnosis and this application of technologies. So how is a technology for health shaped? I think before having or when having the idea and when going into the development process and an interdisciplinary one, you have to ask yourself a lot of questions. And it could be a good idea to have an ethicist as a part of your team. So what is the purpose of the technology? Is it safe? Can the user take control of it in meaningful respects? Is a discriminatory bias possible, for example, if you have an app developed for detecting melanoma, but you also use data from people with light skin, and then you use it for people with a dark skin, and it doesn't work. So this is a discriminatory bias, a very easy one. Are development and use of the technology sustainable? So AI is not sustainable at all in ecological terms. What materials do you need for building your devices? Getting the minerals and the rare earths from Africa, for example. People living there, mining those things under horrible circumstances and with huge health problems. And then devices are built up in the so-called Western world, and all the garbage goes back to Africa or other countries of the global south. Is that really a sustainable way of building our futures and our technologies? So there are, meanwhile, possibilities to build AI in a more sustainable way, but it is about the amount of data, the way you process it, and so on. What happens to the data? Can there be harmful consequences for the patient, the user, or society? Think of whole genome sequencing that was mentioned before. What about incidental findings? So all of a sudden, the patient learns that, or the research participant learns that she has a BRCA1 mutation. Do you tell her that? Because you can, this is an actionable result, you can do something against it. What about all the other genetic mutations that can be found for 100 euros? But the patient doesn't want to know that. So you have to know before which kinds of results you then tell the patient, or which ones you don't tell him, or even which ones you don't examine and assess, because there's no result coming out of that. And how are responsibilities, accountability, and liability allocated? Because when you have a clinical decision support system, then the doctor can say, well, this is recommending this drug, but the doctor says, well, yeah, that was the algorithmic system, then I take it. But the patient actually would have profited from another drug because of his individual circumstances. So decisions are delegated to algorithmic systems quite lightly. And this has to be scrutinized because doctors, together with their patients, have to decide what they do. So that's now my final information I want to convey. Ethics by design in technologies for health is about the purpose of the technology, about the materials you use, about the functioning and use, the production and the delivery, and all the governance systems around that. And on the right side, you see all these ethical principles or democratic prerequisites that are impacted by that, that play a role in deciding about the purpose, materials, and so on. Futures or the technologies for health are part of the social imaginaries, and here's Sheila Jasanoff from Harvard University, the Nestor of Science and Technology Studies. We have a social imaginary of what health is. And those who build technologies for health have a major role on defining these social imaginaries together with societies. These imaginaries frame and shape how we go forward in building technologies for the futures of our societies and of the health of our societies. And they are collectively held, institutionally stabilized, and publicly performed visions of desirable futures, animated by shared understandings of forms of social life and social order, attainable through and supportive of advances in science and technology. And I cite a French expert I met on a conference on AI for Good. He said this wonderful sentence: the more machines can do, the more human we must become. Thank you very much for your attention.
My name is Patrick COUVREUR, and I will start with the first presentation on advanced nanomedicine for the treatment of severe disease. As you can see on this first slide, you know that the administration of a drug as a conventional chemotherapy is characterized by a poor diffusion through biological barriers, which means that a lot of drugs have a low bioavailability. Also, those compounds, for instance, arising from the biotech, are characterized also by a fast metabolization and degradation after administration. And some other compounds, as you can see on this cartoon, are characterized by unspecific tissue and cell biodistribution, leading, of course, to low activity, even worse, to toxicity. And finally, some drugs in the field of the infectious disease or oncology, of course, are inducing resistance to the treatment. So the idea, as you can see on the right side of this cartoon, is that you can encapsulate this drug into nanotechnologies with a size in between a few 10 to a few hundred of nanometers. And if the encapsulation is well done, well, you can see that you will be able to protect the drug from the fast metabolization. This is true, for instance, for the messenger RNAs. It's just one example. Also, as you can see on this cartoon, it is possible also to decorate those nanoparticles with specific ligands, as you can see in green. And this will allow a better tissue and cell targeting towards the diseased cells and tissues. And also, because a drug will be delivered in another way to the cells, you will be able, using nanotechnologies, to overcome some mechanism of resistance. And finally, as you can see here also, in the same nanotechnology, you can combine both therapeutic functionality and also imaging functionality. And I will try through my presentation to illustrate those different points through two advanced technologies that we have developed in my group. The first one is the so-called squalenolation platform, which is the squalene prodrug approach leading to nanoparticles, and also the possibility to use the metal oxide frameworks to design nanoparticles for a better drug delivery and biodistribution. So I will start with the squalenolation. You know that typically the encapsulation in current nanocarriers, I mean, for instance, in liposomes, in polymer nanoparticles, in polymer micelles, etc., results always from physical encapsulation processes. This means entrapment, adsorption, encapsulation, etc. And this results, as you can see here, to a poor drug loading. This means only a few % of the drug transporter versus the transporter material, either lipids or polymers. And this is the first limitation. The second limitation is that because you have a fraction of the drug which is rather adsorbed at the surface of the nanotechnologies than really entrapped into the core, this results in an uncontrolled burst release of the drug after intravenous administration. And this is a reason why we have moved recently from the physical to the chemical encapsulation paradigm, doing, in fact, chemistry instead of physics. And to do that, we have used the squalene. You know that the squalene is a natural and biocompatible lipid. It is in our body the precursor of the biosynthesis of the cholesterol. And as you can see in the right side of the cartoon, this natural lipid will adopt a molecular folded conformation into water. And we have taken advantage of this compact conformation of the squalene to link the squalene to several drugs, to several compounds, by using, as you can see over there, a biocleavable linker. And by supramolecular chemistry, if you are putting this bioconjugate into water, the molecule will self-assemble, leading to nanoparticles with a size of around 100 nanometers, with a much higher drug loading than after encapsulation by using physical processes, because, of course, as you can see here in red, each molecule of squalene will be able to transport one molecule, at least one molecule of the drug. And also because you have a chemical link between the squalene in red and the drug in green on this cartoon, you will have an absence of uncontrolled burst release. And also, if you are a good chemist, you can design a biocleavable linker which will be responsive to an endogenous stimulus located at the level of the target tissues or cells, and this will allow the release of the drug in the diseased area of the body. And as I told you, this represents really a platform, because as you can see here, we have linked the squalene to small anti-cancer drugs, leading to nanoparticles with a much higher drug loading, over 50%, for instance, with doxorubicin. Also, there are some applications in neuroscience by linking the squalene with some neuropeptides for pain alleviation. I will speak about that. We have linked also the squalene with antiviral compounds and antibiotics to allow the treatment of intracellular resistant infectious disease, but I will not have the time to speak about that today. You can link also, and this is really amazing, the squalene, which is a small lipidic molecule, to RNA, to small interfering RNA, a highly hydrophilic molecule, and in that case you get even nanoparticles with a drug loading over 90%. And finally, it is also possible in the same nanotechnology to combine two drugs, for instance, an anti-inflammatory drug and an antioxidative drug, to fight the uncontrolled inflammatory process associated with the cytokine storm. So I will show you, I will illustrate that precisely, first of all, with some application in cancer, where the advantage of using those squalene-based nanoparticles is that you will get long circulating properties, as I will show you, with an increased anti-cancer efficacy and a reduced toxicity. Just one example to illustrate that. We have linked the squalene on the, you can see the chemistry on the left part. In red, you have the structure of the squalene, the biocleavable link, and the structure on the left of the doxorubicin. And when you are putting this squalene doxorubicin bioconjugate into water, right, you can see that you get elongated nanoparticles. And amazingly, as you can see there, after intravenous administration in green, you get long circulating nanoparticles providing much higher concentration of doxorubicin squalene into the bloodstream than what happened, as you can see here, after the administration in red of the doxorubicin as a free drug. And this is explained, I am sorry, but the pointer is not working. This is explained by the cartoon over there, that the nanoparticle will elongate after intravenous administration through the blood flow streamlines. And this means that those nanoparticles will be invisible for the capture by the macrophages of the liver and the spleen. And this will completely modify the biodistribution of the doxorubicin when linked with the nanoparticles. You can see that you will decrease dramatically the concentration at the level of the cardiac tissue, and you know that the limiting toxicity of doxorubicin is the cardiac toxicity. And on the other hand, right below, you can see that after 24 hours, we will increase dramatically the concentration of doxorubicin at the level of the cancer nodule. And the result of that is that you will have an increased pharmacological activity on the left side of the slide. We have tested that in a human pancreatic preclinical model and also a murine lung cancer. You can see with the black line the tumor evolution. You can see in red that after treatment with doxorubicin as a free drug, you have really a resistance to the treatment because you don't inhibit the tumor growth. And you can see in green that after administration of the doxorubicin squalene nanoparticles, well, in that case, you completely inhibited the tumor growth. And on the other hand, on the right side of the slide, you will decrease the toxicity, the cardiac toxicity. And we have tested that by using a rat hypertensive model. If you are administering 1 milligram per kilo body weight of doxorubicin as a free drug intravenously once a week during 11 weeks, you can see very clearly below that in red you will have a huge increase over the treatment of the troponin level. And this is clearly a marker of the cardiac toxicity of doxorubicin. On the other hand, if you are looking at the end of the experiment for some biopsies of the cardiac tissue, you can see that there was in C a very, very big vacuolization of the cardiomyocytes, which is another illustration of the cardiac toxicity of doxorubicin for those rats who survived to the chemotherapy. And now, after administration of the doxorubicin squalene nanoparticles at the double of the dose and even the triple of the dose, you can see below very clearly that the troponin level are remaining completely normal and also in the top the biopsies. And this means that if you are increasing the pharmacological activity and decreasing the toxicity, of course, you will increase the therapeutic index. Now the question is, well, how are those nanoparticles able to target the tumor cells? What we did was to investigate the interaction of those squalene-based nanoparticles with the blood after intravenous administration, either in vitro with human blood or in vivo after administration to rodents. We did that with the squalene gemcitabine, with the squalene doxorubicin, the squalene cisplatin, with different anti-cancer compounds. And for instance, with the squalene gemcitabine, which are giving spherical nanoparticles, you can see that after incubation with human blood or injection to rats, there was a very, and this was really amazingly, a very rapid degradation of those nanoparticles, disaggregation of those nanoparticles, with the release, for instance, of the gemcitabine squalene as individual molecules. And because you can see the chemistry there in blue, you have this squalene part of the molecule, which is a precursor of the cholesterol biosynthesis. This bioconjugate has a very strong affinity for the cholesterol-rich lipoprotein, including the LDL into the humans and the HDL into the rodents. And you know that at the level of the cancer cells, as you can see on the right, you have the hyperexpression of the LDL receptors, because, of course, the cancer cells need to feed with lipids to make membrane and to multiplicate. And so this was a new approach of the drug targeting using, in fact, the LDL or the HDL as an indirect nanocarriers for the tumor targeting. Now I would like to move to a second illustration in the field of the neuroscience, showing you that it is possible also to treat some neurological disorders with those nanoparticles without the need to pass through the blood-brain barrier. One example is the adenosine. You know that adenosine has a very important role in energetic metabolism. It is a neuromodulator, and normally this molecule should have very strong pharmacological activity in neurological disorders, like, for instance, a stroke. But unfortunately, it is not the case, just because, as you can see, the adenosine is very, very rapidly metabolized after intravenous injection, and the half-life is only 10 seconds, which is really a fast degradation. And this is due, in fact, and you can see that on the right side of the cartoon, we have linked in green the squalene to the amino group of the adenosine to protect this amino group from the deamination by the blood deaminases. And this bioconjugate is giving spherical nanoparticles this time, and we have tested those adenosine squalene nanoparticles for the treatment of the brain ischemia. And to do that, we have used a classical model where you are inserting a filament in the right carotid artery of a mouse, and you will induce, as you can see here on the right, an infarct volume, very clearly visible. And if you are administering adenosine as a free drug, comparatively to the vehicle treatment, you can see that there was no decrease of the infarct volume. And if you are administering the squalene adenosine nanoparticles either pre-ischemia or post-ischemia, or even after 24 permanent occlusion, on the right you can see the very strong diminution of the ischemic volume. And this represents, of course, a neuroprotection pharmacological activity. Again, we investigated what is the mechanism behind that. And we investigated the biodistributions of those nanoparticles after intravenous administration, and we observed, amazingly, that those nanoparticles didn't translocate through the blood-brain barrier and were unable to reach the brain tissue. And on the contrary, those nanoparticles remained into the blood circulating with long circulating properties, representing really a sort of reservoir of intact adenosine into the blood circulation. And you can see on the right that in fact, in the brain microcirculating along the vessels, you have the A2B receptors, which are adenosine receptors. And because the adenosine into the nanoparticles is released very slowly as an intact molecule interacting with those receptors, you will induce a vessel relaxation, a decrease of the number of ischemic capillaries, as you can see here on the right of this cartoon, less vessels which are clogged with platelets and erythrocytes. This allowed a better reperfusion and the neuroprotection that we have observed without passing through the blood-brain barrier, but by interacting with peripheral receptors. And we have continued along this line of reasoning to treat pain, because of course you know that pain alleviation represents a very important medical challenge. You know that classically the treatment of severe pain used morphine, morphine derivatives, opioid derivatives more generally, but all those molecules are inducing first tolerance and then addiction, and finally also pulmonary depression. And these represent a national crisis in the USA, also in Europe, with tramadol, for instance. And an option, an interesting option, is to use endorphin, enkephalin, endogenous neuropeptides. because those neuropeptides are not inducing tolerance and addiction because working on delta opioid receptors rather than on mu opioid receptors. But the problem, once again, is that those molecules are very, very rapidly metabolized. And so what we did was to link the squalene to this to the leuenkephalin, which is one neuropeptide, as you know, and we used, as you can see here, different biocleavable linker to be able to modulate the release of the neuropeptides in the blood circulation. And when you are putting those bioconjugates into water, you get nanoparticles, as you can see here, which are spherical and with a size in between 70 and 100 nanometers. And on the right side here, we have tested the activity of those nanoparticles to a model of rat where we have induced on the right paw a very strong inflammatory process associated with pain. And the left paw is a control because it is a healthy paw. And you can see that after injecting the leuenkephalin squalene nanoparticles in blue, we obtained a long-lasting pain alleviation. And in black, after administration of the peptides as a free molecule, of course, there was absolutely no antinociceptive effect. Now, we tried once again to understand what was the mechanism behind that, and we did some biodistribution studies by labeling those nanoparticles with a fluorescent dye. And you can see on the left that after intravenous administration, over the time, those nanoparticles concentrated on the right paw, where occurs the inflammatory process with the pain, and not on the left paw, which is the control paw. Now, this is explainable because, as you can see, when you have an inflammatory process, you have an arrival of macrophages, liberation of cytokine, and you have a leaky vasculature allowing those nanoparticles to translocate exactly where you have the pain with the inflammatory process. And we investigated also the penetration of those nanoparticles into the brain. And you can see on the right in white that those nanoparticles once again were unable to reach the brain. And this explained that those nanoparticles were able to induce pain alleviation by playing the game on the peripheral inflamed tissue and opioid receptors, but without passing through the blood-brain barrier. Therefore, they allowed a suppression of any tolerance and addiction. Now, I would like to finish my presentation just with a quick other example by using metal oxide frameworks to design nanoparticles, and we started this research with my colleague Gérard CREUZET, who unfortunately passed over, and we continued that with Christian. You know that metal oxide frameworks are in fact a cluster of iron oxides which are complexed with different diacids. And depending on the molecular weight of the diacids and the flexibility of those diacids, you can design pores, as you can see here, which will have exactly the size of the hosted molecule that you would like to encapsulate. And we were very interested to use busulfan because busulfan is used in pediatric oncology at high dose, and unfortunately this molecule is inducing dramatic veno-occlusive disease of the liver because of the crystallization process. And we have tried to encapsulate busulfan in liposomes in nanoparticles. It's not working because always you have a crystallization process. And by encapsulating the busulfan into those nanomorphs, each molecule of busulfan is in a sort of cage of pore separated from the night-boring molecule, and this hinders the crystallization to occur. And so we could load around 20% of busulfan in those nanomorphs. More interestingly is the fact that because of the iron oxide cluster, you have a decrease of the T2 signal at the magnetic resonance imaging. And so after intravenous injection, you can follow the nanoparticles into the body and look, for instance, if they are able to reach the tumor and to shrink the tumor or not. And this was the first time that we proposed the nanoteranostic concept. This means multifunctional nanocarriers combining both therapeutic functionality because of the drug encapsulation with diagnostic functionality because the imaging process. Now we observed also that those nanomorphs were pH sensitive but reversibly. Normally, those nanoparticles are prepared at an acidic pH with a size of around 100 nanometers. But you can see that after incubation with serum at a pH of 7.4, there was a microaggregation of those nanoparticles. But this microaggregation, as you can see here in the cartoon, is reversible, is disappearing very fast. And after intravenous administration, because of those microaggregates in the histology, you can see that you are able to target the lung tissue. And now, if you are encapsulating an anti-cancer compound like gemcitabine monophosphate in blue, you will deliver much higher concentration of this anti-cancer compound into the lungs comparatively to the free drug. And the result of that is that in a preclinical model like the Lewis lung carcinoma with those nanoparticles in blue, you will decrease dramatically the number of pulmonary metastases, which was not the case after administration of a free drug. So I am coming to my conclusion. I hope that I have convinced you that advanced nanomedicine, and this is just 2 examples, can protect the drug from the fast metabolization. This was the case with adenosine, with those neuropeptides for pain alleviation. That those nanoparticles can be functionalized to target the diseased area, for instance, in the field of the cancer, depending also to the shape of those nanoparticles, elongated or not, and that in the same nanoparticles you can combine various functionality, therapeutic functionality and imaging functionality, as it was the case in the nanomorph. And I would like to acknowledge all the people of my group who collaborated more or less to one or another example I have given to you. Thank you very much for your attention.
Thank you so much for welcoming me. I'm very grateful to be here and to be able to learn from all of you. So thanks so much, and really a pleasure to talk about the future of medicine and especially the role of artificial intelligence within this context. So I think like everything about artificial intelligence, it's really about how the disease can be cured. And when we say the disease can be cured, it's very important for us to try to choose about which disease we're talking about. In AI, we try to tackle a lot of different diseases. Oncology, cancer, is of course a real like low-hanging fruit. And you have to know that cancer is still a completely uncured disease. When you see like cholangiocarcinoma, where people were not dying from 10 years ago, or a brain tumor, glioblastoma, where pretty much everything is failing. So we really want to tackle diseases. But when you think about the angle of what you want to cure, you can also think about aging. Aging is probably a disease that is maybe more important than cancer. If you are 65 years old, just the fact of having a cancer cured or not might change your life expectancy of only 1.2 years. That's pretty much in median. So maybe, you know, we need to think about aging in a more global process, you know, and pathways. And finally, death is probably the best disease to actually try to cure, and we will agree on that. And so I think the paradigm has to change to be able to cure more diseases. It's not possible that 35 clinical trials out of 36 in brain tumor are negative. The pharma is even stopping to work on it. We need to change the paradigm and find a better convergence of the technologies. And so I think we need to find a new way to see with your eyes, and your eyes don't see as the computer eyes, what we call the computer vision. So how can we see differently? Can we see differently a disease? Can we see differently a patient? And do we have to change the way the system is built? And I think this is just the way to actually change the paradigm. And everything is about how you find biomarkers. Biomarkers are markers of a disease. It's usually a level of a blood count in your cell. A gene can be a marker of some response to treatment, any phenotype. But there is new ways to discover biomarkers that the AI is bringing along. And the way to do it is actually trying to predict from new modalities, such as images, a survival, a response to a treatment, a resistance. And when the prediction works, you have a good accuracy, you reverse engineer. We can also say you open the black box to try to understand what's the biology and what are the biomarkers. And we go, the computer vision sees differently from your human eye and can go above genes or level of blood cells. It can also find markers in the CT scan, an MRI, or biopsy. And the question knows that you find a biomarker. Imagine it's a little part of a biopsy. It's a combination of genes. What do you do with it? You still have the way to think that it's correlated to your outcome. This is how you did the work. You predicted an outcome, a phenotype. Sorry. Okay, sorry. Because I'm very tall. So, sorry. And so the question is, what do you do with this biomarker? The problem is they're still correlated. It's still what we call correlation. And you all know the paradigm. We say correlation is not causation. And actually, artificial intelligence, it's just a non-linear correlation. It doesn't explain way more than a classical biomarker. So how can we go to something more causal? How we can find a biomarker, a gene that will explain the disease. If you actually knock it out, you will cure a disease. You might cure aging, or you might even cure death. It's really about what we call causal inference in the AI space. And causal inference is trying to understand how you can find some things that are linked to causality, and how can you prove it. And this is a huge field of AI. It's trying to understand how you link a certain biomarker, or even better, a combination of biomarker, with more causality to a certain outcome. And it's really a big work, and today there is, we're not still able to go to causality, we can explain more phenotypes step by step. If you try to predict a survival, you might try first to try to predict a cell organization, you might try to predict a tissue organization, all the steps that Elias showed us before. And I think it's really a work step by step to go from good explanation of certain phenotypes to causality. And finally, when you find this type of biomarkers, how you include them in the workflow of a pharma, how does this become a new rationale for combination or a covariate to adjust your clinical trials? So I think this is really the three steps: finding biomarkers, accessing a lot of data of high quality, trying to find what makes sense in biology, opening the black box, and finally trying to understand how that will help a pharma workflow. And finally, you need to choose the modalities quite well. And this is the future of medicine. This is the future of artificial intelligence. And I'm sorry, I would have loved to show that it's CT scan or MRI, because we have pioneers of medical images, Professor ZERHOUNI, Professor Sirota. But we believe that there is something that hasn't been enough used, is the biopsy, the pathology. Here, a pathologist in the hospital would tell you, this is this type of cancer, this type of stage of grade, sorry. But he won't tell you that people will predict, he cannot predict that a patient will resist to a treatment or will die within 6 months. AI can. AI can give superpowers. AI can augment the way to see images, because they find new structure, new patterns. And everything is here, because when you have a cancer, all the information, even if the human eye don't see them, are in these slides. And I will show you an example after. But we think that pathology is the way to anchor all AI in everything's related to most like oncology and autoimmune disease, more than, there is more signal there than, other type of images such as CT scan or MRI. And why is it cool to have this biology into a pathology slide? Is because you can go further with a new technology called spatial transcriptomics. And this is really about how AI can apply to new technologies such as that. The cool thing about spatial biology is that you can actually find in one slide the level of genes, RNA level, as well as the tissue structure in one slide. And you can include the stroma that is linked to all the immunotherapy responses to the tumor itself. And what is even cooler is you can link one gene to a small structure of cell organization within one tile. So you can link genes to phenotype at the largest scale ever done in the past. And at Owkin, we're trying to create the largest database worldwide of spatial transcriptomics. Spatial biology is a revolution in the era of AI, linking tumor to the microenvironment, linking genes to their cell organization, tile by tile. Then the new way of seeing is a new way to see the patient and how we have to think about the patient itself. So we believe that patient data is the way to go to. And pretty much a lot of people are doing something else, which is using cell lines to find new treatments. There's a lot of companies in the US, it's called in vitro, recursion, you can name them. They're actually building huge robots to actually, plate cell lines at a high throughput scale, and without the high resolution. And then they test hundreds of compounds on it. And then they just analyze the images to try to find which is the best treatment to give. But the cell line is not a human body. It doesn't include the microenvironment. It has a huge, what we call, translational gap. So the question is how we reduce this translational gap to create real biology. And for us, the answer is what we call reverse translation. So the classical way to do treatments in the pharma, you find a cell line, a model, which is not a human model. Then you find something and you develop it by step, with always this translational gap that might not translate when you give it to human. So our idea is to start with patient data and to go backwards, what we call reverse translation. And why today reverse translation is not really working well, it's just because first, there is not enough data to be able to find new models. If you find the largest study cohort of glioblastoma, it's around 300 patients. Should be 300,000 patients. In the area of Google, which is our largest investor, I have to disclaim it, and it's possible to gather this type of data modalities. We need much larger cohorts. The second point is we need to be able to link the genes to the tissue and the organization. This is the importance of spatial transcriptomics, for an example. So with that, we really believe that we should be able to find better biology and better, breakthrough. And from there, we would go to understanding new subgroups of patients that resist or not to different treatments and match this subgroup of patients to treatments. This is the area of precision medicine that needs to happen. Precision medicine today, and I would just say a word about it at the end of the talk, does not really happen. It's just only a few people that have a certain gene that can be targeted and drugged. Otherwise, you're out of the precision medicine world and you have the same treatment than anyone else. And to get more data, that's really the idea. How do you do it? We believe that, in my company Owkin, but in general, that data should never leave hospitals. There's too many silos of research, competitive silos, research silos, because people don't want to share data. And sometimes they're right, because if you share data, when the data go on the cloud, well, there is a breach possibility. Let's see when the day where AWS will be breached and hacked. So data should stay within the hospital where they're produced, within the firewall. And we call this federated learning. We are the pioneer of this in the space of medicine. Google is doing the same for your mobile phone. Today, your analytics of Uber, whatever, they go in the cloud. In the future, the analytics, the little model, will analyze your data in your phone, and only the model will travel. But data never leaves what they produce. And that creates a lot of trust from patients, breaking silos of research and competition. It's a complicated technology, but we really believe that this is the only way to gather hundreds of thousands of patients and not only hundreds. And just an example of a project called Melody that we did with the European Union, we actually were able to link 10 large pharma that would never work together on their molecular libraries. They gave access up to half of the molecular libraries to actually train a model on small molecules binding. And this is just much better data than, like public data that are being used for AlphaFolds or else. But if you were asking these 10 large pharma, such as J&J, Novartis, they would never agree to aggregate. What about the clinical trials that failed in brain tumors? There are 36 clinical trials that are siloed in brain tumors and no one is using. Let's build a federated network to try to analyze things and make new insights. So the future for us is leave data where they're being produced, respecting patients' confidentiality and rights. And this is really for us just the way to build trust and sustainability. And another way to do it, that's just how you access data. And this is an example of a reverse translational approach. The slide is not readable, but we published this in Nature Medicine, is how you find a new treatment for mesothelioma. So mesothelioma is linked to asbestosis. It's a really bad cancer. You can die in less than a year. The pharma don't really care because the market is not great. But we need to find new treatments. How we did that? We gathered a 3,000 patients cohort through this federated learning approach. People were agreed to give access to the data. We predicted an outcome. We could produce survival month by month. Then we say, okay, we can produce from the pathology images. Seeing at the pathology slides, we can produce the survival. Why is that? Can we open the black box? And we were able to show that it was linked to the cell organization within the stroma that could make the production possible. We link this to the genes in this convergence of technologies and ideas and modalities. And now we have some genes candidates that are being developed with Sanofi to try to find new treatments in mesothelioma. But this is the idea of AI. You predict, the prediction works, yes, why? Finally, the system, and I'm finishing on that. It's really about how you need to think about the system. It's really the research, AI is being done in a research silo or a care silo. But if you only on the research side, you don't work with patients and physicians day to day, you overfit. You always find models that don't make sense, that are not really far away from what it's been done. And it's different from different modalities. If you take a cardiologist has been using the same items to predict a stroke or an infarct. You're male, you're smoking for the last 20 years with some accuracy around 0.7 AUC. The cardiologists, they would do anything to actually augment their prediction of who is going to do a stroke or an infarct, even if it's completely black box, even if you don't understand the model. The oncologists, they will never accept that. So it depends on the question. You need to be very close from the care and research should share data to advance medicine. And today it's really like going from a solution-based model to an unlimited network and possibilities, creating network effect between academics and pharma, between the pharma themselves, and being able to connect researcher to data science so they can both work together. AI is not an end, it's just a way to communicate. The idea would be that, for us, it's trying to recreate a high-throughput pharma in a way. The classical biotech system is single asset, 7 years, one drug, super high risk. You can lose all your money if it fails. We really believe in being more data-driven because there's still a lot of steps that are not data-driven. Coming from a phase 1-2 to a phase 3 is not data-driven. Toxicity assessment is not data-driven. We really want to go further and build this AI biotech step by step. At the end, what is the outcome? The best outcome will be a way to have new treatments, but also do better precision medicine. Just giving an example of something we developed, it's just being able to help pathologists predict the risk of relapse in breast cancer patients. We just got the approval. Today, if you're a patient, you want to be able to predict the relapse of your breast cancer. You need to pay a test called MammaPrint. It's $2,000. It takes some months. It's genomic-based. The future for what we've built is pathology-based. You just take a scan of the biopsy, take 1 minute, and we have a better accuracy. So it's free. Pretty much, it's rapid, and it brings the precision medicine to a new era of modalities, which is pathology. That means, of course, that the hospitals have to digitize their pathologies, the biopsies, and we are trying to help the hospitals to be up to date. French hospitals are super far from, and I'm so sad there is no one from the government anymore, because it's not the only thing that hospitals in France are far from. French system in medicine is really close from collapse. And I was a physician, and I'm really have a huge admiration for people that stay in the system. But one way to be able to get through the costs and the problematics will be to be able to embrace this new era of precision medicine. Finally, and the courtesy of In Silico, which is an amazing company in Hong Kong, is there a way to approach longevity differently? I have 3 slides anymore, and I'm done. Today, AI can predict your age based on the picture pretty much as accurately as your real age. A very, very small difference. The question is, why is that? Why a neural network can predict how old are you? What is the neural network seeing? Is it your eyebrow? Is it your nose? I don't know. No one knows. And I think just the way to try to understand that is trying to understand how, the biology and the biomarkers that I'm talking about are changing time by time, trying to predict the combination of the right markers, and they can be on different pictures. It can be a lot of modalities that try to converge to try to predict the biological age. And it's really, once again, the approach of AI, trying to predict an age from different modalities and trying to understand how we're able to do that. And let's go to the final concept, which is the concept of aging clocks. Today, we're trying to, In Silico and other companies are building aging clocks based on a lot of different data modalities, such as methylation, photos, epigenomic, as I say, markers of blood, anything. And they will be able to predict side by side what's the modalities that are creating aging and longevity. And longevity is not only about AI, it's also about, cryotherapy, that's making progress, how you can build new models of labs using AI. A lot of technologies, including physical technologies, need to converge. Finally, this is okay, but we still need to understand what's happening for a real human. And we're not there yet. We don't know the cycles of human liver's enzyme that will help us understand if the biomarkers we are finding are just a natural evolution or not. And there is a lot of work to do there as well. But this is the field of AI, and I think it's pretty much a big part, as quantum computers will be, as nanoparticles will be, as other platforms such as mRNA are, a big part of the future of medicine. Thank you.
I want to thank Patrick for inviting me to speak at this meeting. It's a real honor to be here with you. So I am work for a for-profit company, so I need to show you this. You've seen it. Okay, so Over the past 100 years, new medicines have transformed our lives. Many people don't realize that penicillin and the sulfa drugs were only discovered less than 100 years ago. So it really has been a recent revolution. And of course, in the time since then, there have been many other small molecules, many of which many of us take on a daily basis. But other medicines that have been truly transformative obviously have been vaccines. Traditionally, they were made either by killed or live viruses. And then in more recent years, a class of medicines has emerged, which are the biologics or proteins. Of course, the first of those was discovered just a little over 100 years ago at the University of Toronto. They've been celebrated last year, the discovery of insulin. But such molecules as erythropoietin for growing new blood cells, monoclonal antibodies really have addressed many disorders now, and I'm showing factor VIII, which is a clotting factor. Now, with the rise of these proteins as medicines, as discussed by our first keynote speaker today, there are a number of issues. One of them is that they require just very large, dedicated industrial infrastructures. And those infrastructure is not readily changed between different medicines. Now, another issue is that if you're making the proteins in Chinese hamster cells and then purifying them, each is unique. So each needs to be, the research has to be done on how to formulate it, how to stabilize it, and then the ADME-tox properties of each is unique. So it takes quite a long time to develop these medicines. A third thing is that proteins produced in non-human cell lines often display different post-translational modifications than human cells, although I do have to say that the CHO cells that are generally used have a glycosylation patterns very similar to humans. But the fourth limitation is that biologics are injected into the body as proteins, and so they must necessarily work outside of cells. A few are taken up into cells, into lysosomes, but by and large they work in biological fluids and outside of cells. But the problem with that is that over 2/3 of the human proteome are actually intracellular or transmembrane proteins, and so are not secreted proteins, and so are not able to be used as biologics. So the promise of mRNA medicines is that instead of delivering the protein, we deliver the instructions to make the protein and then have the human body make its own protein as medicine. Now, I just want to remind the audience, especially those of you who are not biologists, that mRNA is a essential component of all living organisms, and each of the more than 30 trillion cells in your body contains between 200 and 300,000 mRNA molecules. So you have a lot of mRNA in your system. mRNA is not something that is foreign to the human body. But one of the great things about mRNA and protein is they really have the aspects of classical medicines. So let's take, for example, your circadian clock. So your circadian clock is driven by a set of proteins called the clock proteins. And they appear and disappear every 24-hour cycles with incredible regularity your entire life. They are driven, the production of these proteins is driven by the appearance and disappearance of the mRNAs that encode the proteins. And so this mRNA, every day, you're getting a dose of it and then it's going away, and each day after that it's having the same effect by driving the production of the clock proteins. So mRNAs have all three attributes of classic medicines. They have limited duration of effect because they're temporary. They have predictable dose-dependent effects because the amount of protein produced is a function of how much mRNA is present, and they're redosable. So in theory, they're redosable. Inside your body, you're getting the doses of mRNA over and over again. when we made mRNAs outside the body, we had to figure out how to do the redosing, which we have done now. So what that means is that if one could develop a platform for making mRNA medicines, that we could address many unmet medical needs, such as, for example, inborn metabolic errors where the protein that's missing is an intramitochondrial protein. That just really couldn't be done with biologics. Also, because what you're changing each time is simply the sequence of the mRNA. Once you've figured out how to make RNA, how to formulate it, how to get it to the target tissue, you can easily make a new medicine very quickly and not have to go through all of the toxicity experiments that you need normally to develop new drugs. So here's the thing. In traditional medicines, the timelines have been long and incredibly astronomical. And typically and I'm looking here mostly at small molecules, but this is also true of biologics, and our first speaker spoke to this today, that there is an incredible failure rate for new medicines, and that's why it takes so long and it costs a huge amount of money. And yet, when COVID arose, we were able to make the vaccine and actually get it ready for clinical testing, clinical trials within 45 days. And I can tell you that it still gives me shivers to say this, but we had been working already with the NIH on coronaviruses prior to the arrival of COVID. And so on January 11th, 2020, when the sequence of SARS-CoV-2 was posted to a public web server in China, it took us 2 days. We started working on it immediately. It took us 2 days to discuss with our colleagues at NIH of exactly what protein to make, so what version of the spike protein to make. Then we had put in tremendous amount of effort into learning how to make mRNA medicines and mRNA vaccines for over 10 years. I'll tell you about that in a minute. It only took us 1 hour to design that mRNA. Now here's the amazing thing. That mRNA that we designed in 1 hour, for those of you who got the Moderna vaccine, is the mRNA that went in your arm. So we did it once and it worked. This is a new age of medicine. The ability to make nucleic acid medicines, and there are many other nucleic acid medicines that are coming besides messenger RNA, but it is really changing how quickly we can make safe and effective medicines. So, and of course that vaccine went on to show very high levels of neutralizing antibodies and very good clinical efficacy. And the mRNA vaccines have really proven to be the most efficacious of the vaccines. Now, we're not just a vaccine company. Moderna makes many other medicines. And so this is a picture of our current pipeline. And the pipeline includes many different prophylactic vaccines for infectious diseases, both respiratory, latent viruses, and possible epidemic or pandemic diseases. We're making cancer vaccines. We are making intratumoral immuno-oncology products where we're injecting the mRNA directly into tumors to get the immune system to attack the tumor. We've been working on regenerative therapeutics to use mRNA to help the body grow new blood vessels, for example, in areas where it needs it. We also have systemic therapeutics. We recently announced that we have developed a inhalable form of mRNA that could be used to treat cystic fibrosis. And then finally, at the bottom there, we are working very hard on many of these inborn metabolic disorders, and we've recently announced our first clinical data with propionic acidemia, which is the one second from the left there, where we have patients who've now been redosing with those mRNAs for over a year, and they're staying on treatment. And so this really does work. So the questions that I just wanted to talk about is, well, what enabled us to develop Spikevax so quickly? What enabled us to build out such a large pipeline so quickly? And then more broadly, why is Massachusetts such a hotbed for biotech? And what are the essential ingredients for a biotech innovation economy? So I want to talk about the second two questions first. So one of the big problems with developing new drugs is this so-called valley of death, where you have results and basic years and years of fundamental research that happens in academia to understand the system, but then to translate that into a viable technology that can be used by industry, so many things just are preventing that. Now, the solutions, of course, are long-term capital investment, a willingness to accept risk, partnerships between academic and industry, and then really making it easier for academics to partner with industry. Now, in the United States, we have a very good partnership between government, universities, and industry, all playing their parts to do the early stage research in academics, for the government to help with that valley of death, and then industry to develop the discoveries into new medicines. Now, another thing that we have in Massachusetts is we have an incredible concentration in a very small area of world-class universities that have very strong research programs. And I'm sad to say my home institution is the University of Massachusetts Medical School, which is in Worcester, which is just outside of the area, but it's also part of this industry. The other thing that I think has made the biotech industry so strong in the US is the number of healthcare venture capitalists. So this was in 2020. And what I really want you to look at there is the headquarter country and the flags. You can see that almost all of the most active venture capital firms are in the US. And that really, having those venture capital firms that are willing to invest money into early stage technologies is just crucial. Another thing that we have in Massachusetts is the Massachusetts Biotechnology Council, or MassBio. This is an industry organization where all of the biotechnology companies come together and work together to solve particular problems. We now have over 750 biotech companies in the Boston area. It's really kind of crazy. One of the major problems that we're having right now is talent. We are cannibalizing each other for talent. So one of the things that I'm personally working on is to increase the diversity of people coming into our workforce so that we can take advantage of all of the talent that's out there. Now, one of the things that you might have noticed if you look very closely, the venture capital firm that Moderna was founded under was not on that one slide, but I want to just talk for a minute about flagship pioneering, which was founded by Noubar Afeyan. Noubar's founding principle is to ask the question, what if? So to take very early stage things that normally are just in the university and say, well, what if it does work? Then what could we do with it? And so he has created an incubator for companies where academics, like myself previously, I was a professor for 23 years, academics generally don't know how to create companies. They need help. They need the infrastructure of a CEO. They need business people. And so what Flagship Pioneering does is to create early stage proto companies and explore in very small groups of people, can we get a do that killer experiment that actually shows that this technology is going to work. And then many of those proto companies fail. So that's a very risky proposition, but they're not investing a tremendous amount of money in it. And then some of them actually succeed and go on to found new companies and ultimately growth companies. And here is the current flagship pioneering ecosystem. You can see Moderna is one of many, many companies that came out of the flagship system. And so my message to you is about this is, if you're going to create a biotech economy here in France, you know, it really takes a partnership between the academics, the government, and venture capitalists who are willing to take on risks and you know, allow ideas to flourish. So in terms of Moderna, we actually had invested in our platform technologies for over 10 years before COVID showed up. So we didn't just come out of nowhere. We had been training and building and perfecting things for over 10 years. I'm running a little short on time, I think, so I'm just going to go through this very quickly. But this is the entire pathway from idea to mRNA medicine for an mRNA medicine. It starts with an idea for a new therapeutic protein. That then dictates an amino acid sequence. That sequence then we can convert back into an mRNA sequence. That mRNA sequence then is sent over to, this is all done in Cambridge for Cambridge, Massachusetts for Moderna, sent to our manufacturing facility in Norwood digitally. They create a DNA template. We then use that DNA template to transcribe the RNA, which is then purified, mixed with lipids to make lipid nanoparticles, and then that is fill finished and put in the vial. I'm not going to go through this. I'm going to whip through here for a second. One of the things that we have to do with RNA is you cannot give RNA generally as a naked molecule or mRNA as a naked molecule, because it faces many barriers, many biological barriers. One of them are the circulatory and lymphatic systems, and then of course cells don't naturally take up mRNA. Another problem with mRNA is that, you know, our bodies see mRNA as food. We're eating it all the time. When you eat lunch today, to think about it, you're going to be eating proteins, but you're going to be eating a lot of RNA too, and your body digests it. So there are many digestive enzymes that we have to get around. RNA by itself does not have appropriate tissue tropism, and it cannot get across the cell membrane. And so, as described by our first speaker, we formulate our mRNA in lipid nanoparticles, which consist of our messenger RNA and an ionizable lipid. This is a positively charged lipid that binds to the mRNA. And then a sterol and a phospholipid that form a lipid bilayer around that core, and then a PEG lipid on the surface that keeps the lipid nanoparticles from aggregating when they're in the vial. And what you can see on the bottom right-hand corner is a picture of, a cryo-EM picture of Spikevax. Now, I just wanted to remind people that these lipid nanoparticles are very much akin to the normal lipid transport complexes that your body uses anyway to transport fats around the body. And so on this scale, lipid nanoparticles are somewhere between VLDL particles and kilomicrons. But we can engineer them to have different tropisms by engineering their surface properties. And at present, we have 5 different proprietary lipid nanoparticles that we're using, 4 of them already in the clinic, and 1 of them hopefully will enter the clinic in this coming year. Now, finally, I wanted to say something about manufacturing. So if you remember, I talked about the huge investment in industrial infrastructures required for biologics. In comparison, the equipment used to make mRNA medicines is quite diminutive. In fact, our manufacturing facilities consist of large empty ballrooms, and we can then wheel in the equipment as we need it. It's that small. You can see it there next to a person. And all of the manufacturing is done in disposable plastic bags. So it's very rapid, and one facility can make many different products. The other thing I note is that in the making of mRNA medicines, there is no mammalian cell culture at all. It is purely DNA mixed with enzymes, nucleotides, and buffers. It's an enzymatic process. So it's much simpler, much faster, which is part of the reason that we could make it so quickly when we did. In terms of taking risks, Moderna started building out in 2016, and then we were fully operational by the middle of 2019, our own clinical manufacturing facility. This was considered at the time very risky because most biotech companies don't build out their own manufacturing facilities until they have something that they think is going to be commercial. Prior to COVID, we did not think we were going to be commercial until 2025. But we felt it very important to have control over our manufacturing process so that we could learn from that. And so because of all of the work that we had put in over 10 years to create mRNA medicines, when at the end of 2019, November, December 2019, we had, as I said, had already been working with NIH and BARDA for some time on coronaviruses in particular, and we were discussing with them, should we do a phase 1 clinical trial, should we do a dress rehearsal in case a pandemic shows up. And so then what happens? I'm just going to flip through these very quickly. We started working, as I said, on that vaccine the day it was posted to social media, and the first case in the US wasn't until the 20th. So that is the story of the coronavirus vaccine, but we think that this technology obviously can be used to make many more vaccines. And I note that there are over 225 different viruses known to infect humans throughout the world, but we only have about 25 vaccines available, so we really should be expecting more to make more vaccines. And so to do this, we are building out manufacturing capabilities worldwide. This slide is actually already a little out of date because there's some other ones that should be on there. But the last thing I just wanted to say is that there is no way that we can do all of the research necessary to make new vaccines in-house. And so we have announced the mRNA Access Program. This is a program that allows researchers at partnering institutions to take advantage of our ability to make really high quality mRNA and formulate it. And so, and researchers who are part of this program will have access to all of our different technologies, from designing the mRNA to production. And so I just want to give the website address here, because really the goal is to make this technology available to all academicians across the world. And with that, I want to obviously thank the people that I work with. I wish everybody could have seen how dedicated and hard the folks within Moderna worked during the COVID crisis. There were some people who did not take a day off for over a year. It was incredible. This was the company in 2018. This was the company last week. We are now 4,000 people. We've been growing at an incredible rate. And finally, I just want to thank you. Thank you.
Okay, so hello everybody. My name is Patrick Johnson. As I already said, I'm a head of research in Dassault Systèmes, and I'm very happy to show you in this little presentation what we are doing in terms of modeling, simulation, and big data for healthcare. And pretty much like in the past, what we have been, I hope, catalyzing in adjacent industries, where most of the things were done on paper and then gradually evolved to digitalized paper and then represented abstractions and models like a digital mock-up of a plane. This is happening right now in healthcare, and we are heavily investing in that, and I would like to show you this. The way we see that happening today around the world, and not only in my company's activities, is basically threefold. A lot of activities in terms of generating, Victor this morning talked about generative modeling, generating organ from multimodal data at the cell level, at the tissue level, at the organ level, at the body level, at the cohort and population level. But we also invest in virtualization of hospitals and hospital systems, and even more so in scaling up at the region or nation level, providing political, economical, and sanitarian sort of cockpits, if you will, to conduct a medical policy. We were used, for example, during the first 2 waves in COVID in France, trying to, at the same time, prevent the propagation of the virus with some lockdowns recommendation, but at the same time, trying to still keep some activity level from the economical standpoint. And those are, of course, antagonism that we had to optimize at the same time. There is a paper you will have, of course, on the CAETS website, the presentation. There is a famous paper with all the examples that I will show. Trying to do a virtual twin of a body is a daunting task, and maybe I will be retired, maybe dead, when this is totally achieved. But back in, let's say, 40 years ago, people said exactly the same about aircraft, about cars, about boats. And we tend to think in terms of multidiscipline and multi-scale. As Elias showed this morning, this is about complexity, and this is about precision at once. And so, of course, we need to gather, at the same time, a lot of knowledge and know-how in every discipline related to physiological complex systems, cardiology, neurology, oncology, and so forth, but also multi-scale it. It's not only at the organ level, it's about chemistry, it's about biochemistry, it is about medicine, it is about metabolism, physiology, in an integrative way. And the bet that we have been taking is that, in fact, pretty much like in adjacent industries again, people were representing aircraft with blueprints and drawings, and believe it or not, they had to do thousands of blueprints and drawings to have that precise, accurate representation of a plane. This is how it was done before. And now they completely shift gears, and they are all relying, the manufacturers, on the digital representation of the plane. We believe that we are going to supplement gradually the DMP, the health record that we all know, potentially, gradually, and then ultimately that the best integrative abstraction of health data in our daily life, not only when we are sick, is some sort of integrative model, a twin. And we are developing a platform for practitioners and patients for that. Some example, and I will show you some example of research that we are conducting with academical partners and also with industry. And you see some example here. We have an example in cardiology, of course, what we call our Living Heart program, and I will show you 3 aspects of that. We have in neurology, in lung, respiratory, in microbiota, which is a very hot topic right now, in cell and skin, and in cancerology. But we don't do models for the sake of the model and, let's say, the technological and scientific interest. Always, we try to generate models associated to a medical practice challenge. It is always driven by either a status quo, something that people do a certain way, with some limitations, with some constraints, and we try with physicians, with practitioners in the hospitals, in the block, to use models to change and transform their practices. So it's a very long journey because it's medical practices by medical practices, coded practices by coded practices, but on and on. Our bet, and this is why I'm getting up every morning, is to be able to gather all the industrial assets to provide ultimately an industrial platform that will not provide those twins case by case like we do like we see in the literature today, but something that would, let's say, cover 80% of the current needs. Of course, per organ, per therapeutical disease, therapeutical area or disease. So let's take an example and start with cardio. I have three examples for cardio. I'll launch the video and let you know what we see here. This is done with the Boston Children's Hospital and the Dr. Hopkins. In fact, Dr. Hopkins is able to reconstruct a personalized heart for kids. He's working in pediatry. In this example, an 18-year-old patient had, once very young, a congenital pathology with a single functional ventricle. And so this patient had to undergo a very young intervention, establishing a shunt between the inferior vena cava circulation and the pulmonary artery system. And so later on, this became very dangerous because it created a non-normal flow of the air and the blood. So what we had to do, and I will relaunch again the video if I can. Whoops, was too fast, sorry. What the doctor had to do is to modify that shunt and to basically design a specific device to be implanted in that specific heart, because the impedance, if you will, of the air between the valve and the artery was not fitting. And so this is what you see here. He recreated the very specific heart of that patient, and then within a modeling solution that we have, created this specific device, this patch that you see here, first in 3D, then unfolding it to be able to print it in specific bioresin, and then reimplant it so that in the end, the patient was cured. And the patient is going very well right now. The key message here is that this is twin, not to represent. This is twin to guide specific designs of medical device. So towards the personalization trend in manufacturing devices. Second example, following that example, such as the one I presented, FDA contacted us. FDA contacted us because they believe that therefore, since they had seen that in either medical practice and device design, maybe the twin could also help transform the clinical trial of the device themselves. So here we're not talking device specialized for one guy, but we're talking about a device that needs to be accepted by the regulatory bodies for a whole cohort. So how could twin help that? Well, and if you see the little table, which is very small, I apologize, a lot of companies in devices have been trying, for example, in this case, to solve a cardiomyopathy case. Out of 20 tries in the industry, only one device was accepted by the regulatory body. So a lot of cost, a lot of money injected, and a lot of waste. So here what we did at the bottom line is we reused the model that you saw before, adapted, and then we generated fakely, if you will, cohorts by varying all the different physiological parameters of the twin. Size of the valve, deepness of the borders, the speed at which this is clipping or not, size of the body, size of the ventricle, all the hemodynamics within going through the valve and so forth. And so we created a design of experiment and therefore generating 100,000 virtual patient hearts and valves. We use modern sim for that, as well as AI to be able to generate that. We generate surrogate model, if you will, pretty much the same thing as what we do in the aerospace industry. And therefore, FDA was very interested because they wanted that approach to be basically a standard practice adopted by the manufacturer devicer. And so we are engaged right now in a 5-year project with FDA to transform the current processes of clinical trials for medical device. You see here the very detailed of the valve itself, and then on the right side, the cohort. And the idea is to more and more abstract that, to make it available to more and more prostheses, ortheses, specific devices, and so forth. So of course, we are not replacing the real patient. Those are complementary cohorts today. You could call that a third leg or a third arm, but on and on, this will be pretty much as the digital mock-up became complementary to, let's say, in-flight testing in aerospace. This is coming more and more as a standard recommended practice by FDA, and we are in talks in Europe to do the same with the equivalent regulatory body. And last example, so specification of personalized device, clinical trials, and then connection with pharma. Twin can also be used, and we saw a very interesting talk this morning about safety, for example. Twin can be used also to better assess the safety part of a new drug. You know that the different clinical trial phases, phase 1, phase 2, phase 3, are mainly to first assess the toxicity. Does the new compound, does it do harm or does it do well? And then, of course, efficacy. Does it treat better than what we had before? But efficacy comes after safety. And so safety is very important. In this example that you see here, we are addressing anomaly in a cardiac rhythm, tachycardia, if you will. This is, of course, a very known disease. There are some drugs, but some of the drugs, if not well taken, if the observance is not the one recommended, and we all know that observance is a matter in pharma, sometimes the very same drug that is supposed to help is generating some problems, like side effects called torsion de pointe. I heard that in English it's the same term, torsion de pointe, which is an added arrhythmia plus a torsion in the pounding of the heart. So what we have been doing multi-scale modeling of the heart, modeling the drug, the candidate compound, modeling the way that drug was inferring the ion channels in a cell, in 11 specific ion channels, then scaling up all those cells, doing some sort of a big data, big cell model at the tissue level, and then observing the cardio endpoint as a consequence of those pathology. And what you can see in the next slide, it's a little bit busy, a little bit small, but on the right side, what you see is the different dosages and what the simulation is showing. Here we show on the upper left the polarization of the ventricle, and then the tissue response, which is the deformation on the fourth line. And you see the columns are no drug, posology, 3 times the posology, 5 times the posology, 20 times the posology. And what is amazing, and to be honest, I don't really know why, from a pure scientific standpoint, because this is a multi-scale, let's say a Russian doll kind of approach. This is, in simulation, replicating perfectly what we observe, at least for the 3 first quantitative example. So here we are able to replicate some sort of a nonlinear, chaotic behavior of interaction of a drug on an organ in a pure, complete in silico way. And therefore, some pharma are contacting us to also, as a complement to their clinical trial or their safety phases understanding, to have a lot of simulation being done with the same approach so that they can at least understand the effect of the candidate compound and eliminate some of them. So those were 3 examples: personalization in device design, change transformation in clinical trials, and new kinds of connection in terms of pharma for safety and let's say clinical trial go-to-market process. On neuro, and here this is just, I guess, a teaser to my colleague Professor D'Angelo and to my other colleague Professor Girsa, who will talk tomorrow on the same thing. Neuro, of course, is a hot topic also for us. Here we're using a virtual epileptic patient model, again, reconstructed totally from a specific case to anticipate a surgery. This is for epilepsy, for drug-resistant epilepsy. You know that today 1% of the population globally will be affected by epilepsy, and a large number of those epilepsy cannot be treated by drugs, unfortunately. So they need to go to surgery. And unfortunately, 30% of those epilepsy, cannot be treated by drug, and unfortunately, even by surgery, sometimes it is not conclusive. So here what we're trying to do, and we are working with our friend in APHM Marseille, Université de Marseille, coming from the Human Brain Project and eBrain, we are generating white matter, gray matter, tractography, all the fibers, vascular system. So we are generating a true personalized twin of one epileptic patient. And then, thanks to some technology derived from the Human Brain Project, we are able to simulate Before we simulate the blood in the heart, here we simulate epilepsy spike and strike. And doing different virtual crisis, virtual stroke, we are, like in Google, zooming in and zooming out to find out what would be the minimal, first, where the epilepsy strike is being born, and what would be the minimal volume to be resected so that we don't have any larger side effect or impact after resection. Right now, this is a research hospitalo-universitaire in France. There are, if I remember correctly, Victor, correct me if I'm wrong, more than 60 patients already enrolled, virtualized, if you will. And so we are developing on and on those technologies here for epilepsy. But after that, and we are talking new IHU in France, to expand that to new therapeutical diseases, of course, the one that we all think of, Parkinson, ALS, and so forth. And so there's a lot of challenges for that. We do, of course, a lot of AI on imaging, and I'm sure that my colleague will expand on that. We do, of course, a lot of AI, but driven by some neurological knowledge. This is always, I think, a key message in terms of twin. It's not only AI taking a whole bunch of data and generating something. It is always AI, but guided by a medical knowledge. So here we have fed into the AI technologies, deep learning and stuff, the knowledge of deep learning, the knowledge of neurology, the knowledge of Parkinson, the knowledge of epilepsy, so that the learning system is not generating something blindly, but it generates something that makes sense for a practitioner or a neurologist. And we have, as I said, this unique technology for spike simulation. Moving to oncology, this is something dear to my heart, starting from imaging and then moving on to a virtual tumor. In oncology, to detect tumor lesion, usually many CT scans need to be performed. And as you may all know, a radiologist need to look at all the slides. It's a very painful and long process, and select and annotate the different lesions that exist on those CT scan according to a specific nomenclature, which is called RECIST, which is a gold standard. But in fact, it's a very long and tedious process. So what we have been doing, leveraging a lot of imaging that we have through expansion of my company with a clinical trial company that we acquired, we taught an AI system to recognize all those guidelines of the gold standard RECIST criteria. So the system avoids the radiologist to look at every slice one after the other, does the propagation of the identification of the tumor slice by slice, and more than that, reconstruct a tumor in 3D. And after that, what we do, we can do, if you will, a difference between a CT scan done in January and a CT scan done in February, for example, and with some simulation model and statistical model, try to predict the evolution of the tumor. So, of course, we need a lot of data to do that. So we are partnering with a lot of institutes, hospitals, and even pharma who agree to at least for research purposes, share their data. And also, we are expanding the criteria themselves, because what the AI is able to show is that sometimes those guidelines miss some point. So there is also a feedback from those technology, which is evolution of, let's say, the standardization and the processes. And last but not least, of course, we are evolving also the technologies of AI, because in fact, right now, what you have seen on this demo is, let's say, classical visual computer vision technology, and we're leveraging some new technologies that maybe you have seen this summer with some software called DALL-E, which was imaging inspiration, but which basically does what? Explain the context of something. And here we're applying AI to understand the context of a tumor. And to finish with cancer, this is also, let's say, a project that we are doing in my lab. In the end, what we want to do is really to be able to understand the evolution of a total tumor. So what you see on the left side is a cancer cell with its entire protein network, pathways, interacting, signaling pathways, expression of the genomes, So waits, if you will, in a different pathway. So it's a living cell. And as you know, a sane cell is a sort of equilibrium, and a cancer cell, in fact, is another equilibrium, which is difficult to change. And we have been stacking those cells that you see on the left side 1 billion times, generating this tissue, which is the tumor, which is mostly 1 centimeter cube. And here we have been injecting some behavioral modeling in terms of angiogenesis, lymphogenesis, mutation, motility. So to render that as precise, as accurate as possible, of course, all those parameters depending on the location in the tissue where the tumor is. So this is an ongoing project. The real challenges from a technology perspective here is not so much the scaling and the simulation. Here we can do that very well. It's the calibration with the real world. What are the endpoints of the tumor, and how do we relate them with, let's say, the sensitive parameters of the model, and how do we make sure that this calibration is viable enough to help the practitioner prevent evolution in a certain way. So more to come. This is an ongoing project. It has been many years already, but I'm very confident that one day we will have virtual tumors. And to finish, if this thing is okay, here we mostly saw in cardiology, neurology, and oncology 3D twins. But in fact, the notion of twin is not necessarily biomechanical or 3D. As we saw with Thomas this morning, it's also a multifaceted information twin. So it's more data-oriented with some structure. And here I have a little video with voice. I would like to underline a practice that exists since, let's say, 2010. But we have been lucky enough with the Medidata acquisition that we've had to really lead the pack. The idea here is we look at a clinical trial. We all know that clinical trial is divided into 2 legs, what I call the placebo leg. We enroll people and we give them water, nothing, placebo, or a standard of care. So those people are there to control. It's a reference leg. It can be hundreds of people. And we have another leg, which is the patient who are ill, and we give them the candidate compound. So if I summarize it on, let's say, a more economical standpoint, the control leg takes a lot of time to enroll and is basically just a reference. So can we use data of previous trials, even trials that were not thought for that specific new therapeutical areas, and can we try to see if there is already some insight in those data to basically accelerate the control arm and make that useful to conduct a new clinical trial on a new compound. Of course, we are in the sector of clinical trials, so the data is very clean, very structured, so it's a little more easy. But the idea is to take some data, associate them with some, let's say, inclusion criteria of the new compound, and there is some sort of mathematical reprojection so that the selectees, if you will, can enter the trial as a cohort. So let me show you, it's a small video. But this is, the reason why I'm showing you that is that this is right now becoming a standard practice at FDA. And pretty much like in aerospace, we coined the digital mock-up more and more so that aircraft are now certified with the virtual representation of the aircraft. Now it's not everything in silico, luckily enough, because in the end it's health, so we need always to check on real people. But the control arm is being synthesized, virtualized, and entering a new practice at FDA. So this is with sound.
Synthetic control arm can enable scientific research, cut costs, and accelerate timelines in scenarios where a control group is hard to recruit or retain, such as rare or imminently life-threatening diseases with inadequate standard of care treatments, like some cancers. From a set of archived clinical studies, the control database is created by extracting and normalizing the control arms of these studies. The next step is to build the synthetic control arm. We can assume a new clinical trial is running. The enrolled patients receive the experimental treatment and therefore belong to the experimental arm of the study. A subset of the control database will then constitute the synthetic control arm. In a classical clinical trial, patients are randomized according to baseline characteristics such as gender or age, which ensures similarity between the study arms. In synthetic control arms, all enrolled patients are assigned to the experimental arm. So how do you construct the synthetic control arm? This is a matching process. First, candidate patients for the synthetic control arm must meet the eligibility criteria for the new study. Then, each patient in the experimental arm will be matched with the control patient, who has the most similar baseline characteristics. The matching will be performed using a propensity score estimate. The propensity score is the probability of belonging to the experimental arm knowing its baseline characteristics. This probability is estimated for each patient, in the experimental arm and in the control database, by logistic regression. In practice, the synthetic control arm must be created by selecting the control patients who will correspond to each patient in the experimental arm. Let's consider 3 patients belonging to the experimental arm. We know the propensity scores of each patient, between 0 and 1, represented here by the purple gradient. And we know propensity scores of all the patients of the control database, represented here by the orange gradient. Each patient of the experimental arm will be paired with the patient of the control database who has the closest propensity score. This is how the synthetic control arm is obtained. The quality of the synthetic control arm is assessed by checking that the distribution of baseline characteristics in the experimental arm and in the synthetic control arm are similar.
And so this is, of course, helping classical trials, speeding up the process, because we don't have to enroll classical people in the control arm. But also, and this is not, let's say, a light statement, this is also helping trials in rare disease, neglected disease, or any kind of disease where it's extremely rare, it's extremely difficult to find patient. And so this is a new approach. Again, FDA is now taking really over this, and I'm moving on. Already pharma are pushing new drugs, new compounds, right now in phase 2, already in phase 3. I was hoping that for this conference I could announce an approved drug, so it's not yet done, but stay tuned. But you see that this is an evolution, it's a transformation in the pharma practice, leveraging data in a structured way, so this is what I call the synthetic cohort. To finish, so you've seen example, so it's a call for action, basically. We are convinced that the virtual twin are going to change the medical practice, not only by providing a deeper insight for disease, but also by changing the way people work together. And again, this was maybe the best or the most profound findings in adjacent industries. When we did twins for cars, for boats, for planes, for any kinds of manufacturing industries, of course, we had better representation, more accurate simulation, more precise manufacturing. But the net of it was that it was a completely different way of organizing a company, making people collaboration, and having disciplines understand themselves in a much more profound way. So we have opened this program, which we call Virtual Twin of Human. We'll be announcing in the next, I hope, before the year end, an extremely large European project on that, on many, many therapeutical areas. We'll be talking on it next Thursday, on the, let's say, twin event of my company next Thursday. And also, we are animating, I think, the world's largest symposium on virtual twin on a yearly basis. It's a 700 people event, virtual and physical. And the next one is next December. So if you're interested, contact me, and we'll be happy to welcome you. And I'm done.
Thank you, Errol, for having given me this opportunity to be present in this session. I'm purely from an academic background, so not in industry as my other speakers, previous speakers were. Today I'll be discussing a few of the applications that we have been undertaking in our group in the domain of applying AI to biomedical images. So first, with a brief introduction to AI, we go on to some of the 3 applications that I would like to focus on. One is on the segmentation and survival prediction in brain tumors, the other on the screening of COVID-19, and the last one on delineating the pathologies in diabetic retinopathy. So in medicine, you cannot get away from imperfect knowledge, mainly due to incomplete understanding of the mechanisms, imprecise test measurements, There are uncertainties of different kinds, simultaneous presence of more than one disease condition, missing information. Therefore, you need intelligent analysis incorporating some kind of learning, some kind of automated analysis as assistive intelligence, not to transplant the medical expert, but to help the practitioner to handle the big data, and sometimes through interactive or active learning when you have insufficient ground truth, which is a major problem in medical processing. The insufficient availability of annotated information, which is required in deep learning. So first, to a term called radiomics, which is the high volume quantitative information from radiographic images. So instead of the image, you extract certain quantitative features and the machine can work on it. So it involves an intersection of machine learning, deep learning, medical image processing and analysis, and of course, computer vision. So in radiomics, typically we, in this particular publication, we analyzed and demonstrated that semi-automated processing was as good as the manual processing, which is typically done. Because in the manual processing, when the human observer is there, the inherent bias and the tiredness steps in, particularly in today's domain when you have huge volume of data. Then to personalized medicine, as one of the earlier speakers had also pointed out, so you need to offer the right drug to the right disease at the right time with the right dosage, and that depends on the genomic information of the individual. So radiogenomics, again, is an integration of radiomics and genomics. So we want to replace or substitute the genomic input, which is more helpful towards the personalized treatment, of course, but it is invasive. So we want to replace it or substitute it by certain other non-invasive methods like the radiomic features. So when you have an integration of radiomic and genomics, you come into this new term which is called radiogenomics, which is the association. This association has the potential for a patient-specific personalized management of diseases. So as I mentioned earlier, so with the genetic information, the application of a particular drug could have different impacts. So either it could be toxic and not beneficial, toxic but beneficial, not toxic and not beneficial, and again not toxic and beneficial, which is the best possible option. Another aspect which is investigated in the applications of biomedicine is histopathology, which again involves huge volumes of data, and you need, of course, high volume processing and quantitative inputs. And it is the gold standard typically in diagnosing certain diseases like cancer. But with the growing number of digitally scanned tissue slide images, computer-based segmentation and classification is in high demand, but there are certain disadvantages to it, because it is not a trivial task to apply the deep learning methods to the histology slides. It has several challenges like variations in the color of slides to excessive high resolution and lack of proper labeling across different labs, of course. So here you see some of the challenges like the cropped section of the same histopathological image displayed at different magnification levels. You see the huge computational burden involved as you go deeper and deeper into the precision. And then there are tissue handling artifacts typically observed like the image blur, the tissue tear, the tissue folding, staining from a marker. So these are the problems that a computer faces. So then, when we need to apply AI, we need to take care of all these aspects. So AI, as we see, is a superset of which machine learning is a subset and deep learning is another smaller subset within machine learning. The difference between typical machine learning, which we also refer to as shallow learning, and the deep learning is that in shallow learning or in typical machine learning, you need a good feature extractor that solves the problem. So you need an expert to interact with, so you need these handcrafted features to be extracted and put to the machine. So if the extraction of features is not proper, then there can come in problems in the decision making. Whereas in deep learning, you are automatically learning the good features. It is extracting on its own. So very recently, we undertook this study on exploring the genomic features with deep learning. Of course, a key problem with genomic features is that we have to depend on publicly available databases, as these are, in our country, it's not always available. So we had this interdisciplinary work with the biologists in our group, and the genomic input was in the form of RNA sequencing data, and we were trying to predict the overall survival of patients. And we used autoencoder, which is one kind of a deep learning network, for extracting features. So we developed an autoencoder-based feature extraction model, and it was an interdisciplinary application, involved some genomic pre-processing. We selected differentially expressed genes, reducing through correlation-based analysis. Then we did some exhaustive experimental study, and we did some quantitative evaluation to establish the statistically significant performance of the model in terms of the concordance index. And we analyzed the weights of the autoencoder to infer the importance of the encoded genes. So this was some part of the pre-processing which we did, and we compared with different feature selection algorithms and tested the efficacy of these extracted features or the genomic features in terms of existing classifier models like machine learning, linear regression, support vector machine, and so on, decision trees. And the effectiveness of the autoencoder extracted genes were evaluated in terms of the ROC curve. And we compared it with the biological significance of the extracted genes as available in literature, and we could find a good concordance. With that, we move on to some of the applications of deep learning, which we have pursued. So the first one is about the brain tumor. In gliomas particularly, we use the MRI. It has 4 sequences, and different parts of the tumor are differently expressed or differently observed in good manner. So we find that if we take all 4 of them, we get the different subparts very well. And this is, as we see, a sample image of a high-grade glioma and a low-grade glioma. And obviously, the prognosis is very different in the 2 cases, and with the naked human eye, it's very difficult to recognize or distinguish between the 2. Therefore, we went on for employing deep learning. So we designed this model, and we also participated in the MICAI challenge with this model, which is a public challenge which is organized. And this was for segmentation and survival prediction in gliomas. We designed a model which was the multiplanar spatial convolutional net for the purpose. So typically in deep learning, the U-net is used for segmentation, which has a contracting or downsampling path with a bottleneck part at the bottom and then expanding or upsampling path. And there is a skip connection, a set of skip connections between the downsampling path and the upsampling path. And there is end-to-end training. So typically what we observed was that at the boundary level of the tumor, certain discontinuities was occurring in the unit, the conventional 3-dimensional unit. So there was some error at the boundary, and which is obviously a drawback in medical image segmentation because it is the information that the surgeon will use. So we designed this model, where there was a spatial max pooling layer which retains the max locations to be subsequently used during the unpooling operation in the decoder through this particular layer. So these were the different parts of the tumor which had to be modeled in this challenge. So the error around the boundary was a major problem, and the issue of false positives. The error around the boundary was solved by the spatial max pooling and unpooling, where the location of the maximum point was also preserved in the skip connection. For the false positive error, we used different multiplanar representation along the three two-dimensional planes and combined them so that this could be overcome. And to handle the class imbalance, obviously, if we look at the pixel level distribution of the different regions of the tumor, the background region, which is least important, has the maximum number of pixels. This creates a problem of class imbalance because the important part is the tumor, which is very small in percentage, which is 0.23 as compared to 98.46 in the background. So this is class imbalance, and this needs to be mathematically handled. So we introduced a new loss function, which was a combination of the generalized dice loss and the weighted log loss. So this was the experimental setup in TensorFlow in Python, and this was the box and whisker plot and the dice score, Hausdorff distance, and so on, quantitative differences. So this was the leaderboard score of that challenge, and our model was selected among the top performing ones in terms of the dice score, the sensitivity, the specificity, and the Hausdorff score. Then the next part was the survival prediction. Again, since deep learning requires heavy amount of data, in this particular challenge, the amount of data was not that high. So for the survival prediction, we used shallow learning or typical machine learning. We extracted certain radiomic features, 296, and from them, we employed certain shallow classifiers to predict the survivability. So there were three classes as provided by the organizers: the short survivors, mid survivors, and the long survivors. These were the set of features, 296, that we had used of different categories: the gray level statistics, the shape level features, the co-occurrence matrix level features, and the run length matrix level features. And this was some of the quantitative analysis of the segmentation as well as of the survival prediction. In the next part, we employed deep learning again to the screening of COVID-19 in these because we were passing through the COVID period, and of course we had the publicly available data only. So this was on the chest X-ray images. So this is a sample image set distinguishing between the pneumonia caused by COVID-19 and the others. So the data sets used, the interesting part is that, as I mentioned earlier, in deep learning, the main problem is the lack of sufficient volume of annotated data. So annotated means the ground truth should be present. So here also we had a similar problem. So we improvised a new method for handling this. So you see that the data A is the training data that we used. It had only two classes, the normal and the diseased, because of the lack of data, annotated data. And in the test set, which was data set B, we had 4 classes because these were unannotated at that point of time, and then normal, pneumonia, other disease, and COVID-19. So we trained on that 2-class data, and we tested or validated on this 4-class data using domain extended transfer learning. So the domain contained the 2 classes, normal and diseased, while the validation or the test set or the target domain was the larger one, which had 4 classes. So we trained the deep network or the CNN model here from scratch on the dataset A, which was the smaller one, to learn to classify between the two classes. Then we replaced the last fully connected layer of the pre-trained model with a new fully connected layer, now having 4 outputs. And the rest of the weights in the remaining layers were retained. The last convolution block and the fully connected layers were retrained using stochastic gradient descent, and the evaluation was made on the superset of the data. So then these were the 3 popular models of CNN that we tested on. And the visualization was evaluated in terms of the gradient class activation map. And we also participated in a challenge. And finally, this is the segmentation in diabetic retinopathy. Now, diabetic retinopathy is a disease which is permanent, irreversible, and happens mostly in people suffering from long-term diabetes, and in India, which is also the diabetic capital of the world. So there, this is very much relevant. So these are the different pathologies occurring in diabetes in the eye: microaneurysms, hemorrhage, soft exudate, and hard exudates. So these, depending on their density and their prevalence, you grade the particular disease grade, and then thereby it can be controlled or not, or if it is too late. So then we designed this deep supervision-based layer-wise training. We had a multi-scalar resolution of inputs. And this was the data set, again, a publicly available data set. But of course, now we are working in an actual collaboration with real data. And this was the data set and its preparation. We did some patch extraction. And this is the model with the quantitative loss functions which were designed, and the multi-class extension of the generalized dice loss. These were the 2-phase training of the model. And finally, these were some of the visual results. And then, of course, this is the last slide. And very recently in India, we have started this first fully annotated, de-identified, publicly available image bank on the Indian population. Of course, you may realize that most of the data that is available is from the Western world. And if you consider the genomic factor to be very important, then Indian population is also very large in the world context, and we need specific data. And so this is a very good application. This has been started in collaboration with the Tata Cancer Center and the Indian Institute of Technology at Kharagpur. With that, thank you.
So thank you so much for inviting me. It's quite an honor to be here. As Errol said, I'm a big believer in international collaboration, so it's wonderful to see a consortium of national academies and members from all over different countries, and including, very honored to have the US represented here through various people, including myself. So I have the benefit that Errol's already given kind of a synopsis of my talk, so what I will attempt to explain to you. So as he said, I am the director of West Coast Labs for Facebook AI research, now called Meta. So I see we're having a little bit of a problem with getting the slides up there. Oh, thank you. Let's see. Oh, there we go. That's a little better. Thank you. Yes, so thank you for your synopsis of what I'd like to talk about today. I'd like to talk about private AI for human health and genomic data. And let me just start right in with a number of the other speakers have spoken about AI and its use in healthcare. Being a mathematician by background, I like to try to kind of explain a little bit how AI works from the mathematical point of view, and then what we need to do in order to be able to protect privacy through tools like cryptographic tools like encryption. So let me start with where we are today, which is what I call the AI era. I feel like AI is changing our lives on a daily basis. So humans are now more and more relying on smartphones for all kinds of services, from speech recognition, including things like automatic translation, image recognition, which we've heard quite a bit about already today in terms of medical diagnoses, things as basic as finding directions to where you're going, getting music or movie recommendations, and all kinds of applications in health and genomics. So the way this works is that machines actually learn from data in a process which we call machine learning in computer science, because we decide ahead of time as mathematicians and computer scientists what models we want to use, and then we feed those models, we are fed data in order to train kind of the specifics of that model, so basically the coefficients or the mathematical models based on the data. And then in a second stage, once those models have been trained, those models can be used for classifications, to make predictions, to make decisions, classifications. So all the examples that I just gave of how AI is used in society, those are happening real time, like when your phone is in your hand, because the models have already been trained, and you're just doing the second stage. You're just using the model for the prediction at that point. So I like to focus on the case of what we call supervised learning, just because it's easier to understand and explain. And that is that when the training is done, when you're trying to derive this model or function, the data that you're using is already labeled. So you already know, each piece of data, how it's classified. And that helps the model to essentially try to learn how labels are derived from data. In order to do this in a mathematical setting, all of this data has to be represented basically as vectors. When we talk about medical images, we're talking about arrays of pixels that have to be kind of vectorized. So in the genomic situation where you have SNPs, single nucleotide proteins, those are just represented actually with bits. So you'll have a sequence of bits representing genomic sequences. So those can also be thought of as vectors. And all of the other examples that I talked about, all this data from the real world has to actually be translated into vectors or sequences of bits in order to be processed. So that's one stage of the process that is also often kind of ignored or underappreciated, because that process itself can be very important in terms of efficiency. So just to explain AI in the medical context, so the mathematical side of it, but in a very, very simple example. So here's an example of where you might want to do a prediction on some medical data. And what you have here is a task which is predicting whether a tumor is malignant or benign. So each row of this table represents one patient. And as you can see, there's around 15 or 20 characteristics of this tumor that have been measured. And these are numerical measurements, so actually real floating point numbers for each of these characteristics that have been measured. And then on the far right, you see that there's a classification. So that's the label I was talking about. Green if it's benign tumor, red if it's malignant. So the idea is that this is a very small dimensional space by the standards of machine learning today. Like I said, the number of columns here is only about maybe 15 or 20. And so the task here is to try to learn from this data. You would have many, many more rows of data in order to develop a model, and then that model would be used the next time when you're looking at a tumor, be it an image of a tumor where these measurements could be taken, or some other process by which you've gathered this data from a patient. So a very, very simple mathematical model for binary classification. So binary just means there's only two choices. So in this case, it was malignant or benign. Of course, you could have much more complicated classification problems, which are not binary when there's more than two choices. But in this case, I'm just trying to explain what a binary classification problem looks like from the mathematical point of view. So what you do, this is the supervised case where all your data is labeled, and you basically are dividing your data into two categories. The green is the benign, and the red is, in this case I have it in blue here. And these dots are in high dimensions from the point of view of human eye. Like I said, it was about 15 dimensions. And yet here I'm showing you only a two-dimensional image. So this is a bit misleading because we can't see the 15-dimensional space with the human eye. We're just looking at a two-dimensional picture here. But the mathematics can kind of see the 15 dimensions. That's the beauty of the mathematics. So we're creating this kind of picture in our mathematical minds of a 15-dimensional space where all these data points are kind of clustered together. And so a linear means classifier is really, you could say, almost the simplest kind of AI, is just using these labels and this kind of clustering to compute like a mean vector for each of these two classes. So you've got the benign class and the malignant class. And you've got a mean of each of them. A mean is just like the average in high dimensions here. And what you're going to do is you're going to compute a difference between these two means and kind of define the midpoint and define a hyperplane that separates these two vectors. And so now your classification problem is fairly easy because you think of that hyperplane here, just think of it in two dimensions. It's not accurate, but it's trying to give you the idea. Think of a line that separates these two classes, and then when you get your new vector in from your test vector, you're basically trying to decide which side of the hyperplane am I on. Now, as I said, this is a drastic oversimplification of what actually happens in machine learning today. These are just the very beginnings of the ideas, but it should give you the idea of how mathematics is used here in this space. Data from the real world has to be converted and represented in a mathematical form. Then it has to be handled by deciding on some mathematical model to use. Then you have to actually process the data and update the model and decide. In this case, the model is just a hyperplane. It's really what we could call a decision boundary. It just means here's a boundary that tells you how to decide which side of this boundary are you on. So now let's generalize from this to a little bit more complicated machine learning going towards what is actually used today in deep learning. The general strategy is roughly the same. That is, from a large amount of data, you are trying to learn a decision boundary. You're trying to use the mathematics to learn a kind of a rule that can tell you basically, metaphorically, which side of the hyperplane are you on. And the way that this is done in general is by defining a loss function. A loss function could be something as simple as the distance from like a hypothetical kind of decision boundary, and a distance like, a least squares distance, for example, which is telling how far are you from the current decision boundary, and maybe this decision boundary should be updated. So this loss function is then used as a measure of whether you should keep training your model. So should we keep going on this process of training, or should we stop? Do you think we have a pretty good model now that we don't need to update anymore? So step by step, you proceed to try to minimize this loss function. And when the loss function, when it kind of drops, that gives you a good indication, oh, wow, maybe the machine has now learned, and maybe this is good enough for our training. Maybe we can stop now. And this process, it's kind of interesting being from a different background in mathematics. So my field is actually number theory and algebraic geometry, and from that I went into cryptography and security. So I was actually initially very far from the machine learning and AI field, but then again, when I did my PhD more than 25 years ago, machine learning and AI was not the booming industry that it is today. But I was not a big fan of like the analytic part of mathematics. So real and complex analysis were not my favorite topics in graduate school, and multivariable calculus was not my favorite thing to teach to undergraduates. In fact, when I was an assistant professor at the University of Michigan, I developed a course on basically the algebra of cryptography and coding theory just so I could avoid teaching multivariable calculus. But now, looking at AI, what we're actually doing inside of the AI algorithms, you could really think of it as multivariable calculus. So you have this very, very high-dimensional space that you're trying to move around in, and you're trying to understand the relationship of points in this space using various measures, and you're trying to minimize functions within this space. And it's really very high-dimensional multivariable calculus, in a sense. So I wish that when I had been learning and teaching multivariable calculus in college, that I had understood these important applications. But it's nice to be able to come and appreciate a different field of mathematics today. So that's my high-level description of how AI works. And so now we want to think about this and apply this in the medical space. So one of the things that really motivated me in my career was when I was leading the cryptography and privacy research group at Microsoft Research, and I started working on privacy in healthcare and genomics. And I found this to be very motivating because I finally felt, as a mathematician, for the first time, I could talk to people about my work, that I was trying to work on protecting privacy in healthcare and genomic data, and people would actually care, and they would be enthusiastic and think that sounded like a good idea, rather than just telling me how much they hated mathematics as a kid, which is the usual thing that people tell you when you tell them that you're a mathematician. So I really enjoyed starting to work on privacy. And the privacy problem in AI is that, as you could hopefully see from my description, the whole process, the whole AI pipeline relies on using a very large amount of data. So you have to collect a lot of data in order to train these models, and then at the prediction stage, when the models are being used to do classifications or valuable recommendations, you have to input your data again in order for the model to process it. So all these typical AI services which are hosted in the cloud, then you might ask about both the security and the privacy of that data. So recently, just in April, in Siam News, I actually published an article kind of explaining my work in this area, private AI for doing machine learning on encrypted data. So this was what was so nicely summarized in Errol's introduction for my talk, is that we actually have encryption techniques which can be used to protect the privacy of this data, in particular health data. So there's a new mathematical tool as of a little bit more than 10 years ago, which is called homomorphic encryption. And the nice thing about homomorphic encryption is that it provides a new functionality that was not available before, which is that it's encryption that you can actually compute on. So I have two different descriptions of homomorphic encryption that I like to give. So the first one involves Homer Simpson, looking a little bit surprised here. So this is just purely, I'm sorry, shameless just entertainment value here, including Homer Simpson. But in one of my talks on this topic, somebody tweeted that I was talking about homomorphic encryption. So ever since then, I always have to introduce this topic as homomorphic encryption. Numerous executives over time in the industry have complained about our terminology, that we should pick a term which is a lot easier to pronounce than homomorphic encryption. So Homer Simpson is helping us out here to make this a little more accessible. And the idea of this picture is it's supposed to represent the value that homomorphic encryption has for us. So suppose you have some gold, something very valuable, and you want to outsource the work on this gold. You want to have a jeweler work on this gold to make you some jewelry. Now here, Homer Simpson is the jeweler, and he's not looking very happy. Why? Because we don't trust him, and we're going to force him to make this jewelry by working with his hands in this glove box, where he has to operate on the gold through the glove box. So we are going to actually put our gold into this locked box that's got the gloves in it, and we're going to lock it and keep the key ourselves. So he does not get the key. He does not get to unlock the box. He has to work on the gold while it's locked in the box, and then when he's done, he can't take any of the leftover shavings with him or anything. He has to give the box back to us, and then we have kept the key, and we unlock the box and take out the gold. So this is, and the jewelry that he's made. So this is supposed to represent that we have some valuable thing, which in reality now is going to be our data, our health data or our genomic data. And instead of giving it to the cloud in its raw form to have some predictions done about our risk of having some disease or have our images analyzed for what type of tumor we have, we're going to give it to the cloud in encrypted form. And in the cloud service, the cloud can process this data in encrypted form and then give us back an encrypted prediction, which we then decrypt using our key. So that's the kind of Homer Simpson analogy for homomorphic encryption here. From a more mathematical point of view, you can just say, like if someone were to ask you at a cocktail party, what is homomorphic encryption? You could just say, oh, it's encryption where you can reverse the order of compute and encrypt. That is, you can either take your data and you can compute on it and then encrypt it, or you can encrypt it and then compute on it. And when you decrypt, you should get the same thing. So it's a very fairly simple concept, but actually it was not at all easy to find a solution to this problem, to homomorphic encryption. The idea of it was proposed in the 1970s, and the first solution was proposed in 2009, as I said, just a little more than 10 years ago, and it was considered completely impractical. Until in 2011, in my team, actually at Microsoft Research, there was a surprise breakthrough, and researchers in my team proposed the first homomorphic encryption scheme based on a well-known mathematical problem that was related to hard lattice problems in mathematics that had been studied for many decades. And we immediately, because we had a very interdisciplinary team of mathematicians and computer scientists, we immediately started working on making this practical and applying it to disciplines such as health and genomics to protect privacy. And then another big step was in 2016, our team was the first to show CryptoNets, which was the first demonstration of actually being able to evaluate deep learning, deep neural nets on homomorphically encrypted data. And more recently, I've been giving many talks like this, for example, at the International Congress of Industrial and Applied Mathematics in Spain in 2019 about the use of homomorphic encryption to protect privacy in fields such as health and genomic data. So many people, if you are familiar with homomorphic encryption at all, you will think of it as like a wildly impractical algorithm that cannot be used in practice. And that's the kind of work that my team did over the last 10 years, is to kind of make that statement false. So when we started, the overhead from using encryption was like said to be something like around 10 to the power 13. And so that, as you can imagine, that overhead is pretty prohibitive in terms of using in practical applications. And as you can see from this curve, we were able to steadily improve this overhead through a combination of algorithmic improvements, new schemes, arithmetic improvements involving the mathematics, and then engineering improvements, and finally hardware improvements. So there's a whole community of people that have been working on this technology over the last 10 years, so that we can now say that the overhead is more like 2 orders of magnitude instead of 13 orders of magnitude, so that if you process data in encrypted form, you'll be taking something like 100 times longer than you would have taken if you just processed the data in the clear. And when you're talking about operations that take microseconds or milliseconds, you can tolerate about 2 orders of magnitude slowdown
down of your algorithms. And that's what I mean when I say that we've made this technology practical. And I want to emphasize what an incredible interdisciplinary effort this has been. So working with both mathematicians and engineers, my team developed an open source released SEAL, which is Simple Encrypted Arithmetic Library. It's available to download from sealcrypto.org. We also developed an international community of researchers on homomorphic encryption to standardize the technology and released a standard in 2018 that the community approved. You can go to homomorphicencryption.org to read about the activities and the standard of, for homomorphic encryption developed by that community. So I'd just like to pause for a second to see how much time I have left, because it was a little hard to tell with our just 2 or 3 minutes. Okay, so I'd like to then skip to the kind of applications of this particular technology in healthcare. There are actually a lot of different ways that you can use homomorphic encryption. I was alluding to kind of outsourced computation on sensitive data to make predictions, so private AI predictions. But there are other more complicated protocols, ways that you can use it. In many different fields besides healthcare, but certainly in healthcare for analyzing images, so medical diagnoses, and analytics on population-wide, so genome-wide population studies, for example. But I had a few specific examples in health, heart attack prediction or genomic computations. And I also kind of wanted to highlight in the US the role of the NIH in funding a 7-year, so far 7-year competition, secure genome analysis to encourage teams from around the world to come together and benchmark these technologies on various genomic specific tasks. So analyzing genomic sequences and training logistic regression models on genomic sequences, doing viral strain analysis in encrypted form. And so the NIH has been a very important player here in encouraging the development of these privacy technologies. But I didn't want to leave you with the idea that homomorphic encryption is the only tool in our toolbox from the cryptographic point of view. So the last 2 slides that I have here, I just wanted to emphasize that one of the skills of cryptographers is to actually think about and reason about trust models. So often we use the terms security and privacy, and people don't necessarily know exactly what's meant. And I have found various communities to use these terms inconsistently and not really understand. And I use security to mean really the science of securing data, which means controlling who can access it, whereas privacy is more like the science of trying to make sure that data is used in a way that's consistent with how people would want it to be used. And I want to emphasize that you cannot have privacy without security. You cannot control how data is being used if you cannot secure the data. So that's the basic foundation. We have to first be able to do cryptography, security, and we use cryptography as a tool for security. And in this case, we can use cryptography to protect both security and privacy. But you need to be able to reason about who do you trust and what are you trying to protect? What are your assumptions? So this morning we heard Thomas mention federated learning. So that's a situation where what you're trying to do is you're trying to train machine learning models on data from millions of users who don't necessarily want to share their data. So one option you have there is basically to push models out to the edge, what we call the edge, where the data lives, and not aggregate the data itself centrally. Another thing you can do is to try to aggregate the data from a few parties like hospitals centrally and do collaborative learning using a technology like secure multi-party computation. That's number 2. What I talked about in this talk today was really number 3 and number 4, which is where you want the data to be encrypted at the source before it's uploaded to the cloud to do either training or prediction. And then finally, number 5, I'd like to mention differential privacy, which is a tool for building models which don't leak data about individuals when you answer queries, but it actually doesn't protect the data itself. There's no security or cryptography provided in differential privacy. It's about when you hold a lot of data centrally yourself, you're responsible for the security of that data, and now you want to answer queries to third parties. You want to give answers about that data without leaking the data. So this is like the taxonomy that I've just explained of the what I call the 4 kind of privacy-enhancing technologies that we use to protect privacy in a lot of different verticals, but in particular in healthcare today. So I'll end with that, and I'll just leave my slide up here about my our labs, Meta AI Research Labs, which work on an open science model, publishing all of our research and making all of our code available open source. So thank you so much.
Thank you very much, Errol, for the nice introduction and for the invitation. It's a really honor to be here. And it would be nice to see the face of Golgi and Volta after what I will show on the brain and on the circuits that they started to study more than 1 century ago. Okay, I would start with some questions about neuroscience. We are all interested in the brain. I think it's becoming very fancy now. Speaking about the brain is common, and the brain goes on the TV, goes on the media. We're all fan of the brain. However, it's important to break down the questions about the brain in sub-questions. Otherwise, we can't face them. As normally science do, they start with some reductionist approach. So we have to break down the question in sub-questions. For example, when are brain circuits activated and how do they contribute to behavior? That's a very important issue because we know that the brain is made of parts. These parts communicate one with each other. And when we engage the brain in some operation, these parts are activated in sequence or in parallel, depending on how they are taken into play. This is typically a question that is faced by very recent techniques, very powerful, like magnetic resonance imaging, positron emission tomography, magnetoencephalography, that allow to make a map or maps of brain activity. Okay. Normally, when we answer this first question, we don't see the neurons. This is very important to keep in mind. People working in this sector probably spend their life speaking about neurons, but without seeing any neuron during all their career. What happens with the other questions? What do circuits compute? This is more related to engineering. So what happens in the circuit? What is the algorithm of a certain circuit or system of circuits? Yeah. How do circuits operate? This is very mechanistic. This is more for biologists. How does the neuron do their job? And how do many neurons together do their job in a network? Okay. These 3 questions are on different layers, logical levels. And we have to put them together if we want to answer the biggest questions that are, how do circuits contribute to motor, cognitive, and emotional control? If we want to understand something about the brain, we have to face many questions altogether. And even more, if we wanted to answer the questions, but also make something good for biomedicine, pharma, and ICT. We needed to take an approach that is multimodal, multi-level, multidisciplinary, so an approach that is not the approach of a single isolated researcher. We need an organization. I will come back to this point at the end of my presentation. Now, the problem, where does the problem lie? Lie in the multi-scale organization of the brain. As you see from this picture, I've just shown 3 of the main layers. The layer of the single neuron, of the multiple single neurons, which make a network, or of brain areas. At the first level, you see a single neuron very well. You can study almost everything, calcium concentration inside the dendrite, inside the soma, membrane voltage in all the compartments of the neuron. ionic channels, ionic receptors, everything that makes biology so interesting. When you look at multiple single neurons, you lose some detail on the single neurons, but you see many of them individually, all together. And to do this, you need special technologies that have been developed in the last 20 years, mostly based on imaging or multi-electrode array recordings. The example is taken from a 2-photon multi-spot laser microscope recording that allows to see the activity of many neurons simultaneously. And you still see the neurons and their activity. When you jump up to the brain areas, you don't see any neuron. A voxel of the cerebral cortex contains, with a 3D scanner, something like 1 million neurons. So when you say, I have a good resolution, I have 1 millimeter times 2 millimeters times 2 millimeters, that's very good with a 3 Tesla scanner, you are telling me that you are seeing 10 million neurons altogether. Okay, so you don't know anything about the single neurons. Okay, good, this is the situation, and I also show here on the right the organization of the circuit in the case of the cerebellum with the synapses, the molecules, the neurons, the microcircuit, the mesoscale circuit, and finally the cerebellum inside the brain. So this is the kind of multi-layer organization that we have to consider and to face when we want to understand the brain. Now try to think about what the individual scientists normally do. They take one of these levels, they stay in that level for years to understanding them very well, and they know what happens in the other layers, but they do not connect all of them together because or they do not have time, or they do not have the means to do that, or it is even impossible to do it. So the answer to this multi-layer problem in the understanding of scientists in the last 10, 15 years has been to make models. Models that are multi-scale in nature and can bind together all the layers. So here starts the story of the models. We need to generate multi-scale models of the brain. We have to understand what kind of brain models we can make, because you will see there is not one single model, there are many types of models. And then we have to use the models to understand brain functioning. Okay, let's go through these questions. Foundations of brain modeling. We have seen modeling all the day in different forms. This is a kind of modeling that is based on the real causes of the system that we are analyzing. It's not a statistical model, it's a causal model. If you take modeling in its essence, you can go in two ways. Either you know the elementary causes of the system, and you put them together through a function that puts them together. The function basically is the model of the system. And then you generate the observables or ensemble signals. Or you go the other way around. You face the inverse problem, and then you want to break the ensemble signals into components. This problem of going in the inverse direction is very nasty. In some cases, it's not easy to solve. In most cases, it's very difficult to solve, and implies the fact that there can be many sources, many possible combinations of the sources that generate ensemble signal. This issue is called the neurodegeneracy, degeneracy or neurodegeneracy in case of the central nervous system, and implies that we do not have a single solution to the deconvolution of the ensemble signal into the elementary causes. So we have two cases: direct problem solvable if we have enough data and good models. Inverse problem solvable only under certain circumstances and with some assumptions, and will never get out the specific pattern that generated the ensemble. Only one of the possible patterns. But now we can go through this issue more systematically, because in the last year, a group of scientists, including some that are among you here that will speak tomorrow, Victor and Randy, and also we have listened to the presentation of Patrick just before, that we are all working on the same kind of issue from different angles. We tried to systematize the brain modeling story into this figure. We have models that are data-driven and models that are task-driven. Data-driven when we can use the data taken from the brain to make the model, one way or another, direct or inverse. Task-driven when the data are not so clear. The task-driven come into play when we wanted to simulate behavior. Simulating behavior has a problem that the basis of mental representation are not clear to the biologists. Nobody knows how we generate a mental representation. So if we want to generate a simulation of behavior, we have to be very careful. We can't say we put together neurons that generate a mental representation. We're not able to do it. Simply, we can't at the moment. Therefore, we have to take in a different attitude. I will briefly talk of them as well. Now, if we stay on the data-driven models, we can work at the level of neurons and microcircuits. In this case, we have data. We have very good data. Neurons are very well resolved. The microcircuits the same at the experimental level. And therefore, we can generate detailed models of neurons and microcircuits and play with them. You will see the consequences of having this model in action. These models can be simplified and transformed into versions that are computationally very efficient. We have a problem of computational efficiency, and I mean, we can't do whatever we have in mind. Computational efficiency is a barrier for us. So they can be made more efficient and used even further, even beyond the precise analysis of a microcircuit. Then we have the other kind of models starting from brain, where I've written brain. There we start from data taken from the whole brain. And this data, in general, start from MRI, magnetic resonance imaging scannings. And these data are used to reconstruct the connectome of the brain. Once we have the connectome is parcellized into parts. In the different nodes, we can place what we call neural masses, which are condensed representations of the dynamic activity of that particular group of neurons. For example, a voxel or an ensemble of voxels in MRI. So millions of neurons condensed in the dynamic equations, which means we lose a lot about the neurons, but we gain a lot about the dynamics of the system. And eventually we can put together a virtual brain. Victor will speak about it tomorrow, much more in detail. And this virtual brain is a representation of the engine that generates the signals that we can record from the nodes, either using electroencephalography or using fMRI. Okay, that's sort of a miracle, is a wonder, because at the end we can start from data and the regenerated dynamics of the brain activity. You can see here that the neurons and microcircuit models, once simplified appropriately, can be introduced into the nodes of the virtual brain. And therefore, the bottom-up flow and the top-down flow can be merged together. So we can inject biology with a lot of details into the nodes. That was not possible at the beginning. The two columns were separated. A couple of years ago, either you did a reconstruction of neurons and microcircuits, or you did virtual brain models. They could not be merged. Recently, it has become possible to put them together. So we can inject the details of biology into the nodes, and therefore have a virtual brain model that is even more powerful than it was at the beginning. In the case of task-driven models, normally we have to anticipate what the model has to do. The example is drawn with a robot that is the implementation of an artifact that has a behavior in which we write what we want the robot does. It is not completely autonomous. But we can put some autonomy in the circuits by transplanting into the nodes of the controller some spiking neural networks that are taken from the other branches of the modeling system. So again, we can transpose some of the knowledge we have in biology into the robot. So we're generating hybrids, and these hybrids are getting closer and closer to the functioning of the central nervous system. Okay, now I will proceed more using examples than real explanations, otherwise the story becomes too long and complicated. This is just to give you an idea on how the models are constructed. The model of a neuron is, in the essence, a system of differential equations assisted by other functions and other instructions that allows to reproduce the activity of ionic channels and receptors in the membrane of the neuron. And then a master equation resolves the membrane potential in each of the many segments in which the neuron can be fragmented. Just to give you an idea, this is an example of this on the bottom left is an example of the dendritic tree of a Purkinje cell. And this dendritic tree is represented by about 2,000 electrical compartments connecting one to each other. To resolve this neuron, we need 65,000 differential equations, which means it is huge, it's big, it's computationally inefficient, but it's extremely precise. It is able to reproduce all the known behaviors of the neuron. Whatever we do with a neuron, whatever we record from a neuron in the last 20 years can be reproduced by the model, which means it is strongly and heavily validated. Okay, we can reckon on it. In the middle, you see a part, a piece, a module. We have heard about the modules already today. A module of the brain, in particular of the cerebellar cortex, in which the neuron on the left is the small green dot. Okay, it's just one of the elements of the module. The module can be built up to 100,000 or 1 million neurons and still remains computationally affordable using computer clusters. It can be done. Okay, it's slow but can go. And this can reproduce the input-output function of that particular part of the brain. It works very well. So up to this level, we can work. Clearly, there are specific workflows, specific platforms, specific informatic tools that we need, specific organization of the HPC and backend systems to carry out the computation. So we need a lot of technology, but it works. Okay. Now, if we go further on, we see the movies on the left, shows a neuron. This is a Golgi neuron. This is a neuron of Camillo Golgi, the one that was mentioned by Errol. This neuron is working and is receiving inputs from the network. The network is the same of the previous picture. Okay? We have activated the network, and this neuron is inside the network. So we can visualize and measure and record all the parameters of the neuron, because in the model they are explicit. It's another advantage of models, because if you do an experiment, you normally measure one thing at a time, membrane voltage or calcium or something else. With the model, if you have 100 parameters, you can measure all of them simultaneously. Okay. Okay, the neuron is showing its activity, is showing membrane voltage, calcium, activation of AMPA receptors, NMDA receptor, GABA receptors. So just try to imagine what you can do with this kind of models if you want to carry out a pharmacological experiment. You want to test a drug. You can activate or inactivate the receptors or the ionic channels and see how the network reacts. There is a lot of potential inside this. We're beginning to test it. On the right, you see the network. You can you please play it again? The network on the right shows the activity of a multitude of neurons that are activated by an input mimicking the touch of a whisker in a mouse. We have taken the whisker discharge in the neurons, in the afferent neurons. We have played it into the network, and the network is responding. So by, well, now it's not running, but you have seen it. The network shows what is the activity that we expect from that particular part of the brain when it is put at work. Okay. As a form of knowledge it is unprecedented, because nobody has ever been able to do that. So we're gaining knowledge. At least we're beginning to gain knowledge. Okay. So this is an example on how these neurons can be used, and these networks can be used in a continuous feedback between experiments, model, prediction, back to the experiment to validate or test the prediction, back to the model, back to the experiments, back to the model several times. The example is taken from one neuron of the cerebellum. At the beginning, there were electrophysiological characterization telling us the main types of ionic channels in the membrane. We did the model. The model ran well, we were happy, but the size of the action potential, which is the signal generated by neurons when they are activated, was not correct, was too big. And one can say, well, doesn't matter, it's big and that's it. No, it was too big. It was too big, and therefore, what was the reason? So we made an hypothesis. The ionic channels were not in the proper place, were to be dislocated into the axon, which is the filament that comes out from the neuron. And then we moved the channels in the axon, and we normalized the size of the action potential for electrical issues that one can imagine, okay? And at that point, however, this hypothesis had to be tested. We went back to the experiment using immunocytochemistry. We demonstrated the immunolocalization of the channels in the initial segment. So the model was right. That was the first prediction of the model. The second prediction was about the slow state of the model that generated the theta band oscillations that have never been seen. And then we went back to the experiments, and we discovered what we call the M channels, potassium channels of the M type, that are responsible for this kind of behavior. You know that the theta oscillations are one of the main drivers of brain activity, okay? So these neurons have it. Then, next step, the spike has to travel along the axon at constant speed and constant size, and the model didn't do it. So something was still to be discovered. So back to the experiments. And the experiments showed that the ionic channels of the axon, even if they are the same family, which means sodium channels as those in the initial segment, are modulated in a different way. So we went on and discovered with colleagues in New York that these ionic channels are modulated by growth factor that at a certain age enable signal transmission along the axon. And then, by putting this modulator into the model gained the capacity to transfer spikes along the axon at constant speed and constant size. So again, the model predicted properties that were tested experimentally and put back into the model. The story is not finished, and then at some point we discovered additional states of activity of the neuron. The model predicted these states. We went back to the experiments and we discovered the reason, another ionic channel that has never been found in these neurons. So these models are so strong that I just did this example to show you that these models are so strong that are becoming predictive. If they are properly validated, they become tremendously effective in predicting the properties of activity in the neuron and in the network. Okay, so that's the point. So now, well, I skip this in case I will come back to it later. How do we use these models to extend our modeling scale to higher levels? We would like to have applications like in the neuromorphic hardware, in robots, and in virtual brains. So the first step is to have something that is computationally efficient and suitable for the application that we need. Making it faster means simplifying the neuron from 65,000 differential equations to a couple, 3 to 1 differential equation. Is a difficult job, there's a lot of mathematics in it, but finally we have the possibility to maintain the salient properties of the neuron, even if it is simplified. So we can validate these properties, and then we can use the simplified neuron and build a simplified network. This simplified network is computationally efficient and can be inserted, for example, in a virtual brain, like here, or in the closed loop controller of a robot like bottom right. In the robot, we can also make another step, make it running real time, which means very difficult in software. In some cases, we had to transform it into hardware. That's yet another step. There is a lot of technology in the story, so we can't be happy when we have a model. The model can be very nice but useless. We have to transform it until it becomes useful. Okay, so in the case of the virtual brain, we have transplanted the microcircuit of the cerebellum that is on the top into its proper positioning of the Allen Brain Atlas connectome. You probably know that atlasing is another important story. To work on the brain and work on the models, we need atlases. And the atlases represent our knowledge transposed into a virtual space with respect to the position of neuron, the connection of neurons, the organization of the nuclei. And therefore, if we are able to map the model on the atlas or vice versa, we have an extremely powerful tool in our hand. We have done the job with the Allen Brain Atlas. Other groups are doing it with other kinds of atlases, we'll see. But in this case, we have transplanted into the Allen connectome, and therefore we have the cerebellar module running into the brain of a mouse, a virtual brain of a mouse. Another kind of transplant is here. The model has been transplanted into the closed-loop controller of Now, that is a robot that can be programmed to perform some task. I will show a movie in a while. And it has been connected cell by cell, or better, cell type by cell type, with all the components, the motor planner, the motor controller, the state estimator, the agent, the motor commander, and so forth. So in this way, we have a system in which we have the neurons taken from biology, simplified, put in the controller, running in a behavioral task. Okay? So we've done all the job till the end. Okay, let's see how it works. This I will show later. I show the robot first. Can you try to run the movie? Any chance to have the movie running? Okay, thank you very much, it's running. Yeah. Okay, now the robot has been instructed to perform an arm movement, which means generating a circle following the trajectory drawn on the screen. Okay? What one can do is to give what we call the CRUD command to the planner. The planner sends the command to the motor controller. The motor controller contains the inverse dynamics of the robot object system and generates an action, activating the motors of the arm. Okay, so it is not too bad, but then we make a trick. We put a load on the arm of the robot. Now, the arm of the robot is too heavy, and the arm falls down. So you can say it's trivial, obvious, it should happen, it happens. But in this robot, we have connected the cerebellum to the controller and to the sensory system of the arm. The cerebellum is designed for its architecture. It is the Marr theory of the motor learning theory of Marr. Is designed to generate a prediction of the states that the system will visit during a sequence of acts. It predicts the precise timing of correlated events. This cerebellum is a spiking network, is the one that I showed you, is beginning to receive inputs from the cortex, is the desired command, and from the sensory system is the actual state. Can you play it back, please? Can you play it again? It happens that the robot begins to use the cerebellum. It begins to use the cerebellum, and without knowing it, it is just embedded. We didn't tell him anything. It simply has the cerebellum and begins to use it. The cerebellum has inside the learning rules, which means the ability to adapt the weights at the synapses between certain categories of neurons. And by using the signals that is receiving from the cortex or from the sensory system, begins to change the weights. And the weights change in a way that minimizes the error. So you will see that at the beginning the error is big, so the divergence between the blue and the red line is quite large. But trial by trial, the error is reduced. And at some point, the error will be minimal. Okay? In this way, we have generated a robot with a circuit that operates adaptive learning, an autonomous adaptive learning. So the system is autonomous and is doing what? Is learning to predict the consequences of a command, the sensory consequences of a command. So it's an example in which the implementation of a precise network of the cerebellum is giving added value to the activity of a robot. We are in front of a new kind of plant for the robot. Now, we go to the last point. I hope I have still 1 or 2 minutes. 1 or 2 minutes. This is the last point. It is about the virtual brains. In this case, we're not working bottom-up. We're working top-down. So we're starting from signals that are recorded as ensemble signals using MRI. And these signals generate what we call the empirical structural connectivity matrix that is remapped in the virtual space of the virtual brain. So basically, we remap the connectivity matrix and generate the connectome, and then we put in the proper positions the nodes. The nodes can contain a neural mass, or can contain more complex representation of activity, like mean field models or spiking neural networks. All these things can be put in the nodes. Now, if you run a simulation with this reconstruction, you generate waveforms like the simulated neural activity that you see there. The simulated neural activity is an ensemble of waveforms that reproduce what the brain should do with that connectivity and those nodes. But now the question is it correct or not, or to what extent it is correct? The point here is to match this simulation with some real data taken from the same subject. For example, data taken from a resting state fMRI, generating the empirical functional connectivity matrix. So now the problem, the mathematical problem, is to invert the model, compare the empirical to the simulated functional connectivity matrix. There are procedures of model inversion that can optimize the weights or minimize the distance between the two matrices. What we extract from them are the internal parameters of the system. The internal parameters of the system are those that are of the highest interest for biologists and in medicine, because they inform us about the state of that particular brain. For example, we can gain knowledge, depending on the kind of neural mass or mean field or whatever we put in the nodes, about coupling inside the brain, which means the strength of connection between the nodes, local coupling, which is connectivity inside the small networks, neuronal activity in terms of, for example, spectral bands of activity of the neurons, and excitatory-inhibitory balance, which means how much excitatory synapses or inhibitory synapses are working in the brain of the subject, which clearly becomes critical if you want to understand pathology. So we're beginning to develop case studies on, for example, Alzheimer's disease patients and frontotemporal dementia patients, trying to understand how this data can be extracted from the brain of these patients. Okay. Okay, I think I've given you an overview of all the story, and I go to the last figure, which summarizes all of these virtual brains, robotic controllers. We've also transplanted the network into neuromorphic computers to generate computers that work using the spiking neural networks. One example is a Spinnaker, that is a project run with the University of Cambridge, in which we have transplanted the model into Spinnaker, and now Spinnaker is a supercomputer with the cerebellum inside, spiking. Testing ongoing. Okay. We have transformed the network into GPUs, for example. So we're transforming it into hardware. Very last figure, all of this requires an organization that is implemented and supported by the European Commission, mostly, at least in Europe, in which there are centers for brain modeling, those listed on the left, and there are centers for supercomputing, for atlasing, for data processing, and data banking, and for robotics and informatics. That's all the story. I stop here, and thank to all the people who is working with me on this really big and large-scale project. Thank you very much.
Okay, so today we have two talks on new trends in imaging, and I'll talk about MRI, and Matthias Fink will talk about ultrasounds in general. And then we have talks which are very oriented towards the future about the translation of mechanics and biology. You will see it's quite interesting. And then how to translate the breakthroughs in technology to the market and to the patients. So I'll start to say that we know a lot about the universe, our galaxy, how the cosmos is made of matter, antimatter, dark energy, and so on. And physicists enjoy beautiful theories, and they have also instruments which are key to prove all those theories. But instruments can be used also to generate theories, not only to prove theories. And for instance, this is how the acceleration of the expansion of the universe was discovered in 1998, which can be explained with the dark energy. Unfortunately for the brain, we are not so advanced. We know, of course, that we have neurons, gray and white matter, but we don't have models as physicists have for the universe. But we might have instruments, and I'll talk today a lot about the EASL project that we built at NeuroSpin to have an instrument which is a little bit equivalent to what we have for the universe. So first, very, very quickly, what is MRI in case you don't know? So the idea is to magnetize nuclei, especially the hydrogen nuclei of water molecules, and we can obtain beautiful images of fine anatomy in the brain, for instance, the hippocampus, and because it is not invasive at all, we can even obtain images of fetuses and babies. We can also look at how the brain works with functional MRI. Due to what we call the neurovascular coupling, there is increase in blood flow in activated regions. So this works very well, and we can also, thanks to a diffusion MRI, which I invented about 35 years ago, obtain from the Brownian motion described by Einstein in his PhD thesis, gorgeous images of the connections in the brain. So we should be happy, right? We have everything. We can look at wide connectivity, functional connectivity, and we know that how the brain works, in fact, is a network at different scales of connectivity. But it's not enough. Let me explain to you why. In the brain, so we know that we cannot see everything with neuroimaging. We know the brain is organized in regions and at different scales, down even to the layers and the colons in the cortex. But we know also there are synapses. Neurons are communicating through neurotransmitters, so it goes to neurochemistry and genetics. Unfortunately, with MRI, as we can do today, we are blind to this part of the spectrum. How could we recover visibility there? It's the same as in high energy physics. We have to increase what we call the luminosity of the instruments. That means switch to a very high field, because we know from the physics that by going to the high field, we can have higher signals, which we can trade for instance, higher spatial resolution, higher temporal resolution, or chemical resolution. So this is how the EASL project was born about more than 20 years ago, with the idea to build a magnet of, which is 11.7 Tesla, which is about 223,000 times the Earth's field. So that was a big challenge. And the idea, of course, is not the idea just to make a magnet. Why do we need a magnet or this power? It is to explore this uncharted territory. So we know a lot about neurotransmission. I've shown you some beautiful images of a large-scale brain structure, but in between we have nothing. So if we can address this scale, we can, non-invasively in the human brain, we can perhaps better understand diseases, such as Alzheimer's disease and other diseases. So we go to very, very fine anatomy, and we also can look at new things, discover new things which are not aware of today. So this is how I started the project in 2001 at the CA in Saclay. At that time, the highest field available was 7 Tesla, which is already a lot. I was considered as pretty crazy with this project. But anyway, I knew that I could count on the knowledge of the physicists and engineers from CEA, because they are the ones who build the magnets for the atomic projects first, like, for instance, nuclear fusion, but also for the high-energy physics at the CERN in Geneva. So I went to them and, could you make 11.7 Tesla for me? So initially, they were a little bit surprised. Maybe they laughed. But finally, they understood it was a very serious idea. And this is how NeuroSpin was born, to house this outstanding instrument. So this is the work from the architect, Claude Vasconi. This is how it looks. And each of those arches has a magnet from 3 Tesla up to 17 Tesla, not for humans. This magnet is designed for humans, but for rodents. So we have a complete spectrum of instruments. So the CA physicists and engineers worked out this project, and the idea was to build a huge magnet. So it's, the internal diameter is 90 centimeters. What's also very special is that, I will show you later, it's cooled down to 1.8 Kelvin, and the currents there is much higher than what you find is in standard MRI machines. So I don't have time to go into the details, but the design of this magnet has been imported, in fact, from what has been done for the LHC in the CERN. It's not a solenoid. It's made of a double pancake, and I will show that in a few moments. And, of course, we had to find the money, and we were very lucky to have this project financed through a European French-German partnership with industry and with academic partners from Germany and France. So the magnet was built in Alstom factory in the east of France, where the first French trains were made, where the TGV is made, and when, of course, turbines for electricity are built also. So sometimes, as a joke, I say we went from trains to brains in Alstom. And so this is the design of the magnet based on double pancakes, so not a continuous solenoid, which would save a lot on the quantity of material used to build the magnet. Also, the Alstom staff had to build a very fancy equipment to make those pancakes. So you see, this is how they look. There are 168 of them to build the magnet, and they are stacked like that with an accuracy of less than a millimeter. So it's really a tour de force, and they succeeded. This is the magnet almost at the end, just to scale. When you see the magnet, you feel so small. I mean, it is incredible. And the magnet was completed in 2017. So you see, it took 17 years from the first conception to the final magnet. To bring the magnet to NeuroSpin, so very close from here, about 15 kilometers from here, was not easy because the magnet was so big that it could not go under the bridges. Also, we were afraid about the vibration. So the magnet, in fact, went from Alstom, a factory, on a special truck with two cabins, one to pull and one to push, with 150 wheels. And on this truck, the magnet went to Strasbourg, and then on the Rhine River, on a special boat, going to Germany, to the North Sea, and finally back to France. From the Seine River, it went under the bridges of Paris, was then delivered to Saclay. We had even to move the track of the RERB line, so the train line, at 2 o'clock in the morning to be sure that we could put the magnet there. So it was very impressive. It took 3 weeks, and the magnet finally came to NeuroSpin. I was so moved when this magnet came, as well, of course, all my colleagues. Our dream was completed, if you want to say. So this is the magnet in the arch in 2017. It's a necked magnet. And as I said, the magnet is cooled not to 4.2 Kelvin as current medical magnet, but it's cooled to 1.8 Kelvin. So sometimes I say the temperature is lower than the temperature of the back of the universe, which is 3 Kelvin, as you know. And so that means that we have to produce a liquid helium on site. We have a cryogenic plant implemented at NeuroSpin. And all of this has to be monitored continuously. The magnet is on an external power supply, which is also very original. So we have to be sure that everything remains stable. So we have also to be careful that there is no power outage or anything. So the magnet has to be controlled 24 hours a day, 7 days a week, for more than 10 years, including French holidays, there are many, strikes, viruses, wars, everything. So, so then we had to make it work. It took also some time because we wanted to monitor each step carefully, not to break or to induce a quench in the magnet. So you see we went up and down, and which is possible because the magnet is an external power supply. And finally, on July 19, 2019, we reached our 11.7 Tesla target. That was also a big event for us. But MRI is not only a magnet. We have to use all the equipments, so-called gradient coils, to localize the NMR signals, and this is provided by Siemens. But also radio frequency coils and 500 megahertz, which is a resonant frequency of hydrogen at 11.7 Tesla, is something very special because the wavelength is shorter than the size of the head, so it's a source of big artifacts. So my colleague at NeuroSpin designed some special coils, new encoding strategies to localize the signal. So you see there is a lot of novelty inside this magnet. I don't have time to explain all of that, of course, but some techniques to acquire data in a very fast way to also decrease the amount of radio frequency exposure because radio frequency can heat, so we don't want to cook brain, right? So, so this has to be done in a very controlled way. And my colleague Nicolas Boulan was successful in getting a grant, FET, so grant from the EU, with a collaboration with the key partners in Europe about this. So the first images were obtained 1 year ago at the Halloween time, so we had a pumpkin. In fact, it is a tradition with MRI to have the first images of vegetables or fruits. The images are really gorgeous. This is the image obtained from the machine. And we are not yet allowed to scan normal subjects, normal volunteers or patients. So we scanned a brain from a museum in Paris, and you see that it's very promising. Okay, now we have a magnet, an MRI system, which is outstanding. It's the only one in the world, There is one at NIH at 11.7 Tesla. They bought, they didn't build, but it's not functional yet. I talked to them recently, and they don't have the helium yet. That's quite surprising. So they cannot use it. So what could, I mean, the total cost of this instrument was 75 million euros. I mean, not the materials, but all the work of the people. About 200 people worked around this project. So if we have such an outstanding magnet, we cannot use it just to do a little bit better than other people do, or to do research that can be done with the lower field magnets, just to see a little bit better. So we have to have very futuristic projects about this. And I showed you this picture at the beginning of my talk, but in fact, we don't know the link between right and functional connectivity. We know that the avenues, the highways we have in the brain, but we have no way to watch the information progressing along those lines. So recently I introduced something which I call the relativistic space-time model, and modeling the propagation of action potentials in the brain as a random walk process. Of course, it's not random, but it can be modeled mathematically and physically. But there is an issue. So these are the axons, and again, along those axons, the action potentials will travel, but we know there is a speed limit. It's about 100 meters per second. So because there is a speed limit in the propagation of influx, we have to consider the relativity theory. I don't think many people thought about that. It's like when you look at the sky at night and you see all the stars, they don't light up at the same time. It takes time for the light to come. I think everybody knows that now. It is the same in the brain. So when neuroscientists look at images like that, where you see several areas activated, we think that when you do something, all these areas light up at the same time. This is completely wrong. Each area here cannot see what's going on in other areas at the same time because of the time it takes for the action potentials to travel. It might be hundreds of milliseconds. It's short, but it's not zero. So there is a speed limit. And if we consider that, then you can rewrite the equations of relativity to explain how the brain works. It can go even to the general relativity. So the idea is that if you consider that each region of the brain is activated at a different degrees, it's like a mass in some sort. And the action potentials will converge or diverge from each node along geodesics. So you can rewrite all these equations, and it goes very far. For instance, if you look at schizophrenia, we know that schizophrenic patients hear voices. They do hear voices. They activate the auditory cortex. We talk to ourselves all the time, right? We know it's our brain, which is at the origin. But in schizophrenic patients, we know that the connections are not working properly. So if you work out these time delays using the Minkowski diagram used for relativity in space-time, you see perfectly that there is a disconnection. That means that those patients hear voices, but their brain is unaware that it has been produced a few milliseconds or 100 milliseconds earlier. So they believe that something is coming to the brain, that God is talking to them, for instance. We know also that in vegetative state patients, the connections are getting loose and lower. So this has been published, and using DTI, the diffusion MRI systems that I described earlier, we can visualize those connections. But this model can completely reproduce the pattern. So this is normal connectivity and decreased connectivity in a vegetative state. But this model also predicts that if you have a vegetative state patient and you stimulate the brain at a particular target, you can curve the brain's space-time and restore the communication. So you can have the patients switching from a vegetative state to a conscious state. So we've done a preliminary study for that. Unfortunately, I cannot trigger the movie here, but we have an anesthetized rat here, and we can send a small current in the central median nucleus of the thalamus. I will try. No, it doesn't work, because there is a movie there. You see the animal wakes up immediately. So basically, we have curved the rat's space-time, and we can modulate, we can block this by blocking neuronal swelling using furosemide, or facilitate the awakening of the animal using water, basically. So this is something that we have to look very carefully, because the future is that we might be able to wake up patients in vegetative state. So this is, I think, the vision I have, at least for the research which will be conducted at NeuroSpin. So that's the end of my talk. So if you want to know more, because of course I was very quick on the magnet, so is this paper I published with Thierry Schild, who was a project manager for the magnet. And those are more general public books, most recent one about this relativity theory. So Einstein's error, I will not say Einstein made a mistake. He said he made mistakes. It's a cosmological constant, dark energy, and I explain how this can be used to explain some working of the brain, especially how we interact. And of course, I would like to acknowledge all the people who have been, so supportive for this crazy project. I mean, now in China, in the United States, there are projects to go to 11.7 Tesla or even higher. And when I talk to my colleagues, they say, well, 20 years ago you said I was completely crazy, and you see I'm not. And they said, no, no, you are still very crazy. The problem is that you are very contagious, and there is no vaccine. So that's it. Thank you very much for your attention.
Good morning, everybody. I will speak of ultrasound, and I will speak mainly of what we call ultra-fast ultrasonic imaging. Let us begin by ultrasound imaging. Conventional technique use all the time what we call an ultrasonic probe. Sorry, there are two-time piezoelectric elements, but it's okay. And this probe is made of 100 piezoelectric transducer. And coupled to this probe, you have an electronic which is in this box. How do you play to make an image in conventional imaging? You want to create a very thin beam that explores tissue, and you listen the echo from tissue. To make a very thin beam, you make the assumption that you know the speed of sound in tissue, which is not really true. Once you make this assumption, you can do what we call confocal imaging. That is to say, when you have your array of piezoelectric transducer, you can control electronically each of them with some delay to focus somewhere in tissue. And this is what we call focusing in transmit. Once you have created a small focal spot here, you can also do in reverse during the receiving mode focusing. That is to say, if there is a target, you can put some delay line to make all the echo of this target add constructively. When you are doing this, you are doing what is called confocal imaging, but this is just one beam. To make an image, you need more beam. And what you do in conventional ultrasonic imaging system, you make your beam and you scan this beam electronically, and you change the focal distance. And after some time, when you listen all the echo, you have an image. And because you have to wait all the echo to come back, you cannot have more than 50 to 100 frames per second. All this is based on electronic that use analog device, especially delay line. And this is the way conventional ultrasonic system work. When you look the kind of image you have, for example, you see this kind of image of fetus, or here, and what you see is that in this image there is what is called a speckle noise, full of small reflectors that give rise to echo. And here, in the cardiac imaging, you don't see anything, but in fact there are small echo from blood cells, which are very low level compared to the echo from the rest of the tissue. When you have this, for example, you can just look the echo from the red cell, and it allows you to track the blood motion, and you have this very nice Doppler imaging flow. This is conventional imaging. And people were very happy with 50 frames per second and 100 frames per second. And this was using analog processing. In the 90s, in my lab, we decided to go to a new concept that we called time reversal mirror concept. And this concept was possible only because in the 90s, we arrived at the digital era. What it means? It means that each piezoelectric transducer you have can be connected through an analog to digital converter to an electronic memory, which is very fast. This electronic memory arrives only in this time scale. And when you have this, for example, if you have an acoustic source sending a wave through a complex medium, the time reversal mirror just used many piezoelectric transducers that are microphone first, all the data you receive are put in electronics memories, and once you have done this, you read the electronic memory in time reversal chronology, and now each microphone becomes a loudspeaker. This is what you can do with piezoelectric transducer, and you will recreate back a wave that revive his life. That is to say, the wave will exactly come back to the source. And if you do this, you can destruct a source. And we begin by making apparatus for lithotripsy, to destruct kidney stone or to burn tissue. But this concept allows us to develop this electronic, which was the first electronic in the world to have 128 digital converter, huge memory, and you can manipulate the memory. This was much more complicated than analog processing. And from this, we decide to transform this in a way to create ultra-fast imaging. Imagine that you send just a plane wave on a medium and that there are many scatterers. And if you record in the electronic memory all these scatterers, if you know the speed of sound in the medium, you can do directly in the computer the time reversal and the backward propagation. That is to say, in one shot, you can make an image of tissue. And if you are doing this, compared to the image, which is a confocal image, that takes time because you have to send many, you obtain very quickly, and you can get 10,000 frames per second of tissue, and it's ultra-fast imaging. But when you are doing this, you lose something because you have not focus in transmit. You have only focus in receive by using time reversal. And the image is not as good, but you have ultra-fast imaging. Now you can improve this. Instead of sending only one plane wave, you can send a set of plane waves with different angles. And for each of them, you record the ultra-fast imaging, and now coherently you add this image to mimic focusing in transmit to each point. And when you are doing this, instead of having 10,000 frames per second, you have typically 1,000 frames per second. And when you are doing this, you have an image which is as good as the image that is obtained by confocal imaging. Now, is it interesting really to see the body with 1,000 images per second? When we developed this in the 90s, everybody was thinking it was not interesting to see the body so fast. 100 frames per second is enough. But what we show is that when you have ultra-fast imaging, you can see two things that are not seen usually. You can see what we call the seismic activity of the body. The body is a solid tissue, soft solid, and in soft solid you can have compressional wave that propagate, but shear wave. And shear wave are very interesting because they propagate slowly, and the speed of a shear wave depends on the elasticity of your tissue. And we developed the concept of shear wave elastography that has a lot of success today. The second thing that you can do when you have 1,000 images per second is to revisit Doppler. Now, if you look just the motion of red cell, because you have so many frames per second, you have a sensitivity to very small vascularization. And you can do functional ultrasound imaging in the brain. Let us see the first thing, shear wave elastography. As I say, two kinds of waves propagate in the body. Compressional waves go fast, 1,500 meters per second, and ultrasound are only compressional waves in the body. But in the body, you can have shear wave of low frequency that propagate at a speed which is quite small. Shear wave typically propagate at speed between 1 and 20 meters per second, and it depends on the shear modulus of tissue, which is exactly what a doctor feels when he's making palpation. Now, you can use the ultrasound in ultra-fast imaging to observe propagation of shear wave. How shear wave can be produced. This is one technique, and we developed this technique in the company EcoSense that gives rise to FibroScan to measure the elasticity of liver, but it is not imaging. Now, you can use the shear wave created by cardiac motion and by when I am speaking, when I do vocalize. And you can also use what is called radiation pressure due to ultrasound. But the interest of this is the following. Let us look here. This is an image of a phantom where you have an hard inclusion. But ultrasound does not see the hard inclusion. There are small scatters everywhere, and you don't see. But if you look the movie I showed just before, if the movie come back, the movie of a shear wave that you obtain contain an information on the speed of a shear wave. And if you just look what is the speed of a shear wave at each point, now you have this image. And from an image where you don't see anything if you have at low frame rate, suddenly ultra-fast movie, if you focus just on the displacement induced by the shear wave, you have a shear map, shear elasticity map. And from this, you can do very quantitative things. And when we have done this, we create a company, Supersonic Imaging. The company developed the Explorer, and the company was sold to an American company, Hologic. And in fact, now everybody is trying to do this. Why? Because if you look, for example, classical reflectivity image in breast imaging, you have the impression in these two cases you have the same kind of pathology. But now, if you look the shear modulus, you see that one is really very hard and the other is not hard. And you can make discrimination. For breast imaging, it is a big success. It is valid for thyroid, for prostate, for many things. And now, if you look liver, for example, depending on your liver, the color you have is different. That is to say, the elasticity is different, and you can follow pathology with this. But this is one application, shear wave imaging, and now all companies are putting shear wave elastography in their device. And now the second application of ultra-fast imaging is to observe blood flow. Instead of having classical technique to observe blood flow, when you have sequential imaging that give you, for example, on the rat brain, an image like this one, now if you used ultra-fast Doppler by using nearly 10,000 frames per second, you can quantify very small blood flow motion, and suddenly you will have a Doppler image which is quite better, where you see the vascularization of the brain. Of course, this is on small animal, but you will see that this can go one step more. You have power Doppler, which is enhanced by ultra-fast imaging. But now, for example, if you take a small neonate, a small baby, through a fontanellum, and this is what Michael Tanter developed in his group, you can make an image of the vascularization in the brain. And this image, you have many frames per second. That is to say, you can follow in real time during the cardiac cycle how the blood is moving back and forth. And it gives you a lot of information. This is quite interesting functional imaging, but it is limited by diffraction. The size of what you can see depends on the frequency of ultrasound. And typically, you can have sub-millimeter scale, typically 100 micron resolution. Now, an idea that we developed in our lab and that Michael Tanter has. Well, this is the same, just to show for the fontanellum the technique that uses a probe that can move to explore the brain of a baby. Now, as I tell you, the image you obtain are limited by diffraction, typically some 100 microns. But there is an idea that we developed with Michael Tanter and Olivier Couture that is an idea to beat diffraction limit. And the idea was to inject a small number of microbubbles in the blood and to look, because we have 1,000 or even more images per second, you can look the position of each bubble. And because at each time there are only a few bubbles in your area of interest, you can learn how to find the localization of each bubble with a very good resolution. You are not limited by diffraction limit, which is the distance between two spots. Now you can look this. And because you have this 1,000 times per second, you can follow at each time the motion of a bubble. And when you are doing this, instead of having this image of a brain, look what Michael Tanter observed with his group, you have this image. And suddenly you can see all the bubble position during time, and you can have a map of the vascularization of the brain, which is ultra resolved. Typically, you have 10 microns, even if you use ultrasound with 200 microns of wavelength, and you do super resolution. This has a lot of interest, and the group of Michael Tanter is pushing this for many applications on the brain. Now, before the end of my talk, I want to speak of another application of ultrafast imaging with ultrasound. When you use an ultrasonic probe, usually you put the probe on the body, and you need to put some gel, some gelatin, to have contact. 10 years ago, I was thinking that in the bumper of cars, people used transducer, ultrasonic transducer that work in air to look the distance of obstacles. And we decided to push a crazy project to make an ultrasonic probe in air. And we decided to create an ultrafast imaging system made of piezoelectric transducer working at 40 kilohertz. This is exactly what you have in the bumper of your car. And we put 100 of these transducers in this panel. And you put the panel in front of a guy, and the guy has cardiac motion, he can discuss, and you just observe the echo of the chest of a guy. And we used ultrafast multi-illumination algorithm where we send different plane waves, and we make 1,000 frames per second of a human chest. And we can make an image on typically 1,000 points on an area of 40 centimeters by 40 centimeters. And because it is ultrafast Doppler, we have a sensitivity of displacement of microns. Now, let us look what happens when you put a guy just in front of this. If you put a guy just in front of this, you know that cardiac motion is quite big inside the body, but it's the motion reveals skin motion. And if you observe micron displacement, you can see during the cardiac cycle the activity of a guy. It is a non-contact device. The body is just in front, and you see this. Now, when you see this, you can look now every millisecond what is moving on your chest. And if you look this, you can see that the mitral closing, you can see the aortic valve opening, and you see, in fact, the kind of shadow of the activity of your heart without touching the guy. And you can play with this, and you can look on one point, and you can compare what you have here with the electrocardiogram. You obtain much more information, aortic closing, and you obtain this without any contact. The same apparatus, if you are in front, you can see your cardiac activity because it's ultra-fast imaging. The same apparatus, now you can ask to the guy, say, ah. And when you are doing this, in fact, your vocal cord emits a sound which goes through the pulmonary area. And you know that a good doctor, usually he puts his hand on your back to see, when he asks you to say, ah, he see where it vibrates. And in fact, you can directly ask people to say, ah, or something else. Huh? It depends on the country. Some guys say 33, some guys say something else. And you can look an LT patient in the face, and you see here that sound is going through one lung, but when you are in face imaging, there is the earth is a problem. But when you look the back image, you see the shadow, the acoustic shadow of the lung. And if you have a patient which are obstructive pulmonary disease, it's completely different. And we are playing with this, and we just create a company, Austral, to make this panel, which is an ultra-fast imaging without any contact. And we think that at the hospital, when you come to the urgency, you don't need a doctor to do this. It is just something without contact. You put the guy, he just say, ah, and you look also his cardiac, and perhaps you can have a detection of what is wrong on the patient. It is not as fine as contact technique, but contact technique need a special doctor that is an echocardiographist, which is not so, which is difficult here. It's something that can play a role in telemedicine in the future, because you can go, in 5 seconds you have your movie. Well, some AI can do the job. Okay, just before the end of my talk, I want to ask what next? As I tell you, we developed this concept of plane wave imaging, which is now all the company want to put this plane wave imaging technique. And you know that you send plane wave, you listen the echo, you put in memory, and you send different plane wave with different angle. When you receive all, it is a huge amount of data, and we call this the reflection matrix. When you have a reflection matrix, the simplest thing that we were doing was to take this and to create a confocal imaging. You can mimic focusing in transmit at one point and focusing in receive and move this, but this is done in the computer. This is the confocal imaging based on this, which is ultra fast. But now you can do something completely crazy. You can, from all this data, make mathematical transformation to mimic from this data focusing in transmit in any point, but at the same time, instead of focusing in receive on the same point, you focus on another point. That is to say, you create virtual source everywhere in your data, and you create virtual microphone everywhere. That is to say, you can, when a wave propagates in the medium, look with this virtual microphone at any time how the wave propagates. And you can do what we call the self-portrait of the ultrasonic wave in tissue. And this is relatively new. I will say it is even completely new. You have your data in the computer. And in the computer, you mimic how you focus on this point. And you see if you focus well or not well. If you do not focus well, it is that the speed of sound you put is not the good one. And if you do this for each point, from all this, you can deduce a map of the speed of sound in tissue. It is not speed of shear wave, it is speed of compressional wave. And in the fat, the speed of sound is typically 1,400 meters per second. In muscle, it is higher. And we begin to have the first image of the speed of sound in tissue, which is a new biomarker. And from this, we can correct the image from all aberration. And I will stop on this to tell you that ultrasound are interesting because they are not expensive, they are easy to use, and ultra-fast imaging change many things on this, and perhaps airborne ultrasound, which was completely crazy idea, perhaps it can be also a very interesting tool to improve diagnosis. It will not be radiology, it will be an imaging system, not for radiologue, but for the urgentist, the generalist, I don't know how you say this, and even for, you don't need to be a big specialist to use this. I will stop here. Thank you.
thank the CAETS organizers for giving me this opportunity to share some of my thoughts with this distinguished audience. I represent the Indian National Academy of Engineering, but also my own institution, Indian Institute of Science, which is located in the southern city of Bengaluru, previously known as Bangalore. You can see I'm a mechanical engineer, and in some sense, machines have some kind of an existential crisis, because we hear machine learning all around us now, and we are not talking about the good old machines, but computing machines. But today I want to hark back to the good old machinery, or may I say eternal machinery, that's going to be with us for as long as we are going to be. And since Academies are meeting here, I would like to remind ourselves that recently the US National Academies of Science, Engineering and Medicine produced a report to identify physics of life as a separate discipline, just as physics of particles and nuclear, plasma physics, all those were there. It's on the same level of physics of life. And physics is there, mechanics, which is a branch of physics, and engineering is not far behind. The micro machinery that I'm going to talk about, you can imagine this as the workshop for looking at biological cells. The stage is set with the previous two talks about the imaging, but let's see what we can do if we can literally grasp a cell. Of course, not with your fingers, but you need micro fingers or micro hands to do that. What can we do if we are able to do that is what I'm going to talk about, which I call, or people call, mechanodiagnostics. If you open the hood of a car, a mechanic would be able to see what's wrong or what's right. And in a similar way, if you don't have to open up a cell, it's a minimally invasive or non-invasive way of diagnosing a cell, if it is normal or not normal, when the cell is still alive. I put here a view of biological cell. It's a tiny water balloon filled with granular matter. You might think it's an engineer's view, but actually it's not. Not only an engineer's view, it was said by one of the famous biologists of all time, Francis Crick. This is a quote from his paper where he says, if we were compelled to suggest a model for a cell, we would propose mother's work basket. For that time, it's okay. You can call it father's work basket also. A jumble of beads and buttons of all shapes and sizes with pins and threads in good measure, all jostling about, held together by colloidal forces. What he's saying is cell is very complex and a lot of things moving around. It's a granular matter. And he did these things to cells, what I've highlighted: twisting, dragging, and prodding. That was, he was actually mechanically probing biological cells using what we call today magnetic twisting cytometry. That is, he used magnetic forces at the time. And this was his paper. If you look at the date, it is was received October 5th, 1949, published 1950. That was just 3 years before Crick and Watson discovered double helix structure of DNA. So he was looking at physical properties of cytoplasm at that time. If you take 1950, doing this is very early. There are papers going back to 1926, more than 100 years ago, where people looked at the physical properties of erythrocytes, red blood cells, and looking at extensibility of protoplasm, and also the tension at the surface, meaning membrane, cell membrane stiffness. And I also want to say that if we are able to do that, what do you want to gain? And this will be mechanodiagnostics or biomechanical assays as an alternative or alongside biochemical assays that we do today. And why do you want to do it? As an engineer, I would say it will be less expensive. We need much smaller sample. Just a drop of blood will have a lot of cells, if you're looking at cells that are there in blood, or small tissue sample. And it will be quicker than biochemical assays, and also it will be eminently portable, so point of care, or even could be even wearable. Cell biology and mechanics of deformable solids were born out of the same mind of Robert Hooke. He looked at the cells, the cork, under the microscope and coined the cell and formed the basis for cell biology. And with this interesting anagram, Wood tensio quiz, he also gave us what we call the linear elasticity or Hooke's law, which relates stress and strain using Young's modulus. So we have chem, which is also an anagram for mech. And what we can do with biochemistry, we can possibly do with biomechanics. Another thing to note here is an engineering way. Whenever we want to measure something in engineering, mostly we don't measure that thing, but we measure something else to relate. For example, stiffness. I would measure force and displacement. If I know the spring constant, I divide force by displacement, I get that spring constant, the stiffness measure. But in biology, whenever you want to measure something, you look at that particular thing and measure that particular protein and quantify what you're looking for. It's not indirect measurement. Whereas if you want to take an approach from engineering, we can do this indirect measurement. If we do it well and for a long time, it could eventually lead to mechanodiagnostics. Here are a few questions and problems that I will describe briefly. Because scientists usually ask questions, engineers pose problems. What I have here is a mix of both. How do we measure force? Again, indirectly. And what do you make out of mechanical response of single biological cells? And what do you do with that? What are the biological implications that you can infer from the mechanical behavior of single cells? The tools that we use for that, we call them compliant mechanisms. They're deformable structures that can be made because they don't have any joints. We can make them at any scale. What I've shown here is various materials, including silicon, a lot of micro devices that are made with these deformable structures. But you can also use very flexible polymers and make tiny grippers. In some view, in one way, proteins are also nanoscale machines. So here are two movies for you to see how we can use these compliant mechanisms or deformable structures to make a gripper on the left side. On the right side, we have something where I can actually roll an object, and that object can be a single cell, as we will see. Here, with these things that are made with photolithography, you can see at the top where a breast cancer cell, MCF7, is being handled, grasping. We can squeeze, we can pierce, we can roll, we can peel, bend, and probe and stretch a single cell many ways. And in the previous talk, we heard how doctors palpate abdomen. We do that to the cell to see what is normal, what is not normal. And this is our setup. Of course, there is a microscope to look at things. And we have an XYZ stage that probes these tiny grippers or graspers, or the tools that we make, so that we can press it, squeeze it, bend it, and see how it feels. And here is something. You don't have to have trypsinized cells that are suspended. You can have cells that are adherent, which is what the native way of most cells, other than those in the circulation in blood. So here we make an accordion type flexible structure made of SU8 polymer using photolithography. We can stretch and compress if the cells are attached to these beams. And how do you know that they will attach? We just take a large array of these cell structures and seed the cells with very low density. Somewhere they would attach the way you want, like this. If there is a cell that is attached to two beams, if I stretch and contract, I would be able to do that experiment on the cells at scale. And this is something that we can do very easily and put the cells on these substrates and do that. And to our surprise, our device's stiffness was so low that cells themselves were able to move these beams. That is, we didn't have to do. When they stretch and relax, as we say, it actually applies forces and we can measure it. So we have these beams that you see in these pictures where what in the bottom, I'm showing the nuclei of the cells. And you can see when I'm undeformed on the left side, middle is stretched and compressed. You can see the nucleus also is changing its morphology. And this is what I'll be talking about now as an example. This is real time. This movie is playing and the cell actually divides. When it divides, it detaches. At the time, there is no force that beams have, and we can actually measure over a period of time. And this is just a force plot. We are measuring force in micronewtons over about 12 minutes, just at the time these cells were this cell was dividing and then reattaching. We can measure the force that they are applying on the device. And how flexible is a biological cell? I just want to tell you before I proceed. So let's say we have this pen that CAETS organizer has given us. There is a spring in it. How flexible is it, or how stiff it is? The way you do it indirectly, if you want to know stiffness, you say to compress it by 1 centimeter, we'll be applying maybe a couple of newtons, let's say 1 Newton order of magnitude, then it will be 1 Newton per centimeter stiffness, that is 100 Newton per meter. In comparison, a biological cell stiffness could be as low as 0.001 Newton per meter. That is, you need only millinewton to displace it by 1 meter. This is one of the most flexible things that I have come across as a mechanical engineer. And measuring this stiffness was done as early as 1939 by using a simple lever, this pivoted at the point you can see over there, and stretching a cell. This is 1939, and they arrived at the stiffness of 1.4 millinewton per meter. Of course, they gave in dynes and centimeter, converted, that's what you will get. So you can do that right on the dot. Today we measure the cell stiffness to be of the same order. And here is our gripper that's made of a combination of a polymer that is stiff, bigger device, and a smaller device made of SU8 for the micro gripper. And measuring displacement, we don't measure where the cell is because that's already occluded, very crowded to see the displacement properly. We amplify that somewhere else using what you see, this device over here. You don't see much displacement there, but you see a lot more displacement there. And we measure that amplified displacement so our sensitivity increases more like a spring balance. And of course, it's not as simple. We have to solve an inverse problem when you are measuring displacement multiple points and infer forces. It's called Cauchy's problem, French mathematician. And we do that. I don't want to go into the details of how we do it. It's an inverse problem, and it's ill-posed as usual, and you have to do some calculation over it to determine properly. But the end of it is vision-based. We are looking at the displacements of our tiny springs, and we know the stiffness of those tiny springs. We can infer forces. We can just use a digital microscope or a microscope of any kind and measure displacements and do computation, solving this Cauchy's problem elasticity to infer the force and hence the stiffness. Now I will tell you what we make out of that mechanical response. Let's say I have two cells. It's just a schematic to show that there is a nucleus with a nuclear envelope and a cell with the cell membrane, there's cytoplasm, nucleoplasm. Let's say there's a normal cell and an abnormal cell. I hold both of them and then look at its bulk stiffness. And we can do, right now we are doing stiffness, static measurements, but we can do strength and stability and viscoelastic behavior, frequency, and all of those we can do, but I'm going to talk about only stiffness that we have done a little bit. Normal cell, if you do, it may be more flexible than a cell that is abnormal. These things have been done on blood cells for a long time, including last decade or so as well, many people looking at it. If we know the bulk stiffness is different, is that enough for us to do diagnosis? Definitely not, because we need specificity in biology. So if stiffness is different, how much it is different? Of course, we can quantify. We can quantify just like ultrasound would quantify at the tissue level, we can at cell level. And in what way is the stiffness different? Is it in compression? Is it in tension? Is it in shear? Or some other mode? And also, more importantly, what changed the stiffness? Is it organelle? Which organelle of the cell contributed to the change? And that's what we do, the elastography for a single cell. If you hold the cell in multiple ways, let's say I have a schematic again here. Let's assume that compared to normal cell on the left side, the cell on the right side is not normal. Its cell membrane stiffness has gone up, and nuclear envelope or nuclear stiffness has gone down. They may both give you the same bulk stiffness. How do you tell them apart? Here is where we solve another inverse problem, which is called Dirichlet to Neumann map, where we look at, for a live cell, the forces and displacements on the boundary, and then infer what is inside. Like elastography for a single cell, of course, we are making a lot of assumptions here that it is isotropic and so forth, right? But that may be even a reasonable assumption. But we are doing this Dirichlet to Neumann map by considering several sets of force and displacement on the boundary. This is not the number of sets. If I say it is not like machine learning, we have to do a lot of them. It will be just 10, 20 of that kind. And it is just an experiment, a plastic thing. There is a stiff inclusion. If you close your eyes and feel it only on the boundary, can you tell which one of the inclusion? You can obviously tell. But if I ask you to tell me the shape of that inclusion, like it is seen here, barely, and with computation, we can actually predict that. Again, this is another inverse problem. We are able to get this elastography inside. So based on that, we can say probably which organelle contributed to that. Based on this, now, how do we guarantee uniqueness here? We take several data sets, like I said, 10s, 20 maybe, and we establish uniqueness of this elastography inside. The last part, what are the biological implications? For that, I'll tell you about one study that we have done where we are looking at the effect of hepatitis C virus, HCV, on the liver cell hepatocytes. Here we have a control which is normal HUH7 cell line, and the one that are HCV replicon cells which don't have the structural proteins of HCV, but non-structural proteins is easy to work with, not infectious. We work this. In the few slides from now, the gray means control, orange means replicon cell that are affected by HCV virus that does not have structural proteins, but it has the other things. The first thing we noticed with confocal imaging is that the nuclei of HCV replicon cells look larger. I'm showing one image, but we looked at several of them. And also, if you look at the side views, it actually thicker. So overall, the volume of the nucleus has increased in HCV replicons. You see that, and you think like a mechanical engineer, and then say, what might make a nucleus bigger because of this virus? And we did, of course, some of the AFM studies, atomic force microscopy, to look at them. And before that, we looked at the projected area, the volume of the nucleus, and also eccentricity of the nucleus. We find that HCV replicon cell nuclei have larger volume, and their eccentricity is a little bit less. What might cause that? So we looked at a mechanical model. When a cell is adherent on a substrate, the nucleus left to itself would have been a sphere, but now it is pushed down by the cortical actin tension of the membrane when the cell stretches. And so this particular sphere becomes more like a pancake. And we made a simplified model. Engineers love to make simplified models to understand, just like physicists do. And here it's between two plates. If you squish the nucleus, which is sphere, and we arrived at two, in fact, this was a tire model that was used, automotive tires. And we used that model to look at two non-dimensional parameters that kind of relate this shape of the nucleus to what is the cortical actin force, F, that is acting, and also nuclear envelope stiffness. And there is, of course, the pressure, osmotic pressure, chromatin pressure, microtubules, all of them will be working on the nucleus. And in spite of all that, it's actually becoming bigger when the HCV is replicon. We can infer the elasticity modulus for the whole thing. And of course, we confirmed this with AFM, atomic force microscopy techniques, with you can see the number of cells, 21, 23, of that order. And we find that HCV protein nuclei have reduced stiffness, and cell stiffness is increased. So here we're looking at it. Then we want to see what proteins might have been implicated in this. Normally you do a array of protein analysis and then identify proteins. Here we didn't have to do that because we inferred mechanically that cell membrane is becoming stiffer. That could be that cortical actin, beta actin could be upregulated, and nuclear is becoming larger, so its envelope has become more flexible. And with that, we could go and then look at lamin AC proteins which are there in the nuclear envelope. They are downregulated. In fact, they were. We did the gel electrophoresis also, and also tagging that you see here in the middle one. The lamin AC proteins are downregulated in replicon cells, and the actin, beta actin, is upregulated. Both of them we could get that mechanically first and then verify with biochemical assays. And now, whenever we talk about biology, we say there is a lot of heterogeneity. But then one thing we understand now is that biological cells cannot escape physics. Even though these nuclei here have lots of different shapes and sizes, very heterogeneous, but if you were to plot their volume, projected area that you see, but also overall surface area you can compute using confocal images. And because we have this analytical model, these three things, because the shape is like a pancake of a squished nucleus under the actin pressure, there is a relationship among these three physical parameters: volume, projected area, and surface area. They all lie on a surface that is determined by the mechanics of the squished nucleus. And we can see that different types of cells would lie on different places on this surface. And we also defined two non-dimensional parameters which indicates the flatness of the squished nucleus and the size factor, how much bigger it has become, the tau and lambda. And they parameterize this surface, and based on where the cell nucleus lies, we can say what type of cell it is. And we have done it with 5 different cell lines, and each of them, as you see at the bottom, they all have their own place. In fact, with this measurement, we are now working on to tell the phase of the cell cycle as it goes from G1, G2 and other phases. We can tell by just looking at these physical parameters. We didn't even have to go to the stiffness and other things yet. So heterogeneity does not matter because laws of physics dictates the shape. Just to conclude, mechanical response can be measured with miniature tools. Of course, it's not as non-invasive or minimally invasive as ultrasound imaging is, but there are times when you take biopsy and things that are in circulation in blood can be easily accessed, and we can do that. And when we go to dynamics, maybe there are other things that we can look at, cell level elastography, and we can establish specificity with, again, computation being there, and we can understand the biological mechanisms. In the case of HCV, my colleague Professor Mitra Das, who is a biologist, told me that, okay, you are now telling me the nuclear envelope is becoming more flexible, and he noticed that in his research, he had noticed the proteins that are not in the nucleoplasm are there when HCV affects hepatocytes, and what are not in cytoplasm are there, meaning that virus using this mechanism of disrupting the nuclear envelope so that it can have traffic across the nuclear envelope to do its job. And of course, we can intervene mechanically in many ways, and of course, the anagram of chem and mech. And these days I say that there's a whole new meaning to when we say life is very mechanical. So I'll stop there. Thank you.
Good morning, everyone, and I want to switch now to looking at some of the technologies that you've heard about today and yesterday too, and how you're going to get them into the market. Is the market ready for them? And so the Australian Academy of Technological Sciences and Engineering's mission is to help Australians understand complex technologies. So we undertook a 3-year study to look at the new technologies that were coming in, being developed by our local industries and our startups and in academia, and assess the country's readiness to adopt them. So this study was funded by the Australian Research Council, so we thank them very much, and the methodology developed by ADSI. And I'm talking today about the application to health, but we did apply the technology to several sectors. So Australia's healthcare system has increased the life expectancy of people. We're incredibly unfortunate in Australia to have one of the best health systems in the world, and it's improved people's health in the time that it's been available and their quality of life. But there are still inequalities between the metropolitan areas and the people who live in rural and remote areas. Now that the healthcare system around the world have been stressed dramatically by COVID, by the increasing incidence of lifestyle diseases like obesity and preventable chronic diseases. And so the problem is it's still focused on cure rather than prevention. It still relies on face-to-face and long waiting lines in hospitals until recently, until the pandemic allowed some telemedicine. And it still communicates through outdated technologies. So how can we take best advantage of the existing and emerging technologies and kickstart the shift into a digital approach? Currently, the industry trends are, particularly with the younger generation, is that all solutions will be mobile, and the personalization can be driven by genomics, and therefore they want personalized medicine. They want to know something that relates to them rather than to just the general public. Point-of-care diagnostics improve public health, and we saw that a lot through the COVID pandemic. And machine learning and AI are also very important in this area. Most of our young people, and most of us, I think, are using wearables and the Internet of Medical Things. So the Academy looked at the industry, at the readiness over with a number of parameters. And these were the infrastructure and systems readiness, the skills availability, social and ethical readiness, economic and commercial feasibility, and the policy regulations and the readiness there. So we need to take advantage of all these new technologies and the efficiencies they bring, and to change the focus from curing disease to prevention and staying well. We spoke to about 200 different organizations in the healthcare, biotech, from industry, from research, from government, and we ran a series of workshops and round tables, and overall they said that good work's already being done, but there are some areas that need attention. And so these are the results that we obtained. And you can see the scale at the top. Oh, sorry. There it is. You see the scale at the top there. And then we found that some areas were about half ready. It was interesting that in most cases the social and ethical readiness was ahead of some of the other areas. So I think this was a really simple way to look at whether the country was ready for these new technologies. So in order to address these issues, and for Australia to be ready for the opportunities presented by its technological solutions, ATSI's developed these robust and forward-thinking, achievable recommendations for the stakeholders involved. And they're the government and the industry and the research organisations, and also the training organisations. In ATSI's vision for 2030, there are no more paper files. There are universal, secure electronic records. Those records are going to be available for everybody. And our health system at the moment is divided. And I know that's not the same with all countries, but we have a federal Minister of Health, and his responsibilities are different to the states who are responsible for the hospitals. And so there's that division. The states all have different levels of digitization, and they don't all talk to each other. And so it's going to be really important that health professionals can share the patient information, they can share data, they have appropriate protection, and they are able to improve outcomes. So that's the first area, is it must be digitized. Analysis of the big data is going to enable an accurate monitoring and informing and enabling governments to act early, and to invest in large-scale preventative or control measures for emerging lifestyle and infectious diseases. So we need to change our healthcare to digital. We need to change the focus to prevention and wellness. So there's a social license at the moment for electronic health records, but the problem is it's still not there yet. And it's not there yet because people are nervous about cybersecurity, about privacy, and so we are recommending the development of a national framework to improve health data privacy and usage. There are still questions around the best way to integrate novel, mobile and digital technologies, as we need to connect those into the main health system through phones, wearable devices, and to the GP's clinics. General practitioner, sorry. The use of remote consultation is now beginning to open up better, and it's changing the treatment options for medical specialists. And you saw yesterday in the videos, the winning video from India, where they're using telehealth very effectively. We in Australia haven't been using it terribly well until the COVID pandemic. It wasn't reimbursed, so there was no incentive for the medical profession to use it. And so they preferred to have people to come in because that's the only way they could charge the patients. However, the national workforce needs to be supported. I am involved with a company, which I'll just talk about a little later, which had some technology, and the workforce just wasn't ready to pick it up. And so we actually need to change the training of our medical people and the rest of the allied health workforce. And I recently have been asked by one of the local universities to help them change their medical degree and to look at what areas they needed to include. And that included more digital training, more understanding of how digital records would work, but also training in genomics and genetics, which a lot of doctors had not been having. And then there was the idea that micro-credentialing would then allow patients, doctors, to come back and learn at the university on specific topics and specific new technologies. So equity of access is the other area, and this picture shows you just how remote some of these places are. I was in a small town in the north of Queensland not long ago, and there was a 200-kilometre drive to the nearest pharmacy, and the medical professionals were even further away. It's a real problem in Australia, where we have such a big country with a small population. Oops. So these are the recommendations that ATSI made. Transition to interoperable health records. Improve the equity of access to healthcare through technology. Support the future and existing healthcare workforce in the transition, and to provide targeted support for a thriving health tech sector. And that was really to provide more support for the incubators, the universities developing technologies that are planned to be in the market in the next little while. So the COVID pandemic changed things for us. One of the things it changed was that we could the telehealth services, I mentioned that before, and that's now accepted, reimbursed, which is the important issue, and is now available and being rolled out into more remote areas. So this will begin to provide equity for patients in remote areas. The digitised healthcare system can better identify and monitor COVID-19 or any other pandemic in real time, and that has really been valuable. Agile manufacturing and supply chain support that was considered a real problem during COVID because we closed all our borders, not only the external borders to the rest of the world, but the internal borders between states. And so if there wasn't supply in a state, it was very difficult to get the products that you needed for diagnostics and for the medical profession. So I'll now turn to a couple of case studies that will just give a bit of colour to what I've been saying. FitGenes is a company, and I am involved in that company. It's a DNA-based health tech company. The company was founded in 2009 by an engineer, actually, who decided that there needed to be a better way to stay healthy and to get information to stay healthy because both of his parents died very young. And so he set about looking at what technologies he could use, and he developed the first, or one of the very first tests in the whole world that would pick up a few genes. It was just single nucleotide polymorphisms, so not a complicated technology, but he looked at the genes that you could alter with diet, with exercise, that you could up and down regulate because you could take simple supplements like some vitamins. He was ahead of his time. The medical profession didn't want to know. Most of them were extremely skeptical. And in fact, it got taken up by some of the people that did take it up were naturopaths, which was interesting. So people were going to their naturopaths because they weren't getting the right information from their doctors. But the problem with doctors is that they weren't compensated for keeping people healthy. They're not compensated to keep people out of their office. They want people to come in so that they can charge them. So it's important that we look at what the recommendations we make to the government to make sure that that compensation is part of the equation. So the company now provides a lot of genomic profiling for a number of areas. And so, as I say, it was an early leader in genomic testing for wellness, but it wasn't accepted by the market. The healthcare system wasn't ready, and we had a lot of skepticism. So I'd like to talk now about another company. Now, this is a brand new company that is in one of the incubators in Brisbane. It has a unique clinical trials platform. It's been developed by AI specialists, and they're not medical people. And so they've had a different look at how clinical trials are recruited. One of the biggest problems with clinical trials and getting new drugs onto market is the expense of recruiting patients. And sometimes that can take several years. And that's a real problem for a pharmaceutical company or even a research group that wants to recruit patients into a trial quickly. So this technology allows them to recruit patients through looking at the web and finding people who are searching a particular disease or who are looking for a clinical trial. And so it's looking for patients that are not on anybody's databases, or not necessarily. And they bring them in. The medical jargon in the consent form, etc., is transformed by the AI to very simple language, so that it's in plain language that anybody can understand. There's a participant eligibility form on the site that allows people to self-select. And so the people that are coming to the trial are already curated, and they won't require, obviously the researchers involved in the trial need to interview them, but it's not that very large database and a lot of phone calls. It's a quick way of bringing on patients, many of whom are not on these databases. And they've got a lot of skills with digital campaign optimization. You can see the, well, not very well, but that's the dashboard. And what that dashboard does is it allows the people who are recruiting the patients to see at a glance the distribution, whether they've got the right distribution, male, female, ethnicity. So they're looking at age groups so that they can see whether they're recruiting the right patients, and they can alter their advertising accordingly. This allows multi-site management, and it gives real-time insights. And this is how it works. It starts with the plain language, then it has digital marketing schemes, strategies, and the people in the company are all very skilled in the digital marketing area. The automated eligibility is all done by AI, and the participants are pre-qualified. And that compares with how you do it normally, where a hospital will have a database and they start calling people, and it takes time. However, it is a disruptor, and some of the roadblocks include the medical competition, and that what they found in America as they started to expand into America is that the doctors didn't want to release the patients who'd found the trial on the web. So they didn't want their colleagues to take the patients because of the probably remuneration again. And also identifying the right customer for them in the process was also an issue, because initially they went to the end users, but that was then cutting across the CROs that normally undertake this business. And now they've pivoted to work with the CROs, and that was working very well. So, in summary, each country's got a completely different structure, and their health systems are different, and they all face different barriers to translation of novel health. But ATSI's methodology can be used to understand anybody's technology readiness, identify barriers to adoption, and to assist governments to put measurements in place to accelerate active uptake. And we'll be making our technology publicly available as soon as it's ready, so that anybody can use and adapt it to assess any sector's readiness for future scenario. We've already used this technology, this methodology, for health, for transport, and for the circular economy. So please contact us if you are interested in your countries, and we'll provide that when it's ready. Thank you very much.
Morning. It is a pleasure to be here and to speak to you on virtual brains, how they are constructed, and actually where we want to go with them. Personalized virtual brains is one of the key elements that are actually characterizing the virtual brains as a brain model. They are being derived from individual, non-invasively obtained brain imaging data, typically MRI, but also other modalities can be used in order to contribute to that. And the vision is essentially that we are able to capture inter-individual variability, in particular pathological aspects, as they are represented, for instance, in epilepsy in the epileptogenic zone, that we are able to capture them in the brain model, being able to make predictions that are clinically meaningful and that can be used in diagnosis and therapy. The vision would be, in the long run, for every pathology, a virtual brain for every patient. And for this, I'd like to get you in a particular mindset. What you see here is a representation of a figure that resembles actually what my colleague Egidio D'Angelo showed yesterday. On the left-hand side, you see essentially the generation of elementary causes. This is the organ, in this particular case, the brain, using certain mechanisms in order to generate activity on the source level that is then being accessed by us through sensor signals, as you see represented here by the observed data. This we are trying to capture this through mathematical models or some other forms of symbolic representation in order to formalize a causal hypothesis that is actually representing the generation of these signals. In order to analyze this, we typically use data features. Yeah? So this may be a cross-correlations, so different forms of metrics. Here you see a representation of a functional connectivity matrix that has been also showed multiple times throughout this conference here for optimization. So we are working on this level of data features that we are interpreting in order to try to understand the underlying causes. So we are actually performing an inverse problem from the level of these data features. We are trying to infer what actually the elementary causes are of the dynamics. Yeah? So we are having a causation and inference loop. Yeah? And I want you to keep this in mind, because for the forward problem, in particular when dealing with a virtual brain, but actually any form of modeling, we are assuming a causal hypothesis, that is a mechanistic representation, generating this type of data, or typically in a reduced case, the relevant data features. Clinically relevant, however, is actually the inference, the inverse problem, the inference that you perform on the basis of these features. This is what we use in applications, because we actually want to know the cause of a particular disease or disorder. So we have heard a lot about the mechanistic forward modeling. The inference, when linked to a modeling approach, and modeling is just a formulation of our understanding, our knowledge on the causal hypothesis that analyzes the pathology in its formal form. We want to obtain a parameterization of this causal hypothesis. And what you see represented here is actually some real data. This is an estimation of the epileptogenic zone overlaid of a post-surgical MRI, where actually we are estimating the probability distribution of the corresponding parameters. This is coming from an individual patient. We will hear more about this later. In blue, you see our sampling using quite sophisticated Monte Carlo Markov chain techniques. And you see actually there is multimodality. Towards the right, it's higher epileptogenicity. Towards the left, you see non-epileptogenicity. Depending on the type of metrics, depending on the type of data feature you use, you will actually obtain different results. If you take a second-order approach, very often used cross-correlational analysis, functional connectivity, you have actually a Gaussian parameter, a Gaussian distribution fitting to this, and actually you will not capture the essence of the multimodality. What we are doing very often is that we are going for the center of the mass of the probability represented by the crosshair. Yeah? Which is a maximum a posteriori approach, because this is what we can do in finite time. But I want you to be aware of this issue. This non-identifiability can either come from the mapping from the sources to the sensors. That's one possibility. Then we can improve it with more data on multiple modalities, or it is intrinsic to the system because of the multi-scale nature of the brain. This feature is called neurodegeneracy, that was also previously mentioned, and is essentially one of the essence of our brain to be resilient to outside impetus. So The inference is one of the elements that is aiding us in moving forward in the clinical translation. Yeah? So the causal inference always needs to be linked to a causal hypothesis. Inference extracts a structure that is in the data. If we want to go beyond that and have a predictive approach, we need to formalize our understanding, our knowledge, and link it to a model. Then we talk about causal inference, and we are actually performing inference in the context of a mathematical model. And this is what digital twins are good for. We can use them either as a template for parameter sampling to inform clinical decision-making, or we can use them as platforms to simulate brain activity and the impact of the causes that we are after to understand. And one of the aspects that poses many difficulties to us is the multi-scale organization due to this issue of neurodegeneracy and this intrinsic non-identifiability of mechanisms. We have to live with that. It has advantages and disadvantages, and the more data we introduce into the digital twin model, the easier it will be to navigate from the mechanistic perspective, but the more difficult it will be actually to perform the simulations. We have parameter sensitivity, high dimensionality. All these aspects are rendering the simulations very difficult. eBrains is the key deliverable of the Human Brain Project, and it is a digital neuroscience infrastructure, a European digital neuroscience infrastructure available to the community. And it has, in particular, the objective to integrate our capacity of working with tools, models, and data in the same space in order to render them interoperable, because then we can actually traverse these type of scales of organization and link causal hypothesis to data and the analysis tools. And here, what you see represented on the right is the traverse of scales captured by the three major simulators, simulation engines and ecosystems we have in eBrains. The virtual brain I will focus on, but then on a more microscopic level, simulating microcircuits, neurons as point neurons. This is a nest simulator. And even further down on the subcellular level, we are having the neuron simulation ecosystem. What we are missing, and there we're not doing well enough yet, is a molecular level, capturing reaction diffusion processes, in particular in the extracellular domain, linking and affecting the neuroelectric activity in development, but this is our entry point for drug discovery. Yeah? What I want to focus on is actually on the macroscopic systemic level. And there we are essentially informing the brain models, full brain models, by non-invasive data. So typically obtained from this type of imaging techniques that you see represented on the upper left, MRI providing us with topographic information about connectivity and the capacity for us to build a network out of this data representing the model. But in order to close the causation and inference loop, we need to link it to the machine learning capacities to sample actually the distributions of the probabilities and not just perform optimization. So I want to emphasize this particular point. And this is what we can do in eBrains. eBrains has been actually designed over the Human Brain Project lasted or will last until middle of next year, 10 years. And a whole culture of coordination and cooperation leading to the capacity of integration data, tools, and models has been developed. What you see represented here is a capture of the causation and inference loop. However, now from the back end, from the infrastructure that actually supports the scientific workflow on the left-hand side, the Atlas services, knowledge graph to use fair data principles in order to merge the data in the same reference space and make them accessible, to map them upon model parameters. Highly non-trivial, actually, because you have measurements on the one hand side, and you need to map them on mathematical representations within the model. Yeah? So there is a metadata approach that is actually making this possible. linking it to the simulators on the right-hand side, represented and being supported by a hardware infrastructure, supercomputing and high-performance computing structures, including neuromorphic structures, allowing this, the simulations and the necessary sampling to this. So this is the type of support we need for the causation and inference loop I was referring to. Key is the atlasing that allows us actually to organize the data and represent them in the same reference space to make data that come from heterogeneous data sources accessible from obtained from different modalities and make them interoperable. So you see a representation across scales, all the way down to post-mortem data. I'm talking mostly about the human, but you have also representations of rat data, for instance, rat data atlas. Yeah? All the way down to the cellular resolution using latest technology like polarized light imaging, PLI, which allows us to visualize individual cells. Yeah? And that allows us to get across the different modalities, cells, fibers, all the way to functional data. There should be a third dimension actually mapping the appropriate models, yeah? Because this is being done with these data being mapped on the parameters. That allows us then to represent it in the same space, building a mathematics where the network with this mathematical object is spanned in the patient's three-dimensional physical space and is being using different forms of mean field dynamics. So we have representations of population dynamics of the individual network nodes that can either be simulated by the different simulators to make them co-simulate, cooperate, or what we are using very often, we use mean field approaches from statistical physics and nonlinear dynamics in order to obtain an information compression. This is the type of dynamics that is behind there, where on the left-hand side with N you see the intrinsic dynamics of a regional node. Then you have the nearest neighbor connections represented by the second term, developing the local dynamics when we go to very high resolutions. And then the connectome spanning the connectivity skeleton, undergoing time delays via signal propagation. And as Denis was talking about this earlier, spanning the space-time structure of the connectome, where time delays plus the connectivity strength are determining the space in which actually dynamics can emerge. Subject specific. So summarizing the workflow is we scan individual patients, we map the topography and build an atlas, a network where we are actually connecting the individual network nodes by the individual connectome, and then equipping this with the appropriate mathematics in order to render it functionally active, and then we can insert sensors in the same space. Here in this case for epilepsy we use a stereotactic EEG, mapping from the sources to the SEEG or non-invasively MEG and functional MRI. So this is a workflow that we're using, and in the remaining time I would like to share with you the applications to epilepsy. Afterwards, Sri will talk in more detail about a functionally constrained model, and Randy will carry it into healthy aging applications. But the workflows are very similar, actually. So in epilepsy, we are, in particular, pharmaco-resistant epilepsy. 1% of the human population will have a seizure at one point in his or her life. Seizures can be treated through pharmacological approaches, but about 30% of the patients are pharmacoresistant. For those, the identification of the location of the epileptogenic zone is the major hope, because this can then be treated either through resective surgery or stimulation approaches. And what you see represented here is a virtual brain approach, composing the patient's virtual brain out of the individual brain imaging data. On the top right, equipping it with a model and being able to generate this type of seizure activity mathematically, so it becomes an autonomous platform, better refined by additional information we get about the patient, typically a lesion in the MRI, performed additional personalization by the inference using typically machine learning techniques, in our case Bayesian inference, and then going to the patient and the clinical decision maker, the treating clinician, to help guide the surgery. There is an ongoing clinical trial at the moment where in France, over 4 years, essentially, where we are collecting data in 13 reference centers, where this workflow is implemented, and we have 2 branches, or 2 arms. One arm where the information about the virtualization is actually being provided to the clinician, VP positive, and another arm in which this is not the case. We are collecting the data, we are centralizing them in Marseille, we virtualize the patients, we build a virtual cohort essentially, and for each individual patient we are sending the information back to the clinical center in the VP positive branch thereof. And this is on the left-hand side the type of results that we are finding, or our metric. You see the overlay of the estimated epileptogenic zone overlaid on a post-surgical MRI on the top where it actually coincided with the surgery where it was performed, and on the bottom where it actually does not coincide. At the moment, we are expecting the trial to be finished by summer 2023. It was a little delayed by the pandemic. Yeah? On retrospective data, you see some of the performance. Precision over 54 patients is on the level of 77% with correspondence to the clinical hypothesis. And it actually drops down when the surgery has actually not been successful. So we are making the distinction between seizure-free and not seizure-free after 1 year. Yeah? And this is indicative for some of the predictive power of the virtual brain modeling in this particular case, but actually most interesting in this particular case, when you look at the difference between seizure-free and not seizure-free patients, you see actually that in the case, and I'm explaining this here better along this figure, in the case of where the clinical hypothesis actually did not coincide with what came out of the virtual brain estimation, which is represented in the red areas. Yeah? This corresponded in most cases actually to areas that were not accessible for exploration by the clinician, but they were accessible to the virtual brain because the virtual brain can actually perform the simulations on the source level. But you see actually that the SEG is implanted in the area that are covering the blue areas. And this coincides with the large increase of variance and the number of non-explored areas which correlates to the not seizure-free case. This is the current situation. And we are, the only way to test this causally is in the prospective trial which is ongoing. Yeah? So, in this particular application, you saw the use of the virtual brain as a template for personalization and better estimation of the epileptogenic zone on an individual level. However, the more powerful element in the long run will be actually the use of the virtual brain as a platform for discovery of other potential situations such as therapeutic approaches specific for the patient. Here you see a simulation after a brain got virtualized and personalized for a particular patient. You see her implantation scheme. The activity that is evolving is on the blue dots. Essentially, this is a source level. And I run it again. On the source level, and the yellow balls are actually capturing the activity on the sensor level for the particular seizure that is represented on the bottom. There you see the type of SEG signal that is accessible to the clinician. And here you can now perform virtual surgery. You can intervene, or you can actually start questions about stimulation paradigms. It makes only sense if the inter-individual variability is actually informative. Yeah? And that is the case. This has been tested in animal studies, but also in human studies. Here you see an example of where, here you see an example on the left-hand side where connectome has been systematically altered. These are experimental data. When connectome has been systematically altered and performed for the virtual brain studies and predictions, and where actually the predictive power in whatever your heart desires in terms of statistical metrics, precision, sensitivity, F1 values, it always drops down. It also drops down when you go from one patient's connectome to another patient's connectome for the individual particular seizure case. So there, this is pretty well established nowadays that there is an individual predictivity. Another aspect that is very important for interventions is actually stimulation, brain stimulation. And for this, you need essentially two elements need to be satisfied. You need to capture the individual topography, number one, but and you need to be at high resolution. High resolution is another one of the factors that is actually determining the predictivity significantly. Today, in the clinical trial, for instance, we use 200 nodes as a resolution of the network. This gives us an estimated area of brain tissue, cortical brain tissue, of about 10 square centimeters. Yeah? But we need to go down to the millimeter level if we want to have a meaningful statement about interventions such as brain stimulation. And you see here, we can stimulate on the left-hand side, we typically stimulate either with implanted electrodes, as EEG electrodes, or what is at the moment in development, you see it represented on the left-hand side. We can stimulate also non-invasively with upcoming technologies such as temporal interference, which we have improved by making it multimodal, and we can actually reduce the stimulation focus to 2-3 millimeters, which means we're actually non-invasively approaching the spatial resolution, the precision of deep brain stimulation, DBS. Yeah? So this is upcoming. However, once we are on the level of high-resolution virtual brains, on the millimeter level, we can actually take physics simulators and map the electric field on the cortical surface and the subcortical areas. And here you have a representation, this is where we are nowadays, a representation of a reconstruction of the human hippocampus. Yeah? In much detail with the different areas, CA1, CA3, CA4 that you see represented here, together on the right-hand side with the SEG needles that are in there. And you see a scaling of the patch area when we go from 10 square centimeters, so 1,000 square millimeters, towards the millimeter range, and the quality of the precision of the reconstruction of the electric field. So it drops down, so it scales actually, drops down to 25%, approximately 30%. So high resolution matters. Yeah? And we are becoming capable of doing this today, but it requires computing resources. To finish up, I would like to convince you that we are actually able, on a personalized manner, to stimulate. Here, here you see a representation of where we have personalized a virtual brain for a particular patient. This is just one example out of the retrospective cohort of 50 patients. We have personalized the brain on the seizure. The virtual brain model has never seen any type of stimulation. And then we can actually enter either through the SEG needles, and this is what we have done here, or in the future, hopefully with temporal interference, so non-invasively. But you see 3 different stimulation sites that are represented here, temporal lobe, further in the occipital lobe, etc. So 3 different stimulation sites. It has been done experimentally with the patient data, and you see for the first case, there is no seizure that is being triggered. That is different for the patient data in the second case, but when you go to a different area, you trigger a different seizure with a different pattern. Yeah? on the model that has actually never seen the stimulation data, you find that we are actually able to reconstruct the spatiotemporal propagation pattern as observed in this patient. This is for stimulation sites, or if we change another clinically very relevant parameter, that's frequency or amplitude, we are actually capturing the same type of phenomenon. And at the moment there is statistics. We are performing the statistics over the individual 50 patients. Yeah? I'm almost done. This is my last slide. Thank you. Yeah. So, what I wanted to share with you is essentially the importance of integrating these elements together in the same platform. This yesterday morning from Elias, we heard precision, complexity, and convergence. In the language that I presented to you, but it's essentially the same, I would have said multi-scale, high resolution, integration. But it's just words. The meaning is the same. We need to build platforms that are personalized, that allow us for personalization, where we integrate the tools, models, and data in the same reference space, enabling the execution of the causation and inference loop. In my opinion, it's not optional. It's obligatory in order to move forward, because integration is essentially a form of understanding that allows us to actually add value in the data per se. Yeah? And through this, we are generating knowledge. And the type of platforms that are developed by Dassault System, as presented by Patrick Johnson, or eBrains as offering it to the research community, are absolutely critical in this aspect, because they are the porteur of the workflows that can be developed. I thank you for your attention. I thank my colleagues and friends that are listed here and the agencies that have supported us. Thank you very much.
First, I'd like to thank Victor and the organizers for inviting me. It's been a pleasure. Yes, it's a rather long title, but I'm going to sort of revise that on the spot. Essentially, what I'm going to talk about is how we use the virtual brain to actually diagnose a patient as to whether they have epilepsy or not from just minutes of scalp EEG data when that data is captured when at rest, meaning the patient is just sitting there and resting. Perfect. So a little bit statistics on epilepsy. Most of these numbers are in the United States, but 65 million people or so have epilepsy in the world, 3.4 in the United States. That's 1 in every 26. And of course, if you have epilepsy, you have a higher risk of, greater risk of premature death. In the United States, the annual economic burden is $60 billion to treat these patients, and every year about 150,000 new cases appear. Now, just want you to take, we're going to really dig deep into this one application. So I want you to appreciate, what it takes to actually diagnose epilepsy. So if you have a seizure or a seizure-like episode for the first time in your life, you might go to the emergency room or to a neurologist, and this is what's going to happen. They'll ask you for your medical history. They'll also ask you, well, what happened? Or they'll ask the family members who might have observed the event, say, tell me a little bit about it. They'll take an MRI scan to look for any abnormalities in the brain that could be possibly causing that seizure episode. And then the routine scalp EEG. When the patient is there, they will say, okay, they put on a very non-invasive cap, and they record their EEG for 20 to 30 minutes, okay? And then what they're looking for in that EEG, in particular, are abnormalities that are indicators of epilepsy. So these abnormalities could be particular spikes in channels, what are called interictal epileptiform discharges, or some delta waves that are abnormal, say in the temporal lobe, but it's all done through visual inspection. Now, the problem with this is that, first of all, those abnormalities may not be captured in the 20 minutes because these events are random. So what are the chances that when the patient came and you put the cap on that you're actually going to see these random events? So there's a couple of outcomes here. Is that the patient EEG is normal, has no abnormalities, because they simply don't have epilepsy, or they have epilepsy and you just haven't been able to catch that at that visit. And this is a big problem. At the end of the day, you have 60% sensitivity, I mean actually seeing these indicators that will allow you to confirm your diagnosis. And what that means to the patient is as follows. What we understand is that the majority of time when the MRI looks normal, the medical history is ambiguous, and the EEG is normal, the doctors will still send that patient home with anti-epileptic drugs. Because the chance that they're wrong, the consequences of being wrong are much greater. They'll have a much more increased chance of the next seizure, which could be much more serious, possibly fatal. So everybody gets sent home with drugs, which has some side effects that are not pleasant. So, and with that said, they'll send them home and they'll ask them to come a second time. They'll do another EEG and they'll say, do we see anything? If they don't see anything, they'll ask them to come another time. So this process of getting a confirmed diagnosis can take months and possibly years. So the key issues of standard care is that, well, first of all, if you're visually inspecting that data, you have to have some expertise in this and training. So these are typically read very well by epileptologists, prone to errors because one doctor might see something different than another. False negatives. We talked about that. You have to send them home with medication or the consequences could be quite fatal. And then the delayed diagnosis is really a problem. So what I'm going to talk about is how we basically created a software. We call it EpiScalp. RS stands for resting state because they're just resting. It's going to take minutes of scalp EEG data and produce a heat map. And what this heat map is, if it shows any heat or activity, then it's an indication that this patient has epilepsy. And if it doesn't show any activation, it's an indication that they don't have epilepsy. And this particular algorithm, where the virtual brain is embedded in, is actually independent of abnormalities. We don't need to see anything that's in that scan, at least through the naked eye. So I'm going to start digging into this algorithm and show how we do it. So the actual algorithm, EpiScalp, has two components. It's very similar to the forward modeling and the causal inference, the backwards point. The first, which I call the dynamic network model, is the personalized virtual brain that's going to be built and constructed from that EEG snapshot. Then it's followed by source sync analysis, and I will define source sync, okay? But it's essentially taking the model and its properties and trying to understand which nodes are special in certain ways, sources and sinks. And again, I'll define that. And then it produces this activation map. Okay, so this idea of sources and sinks, which I still haven't defined, and I promise I will, is ubiquitous. It's not a new concept when you think of networks out there, whether it's storage networks, transportation, communication. But what does it mean in the brain? So now, before I define it, this is really important to understand, just think about the problem we're trying to tackle. I have a completely normal scan, normal to any trained clinician. Whether I take a healthy person or this epilepsy patient that had the normal scan, the clinician cannot tell the difference. So what we're trying to do is seeing, well, can a computer tell the difference? Well, the first thing you might say is maybe the computer will find patterns that are not observable to the naked eye. But many thousands of papers have been published with that effort. Instead, we take a different approach. We said, let's build this virtual brain model that tells me about how different regions are influencing each other. Look at those properties to see if there's a set of properties that are epileptic versus healthy. So what could these properties be? So one of my students asked the following questions to identify this property. She asked the question, well, if I have epilepsy and I have this epileptogenic zone whose job it is to trigger seizures, why does an epilepsy patient have seizures all the time? If you think about it as almost like a bomb ticking, why isn't it going off all the time? And her hypothesis was, well, it must be that other regions in the brain are holding it down. This is your body's natural reaction to this diseased part of the brain. Okay? So I've got the epileptogenic zone that while you're not seizing, so during rest, it's being heavily influenced by another group of areas in the brain. So she calls that epileptogenic zone sinks because they're being heavily influenced by sources. And that if you're healthy, you don't have these sources and sinks. So that was the premise. So now let's define sources and sinks in the brain. Sources. So you think of the brain as a network system. And in this case, each node is like an electrode contact, okay, where we're going to eventually get a signal. Now, a node in the network is a source if it has a lot of influence on the rest of the network or other nodes in the network, so a lot of outgoing arrows, but not being influenced itself, okay? So I'll define influence shortly. That comes from the modeling. A sink is the opposite. It's a node in the network that's being heavily influenced, but not very influential. Okay, so a lot of incoming arrows. And her hypothesis was, well, when you're not, when you're right before seizure, this epileptogenic zone, these sinks all of a sudden turn into sources and they trigger a seizure which propagates through the rest of the network. So right before seizure, this epileptogenic zone, they become sources. However, when you're at rest, something's holding them down, so they must be sinks, being influenced, inhibited, okay? So the trick is, can we detect the presence of these sources and sinks from EEG, okay? So the process I'm going to walk through, I'm going to actually open the hood of that episcalp, is we're going to build that virtual brain model. At the end of the day, I'm going to show you it's a matrix. We're going to find sources and sinks derived, just computed directly from that matrix, okay? And then we're going to make some, like a source sink index, okay, that for each region of the brain and display that then in a simple to read heat map, okay? So that's the process. So let's go one slide into this model. So here's what we do. We consider this EEG network. Every contact is a node in the network. Okay? And what we're going to do is it produces EEG data over, say, 10, 15 minutes. And we're going to define each node as 1, 2, 3, and the signal being generated is denoted as X1, X2, and so forth. And we're going to say, and this is the important part, this is not functional connectivity correlation matrices. What we're going to do is build a generative model that says if I have the present activity in the network, can I predict the future activity 1 millisecond later, okay? From X of t, can I predict X of t plus 1? So how do we do that? We take this 500 millisecond window and we build a very simple linear model. Linear meaning X of t, if I just take linear combinations of the present activity, I'm going to predict the future, but only in that 500 millisecond window. So linear means I have this A matrix. If I have n contacts, this is a square n by n matrix. Okay, and we've shown that this can actually reconstruct activity quite well. But we know the brain's not linear, so what do? I slide, and in the next window, I build another linear model. I slide again, build another. So at the end of the day, what we have is what we call a linear time-varying model. That's our virtual brain model. It's a phenomenological forward model as opposed to a mechanistic forward model as in the virtual brain that Victor presented. So I have from this patient, from the scan, I get a sequence of matrices. And at the end of the day, what does that A matrix tell me? It's about who is influencing the activity of who in the future. Okay? So we look at sort of this average, I'm going to call it an influence matrix. And now I ask the question from that matrix, can I figure out which nodes are sources and which nodes are sinks? Because the sinks, if you have strong sinks, that's an indicator of epilepsy. That's our hypothesis. So how do I do that? Well, if you go back to the details of the math, any row that is highly red, high values, that means it has a lot of other parts of the network influencing them. So to be a sink, your row has to be more red, but at the same time, you cannot be influential, so your column has to be blue. Okay, that's where the mathematics comes in. And then so now every node, I look at their row and column, this is the model, and I figure out where they fall in what we call a two-dimensional influential source sink 2D diagram, where essentially what's in the top left, which is computed directly from the main matrix, every dot here is a contact, your sources if you end up in the top left, and your sinks if you end up in the bottom right. And if you have epilepsy, you will see that there are these top sources influencing these top sinks. I know I didn't show you the mathematics behind it, but I hope you get some type of intuition of how we go from the virtual brain model into a notion of sources and sinks, and we turn that into actual quantitative metrics and produce that heat map. Okay, so to test this hypothesis, we're going to do a retrospective study on patients treated across 3 different centers, and this is the premise. If you have epilepsy, from normal EEG, I should see some activation, even though the EEG is normal. But if it's coming from a non-epilepsy subject, I shouldn't see anything. So here's our data. So we are looking at 235 at this point, patients who came in. We take their first EEG scan, and then we actually made sure that these patients all had confirmed diagnoses, even if that confirmed diagnosis happened months to years later. Okay, so we have exact labels. Do they have epilepsy or not? And what we do is we take 10 to 15 minutes of their scalp EEG. We do some artifact removal, eye blinks and so forth, in an automated fashion to get more cleaned up data. And we put it through an analysis pipeline. And although this diagram looks complicated, essentially what we're doing is in the bottom path, that's what the clinicians do today. They capture all their data, they do visual analysis of the EEG, then they make a diagnosis. And what we're going to do is we're going to compare that to a prediction we make based on that heat map. So we take the heat map, if we see activation, we're going to basically that's going to turn into the probability of having epilepsy through a model approach, and then we're going to compare the prediction to the actual diagnosis. Here are just examples of real data. These were 2 patients. One had epilepsy, one did not. None of them had abnormalities in their data, but our source sink map lights up when you have epilepsy and doesn't when you don't. But across all the patients, around 235, here are our results. So if you look at panel A, every dot is a patient. Okay, the y-axis is the probability of having epilepsy. So if it's a good marker, if this is a good heat map, informative, then that probability should be high if you have epilepsy and low if you don't. And as you can see, we have a reasonable separation. In fact, if you apply a threshold at that alpha star, that line there, and say, okay, if your probability is above that threshold, I'm going to say you have epilepsy, and if it's below, I'm going to predict that you don't. We get around 80% accuracy. Now, that may not sound like a high number, but remember, these are EEGs that are completely useless to the clinician because they're normal. So this is actually adding some new information that could help expedite the diagnosis, okay, and not put these people on drugs if you don't. By the way, all 235 of these patients were sent home with drugs, every single one of them. So even if we were very strict and wanted to make sure we predict all the epilepsy patients, we would still have not given drugs to at least half of the patients that don't have epilepsy. Okay, we also showed that this algorithm is agnostic to the center. So the accuracy doesn't change if you're at this center versus that center. We also show it's agnostic to the sex. It's agnostic to your age, and how many drugs you're on. So what's nice about that is I don't want it to be dependent on making sure they're sleeping, no drugs, they have to be in this state. I'd like that tool to work anytime, on anyone, at any center. Okay, and so just to summarize, so with this type of virtual brain plus source sink inference analysis, we don't depend on abnormalities anymore. It's not subjective, it's completely quantitative. We can increase sensitivity by over 25%, and what would be the impact? Getting that first visit diagnosis correct, and not applying this stigma of epilepsy to patients who don't have it, maybe taking them and redirecting them to the right clinician, say if they have psychogenic non-epilepsy seizures or a cardio problem or a respiratory problem, get them to the right doctor earlier is what we hope for with this type of tool. And I'd just like to acknowledge my students. Kristin Gunnarsdóttir, she's from Iceland. She actually showed that the source sync analysis applied to intracranial, the stereo EEG that Victor showed, that can actually localize epileptogenic zone when you're at rest. And Patrick Myers developed Episcout that I just talked about today. And thank you.
My presentation is going to be a little bit of what we've done, but also a little bit of what we plan to do. So it'll be both actual and aspirational. The focus of this work and the focus of the work that I've now taken to Simon Fraser University really tries to get at this question about what makes our brains resilient. And that's kind of the converse of what makes, ensures healthy aging, because by some definition, one would assume that healthy aging actually is the embodiment of being resilient. But the focus really looks at what are the factors that allow us, to, for example, to maintain brain health in the face of things like stroke, in the face of certain kinds of dementia, overcoming head injury, and potentially adapt to developmental disability. It's a fairly broad scope. Resilience is not just a brain thing, however, and that's one of the important features. It's come up a number of times in this conference, that the biology itself is encapsulated in the person's own context. So there's biological factors that determine brain health. The rest of the body, as we've heard already, cardiovascular factors, metabolic factors, all influence brain health. But there's that person's environment as well, the psychosocial environment that they're sitting in. Their friends, their family, their broader social cultural context, and even the environment, broadly speaking, is going to affect resilience and affect brain health. And it's the interactions of these factors that really holds the key to understanding what helps brain resilience and what threatens brain resilience. It's really complicated, and we've heard that a number of times. But the purpose of our session here is to suggest that this can be addressed by using computational modeling. What I'm going to start with is just show you some applications. This is more the actual thus far, where we've taken the virtual brain and applied it to persons with Alzheimer's dementia. To see what we learn from that in terms of the ability to classify people who have various ranges of cognitive impairment, and then also be able to understand some of the biophysical aspects that are related to that cognitive impairment, and see then if we can use that to guide how we might intervene at some point in time. The first application came from a study that was done by my former graduate student, Joel Zimmerman, and took some data from a prospective study that's been ongoing actually in Australia called the Sydney Multiple Aging Study. And the purpose here was not so much just to differentiate Alzheimer's disease from healthy controls, because that's a fairly simple thing to do. What we wanted to try and do is better appreciate those who have mild cognitive impairment, or MCI. So MCI is the transition, if you will, from ostensibly healthy status to some sort of full-blown dementia. MCI can have a number of underlying causes. It can be something like Alzheimer's disease, it could be other dementias, it could be cardiovascular issues, it could be nutritional factors as well. So being able to find out how to differentiate MCI from that which is going to be dementia versus other things is going to be an important capacity as we move forward in addressing these things. And what you see on the scatter plot is the ability to differentiate the different patients based on biophysical parameters that are derived from the virtual brain models. These biophysical parameters represent things like the balance of excitation and inhibition in the local cell populations, plus the global scaling of the connections between these different areas. And that's derived from patients' models that you saw from Victor earlier on. The main thing here is, again, the healthy individuals that are in black And the Alzheimer's patients in red are differentiated quite well. But what's nice to see as well is the MCIs are kind of staged across these individuals. Some MCI patients are more like controls, some are more like Alzheimer's disease, and some are in the middle. And that tells you that there might be, again, different underlying processes that are engaged in these MCI individuals, and that might be a way of being able to differentiate them, stratify them early on in the process. So again, this is being followed up now with subsequent waves in this longitudinal study. Carrying it a bit further now, because we have a biophysical model where we have parameters that are inspired by what we might use to treat a patient, we can also then give virtual pharmacotherapy, for example. And this is some work led by our colleague Petra Ritter in Berlin, involving pretty much all of us in virtual brain. And the idea here is to see if we can re-establish the spectral power distribution in a patient's brain who has dementia. So what we did here is used PET scans of amyloid beta, which is one of the proteins that's thought to be an underlying cause of dementia, and use that to change the excitability of different parts of the brain. Derived what you measure with EEG, and what you see in the red is the slowing of EEG that you see quite commonly in patients with dementia. And then gave virtual memantine, which is an agonist, or antagonist, pardon me, for excitatory neurotransmitters, to reestablish the excitatory-inhibitory balance. So A-beta messes up inhibitory neurons. The drug memantine reduces excitation to sort of rebalance spectral power. What you see happening when you give virtual memantine is that the spectral power increases, suggesting that that's able to reestablish the balance in this virtual model, and suggesting that we can use this as a way to test, for example, interventions before they're done. A great example of testing that is, in fact, what Victor was talking about with epilepsy. The idea here is to be able to extend that into other indications, particularly dementia. This is the aspirational part of my talk. So a lot of what I'm talking about here are pieces of the puzzle that we would need to really start applying these methods to understanding healthy aging trajectories. Now, across the history, if you will, of neuroimaging studies, or actually, I guess, all of neuroscience, really, looking at aging, there are several clues that have emerged, suggesting that the way your brain does things, not surprisingly, does change as you age. And that may actually be a good thing, because some of that may be a way to preserve cognitive function, or maybe adapt to changes to ensure resilience. What you see here is a functional connectivity or correlations of activity diagram showing different parts of the brain that are interacting in young subjects when they're doing a visual memory task. And then the bottom diagram shows you what happens in older subjects who are doing exactly the same task. They have the same level of performance, but the interactions, the networks that are engaged are different, suggesting that they're doing the task differently, and in doing so, are able to preserve cognitive performance in this particular case. There's a number of examples of this through the literature. That aging really change in the functional architecture that may preserve cognitive function. So it's our goal to really understand what are the things that happen to preserve cognitive function. How do we facilitate that to ensure brain resilience? One of the other features that's come up, and this is kind of getting to both Victor's and Shree's ideas, that there's a broader change in the networks. And the networks have a very rich spatial and temporal pattern to them. And that spatial and temporal pattern is going to be important to be able to fingerprint, if you will, how people are changing across the lifespan. This is a complicated slide showing you relatively a simple measure, and that's actually more evident on the left-hand column where you see these colored matrices. These are functional connectivity matrices and how they change across time. So basically looking at the dynamics of network switching. And what you see in the top of that column A is a young subject, over 25 years old, I think this person is. And the green suggests that their networks are switching quite rapidly or quite frequently across time. A middle-aged subject, you see more red, suggesting that their networks are switching less across time. whereas the bottom part in that panel A is an older subject showing there's very little switching going on, suggesting that they're not switching their networks as much. The question is that related to cognitive function? Yes, it does. So that less switching actually seems to relate to worse cognitive function in these individuals. So the brain changes both its spatial patterns of interactions, what areas are talking to one another, but also how frequently those interactions switch. So the space-time structure changes in aging. This is some very nice work from a collaboration involving Joanna Cabral and Gustavo Deco, amongst other people, just looking at, again, the consequences of how this pattern of network switching proceeds and whether it relates to cognitive status. And the main point here is that you can track which networks tend to be associated with each other across time. So if you have network 1, where do you go to next? Do you go to network 2, network 4, network 5? You can analyze this and look at the likelihood of switching networks and how that then relates to cognitive status. And what Joanna et al. Showed is that that switching pattern actually does relate to how well the older individuals show cognitive performance on some standardized tests. So again, the progression between network states seems to be an indication of cognitive status. So we have now some suggestions that networks change. We have some suggestions that the pattern of how these networks change may be critical to be able to evaluate trajectories of healthy aging, potentially also trajectories of non-healthy aging. How do we measure that in a way that can be scalable? So the problem with these metrics, of course, is these all require an MRI, which is not a bad thing, but the MRI is not necessarily accessible for everybody in the universe, or everybody in the world, I should say, which makes it a difficult metric to apply broadly. We have looked at another measure, and this, I'm going to spend a bit of time on this slide. The measure is called multi-scale entropy. And the notion here is that the metric is sensitive both to the spatial interactions that are happening, plus how they're changing across time. So on the top, you see this series of slices. That's the space-time structure of the brain connectivity matrix. And that just shows you taking the structural connectivity, what areas are connected to one another, and just extruding it across time, telling you what areas could be interacting given a particular timestamp. What you can do is measure what's called sample entropy of things like EEG, for example, which she was showing you earlier. And that metric can tell you how much information is in that particular signal. And the information does, in fact, depend on space and time. So you can show that entropy changes as you look at different temporal scales. If we apply that to, for example, regular adults, you look at the curve, and the curve is dependent upon time scale. So as you go from left to right, you're going from very fine time scales, really fast interactions, close areas. If you go to the right, you start going to slower time scales that involve more distributed networks. So the measure is essentially telling you at which organizational level do you have the maximum information. And you see in healthy adults that it's sort of in the middle scale. If we apply this to children, these are kids from about 4 to about 12, you see in general the information that's contained in their brain signals goes down, is less. Not surprisingly, because they're still developing, so they don't have as much information, if you will, in their brain than adults do. Older adults that are healthy have an interesting change in their pattern. They have an increase in the fine scale entropy and a decrease in the coarse scale entropy. So again, increase in fine scale means that there's more interactions, more information at a faster time scale and a more local distribution, closer areas. Whereas the decrease in entropy, decrease in information at coarser time scales involving long-range connections. And that's for older people who are healthy. If you look at older people who are at risk, you find that they don't show that increase in the fine scale and continue to have a higher level of entropy at the coarser scales. And these people, again, this is across a number of studies, are either showing cognitive decline already or will show a cognitive decline at least 2 years from the time that they're measured. So it actually could be prognostic of decline. This is some examples of some of the factors. This is a very brief paper just showing you again the curve in young and older adults, showing you the higher fine scale entropy and lower coarse scale entropy. in relation to their physical activity. So it gets back to the idea that the brain is embedded in a body, and that embeddedness is actually an important feature. And you can actually then look at how they're influencing one another, suggesting that physical activity actually boosts this fine scale entropy in healthy adults. So it could be a measure of brain risk. We do find, for example, in the left panel, that people who, if you parse older adults in terms of high and low performance, you get nice separation between older adults who have high entropy at low scales or from core scales and actually have poor cognitive function. If you look at the right panel, if you look at just, these are individual subjects now, and we're having them displayed based on their MoCA score. MoCA is a measurement of cognitive function in older adults. And you find that those have the worst MoCA scores are the ones that have a very high scale entropy at the core scales, whereas the best MoCA has the high fine scale and low core scale entropy. Again, more information in local parts of the brain, less information in distributed parts of the brain. So this could be a very simple metric. You can actually measure MSE, is called, or multiscale entropy, with one electrode. So this could be a very nice indicator of healthy status. So now we have some targets that we could use then start building these models of healthy aging, which is something we actually need to do to really characterize the population. Everything we've talked about today is really just building these population models so we can use them as a way to compare individuals across time and as they come into clinics, for example. So we now have a target for modeling. We've got changes in space-time structure we can do with entropy, we can do with MRI. What we can then do is take individual subjects now across a large age range and derive the modeling parameters and look at the distribution of parameters in relationship, for example, to age, in relationship to cognitive status, in relationship to perceived risk. So the workflow that we started right now, and this is a really bad diagram showing you one of the projects that we started, taking a cross-sectional data set from the Cambridge Center for Aging and Neuroscience, or called the CAMCAN data. There's about 700 individuals here. We can derive their brain models in TVB and look at the distribution in the empirical distribution, and then in that distribution we start playing with the parameters and start pushing them to the extremes and see how do the dynamics change as we move things like excitatory-inhibitory balance, like global coupling, and see what the parameters look like, and then see if we move it in one direction, do we get a model that's non-plausible? For example, in the top right you see what looks like seizure activity, whereas the lower panel shows a model that would be implausible, or not very probable, but actually shows reasonable patterns, and that could be a sign, for example, of resilience or risk. And so that ends up being sort of a toolbox, if you will, to look at the distribution of potential architectures that underlie healthy aging. We can relate that to cognitive function, then derive predictive models then to see how these particular patterns then relate to cognitive function across the lifespan. I know this is cross-sectional. I'm going to get back to that in a second. You can then extend that. So now that we have a platform or distribution that relates to healthy aging, we can then start adding in the TVB models of different kinds of dementia and seeing where in that distribution of parameters do you start seeing what are called bifurcations, where there's a qualitative change in the properties of the network that's consistent with different kinds of dementia, and how long prior to the onset of dementia do you actually see these changes happening? When does the bifurcation happen so that you all of a sudden have now a brain that's progressing through to Alzheimer's dementia, it's progressing through Lewy body dementia, it's progressing through vascular dementia, FTD, etc. And on the far right on the slide, you can then envision being able to use these models again as a fingerprint to go back out and take things measured like entropy, for instance, and use that as a marker to measure it back to the population parameter. So it becomes a very nice workflow from creating these large space of models, then using the model as a target for evaluating data you can collect pretty much anywhere, like EEG, for instance. So that's a really important aspiration. We're working towards it. We're not quite there yet, but I think this is a promising workflow to go from the computational model, where we're integrating parameters across a large space, to be able to use it in a clinically meaningful way. And this is similar to what Victor was showing in Epinov, so I think this is a similar aspiration. Again, it is an aspiration, we're not there yet, but we'll get there soon. Cautions. So there are cautions in working towards this. One of the ones, as I mentioned, that this is the data I showed you earlier is cross-sectional. And there was a very sobering paper that came out a little while back that looked at the difference in predicting things like brain age versus using cross-sectional data versus longitudinal data. So what you see in this diagram are two different data sets, one of which has been split into two different ways of processing it. And you have the predicted brain age from the longitudinal on the x-axis, predicted brain age from the cross-sectional on the y-axis, and you see across these data sets that there's zero correlation between brain age predicted using cross-sectional versus longitudinal data, which is really troubling because a lot of what we think we know about aging in the brain comes from cross-sectional studies. So a moment to pause to think about what's happening here and reinforces the fact that we really need to use longitudinal studies. I'm not going to read this. This is basically just going through one of the main conclusions. One of the things that's important here is they did emphasize that the brain age is probably more related to early life events like nutrition, like bad health outcomes, like cultural factors, than the biology per se. This gets back to the idea that brain health is determined as much by the environment as it is by your brain per se. We need to incorporate those factors. The other thing is that we do need to be mindful that much of the data that we're using for these studies are coming from Western cultures. So the representation of indigenous people, for example, representation of Asian people, for example, representation of other populations is not prevalent in a lot of these data sets. If you start looking at these other populations in what are called normative populations, you find that they don't get represented very well at all. So that, again, social cultural factors are critical to be able to have a model that's useful and translatable beyond just the Western culture. And that gets back to this my last slide now, just saying that we need to be mindful in terms of the models to be able to take into consideration that the brain exists in a body, that exists in a person who's interacting with their environment, interacting with their loved ones, interacting with the social, cultural, and the political environment within which they sit. And to really capture that complexity, we have to start building models that really span scales. Victor was talking about spanning scales within the brain. The other aspiration is to start spanning scales outside of the brain. That's a rather complicated approach because these scales have different spatial signatures, different temporal signatures, and make it complicated to model, but not impossible. And since it is possible to do that, let's try and do it. Let's try and use these data to really span scales of space and time and span scales of the person within their culture. So final thoughts. So we can build PVB models for healthy aging. There's a wealth of data. We just need a good, robust workflow. If you start doing a Google search for healthy aging and brain data, you'll probably get 7 or 8 pages of hits. There are problems. A lot of them are cross-sectional, which I just showed you may not be the best way to estimate trajectories. There was some discussion about data quality as well. The data quality are not equivalent across these different data sets, so you'd be mindful of the quality control involved in those. I do think that the workflow that we're establishing here is a great potential for personalization and monitoring. We actually saw 3 examples, I think, of personalization here today, using computational model as a method to personalize some of these models and take them into the environment beyond, again, the Western culture. And finally, we've heard this message a number of times, and I just want to underscore that we really need to take into consider psychosocial factors that are impacting brain resilience. And they also, as we saw earlier today, also can impact the uptake of our putative solutions for brain resilience and brain health. And these factors need to be taken into consideration if we're going to be successful in promoting brain resilience across the lifespan. Thank you.
Thank you very much for inviting me to give a speech on stem cells. What is a stem cell? Stem cell is a cell that is able to replicate itself and also differentiate into many cell types. Stem cells are also the unity of development, the unity of regeneration, and units in evolution. Cell therapy aims to introduce a healthy stem cell or immunocell into a patient's body to get a better curative effect. Two types of cell therapy become breakthrough therapy technology. One is stem cell-based technology, and another is immunocell-based therapy. Stem cells are mainly used to tissue regeneration and reconstruction, whereas immunocells such as CAR-T, NK, CAR-NK was used to eliminate non-self or missing self cells and to eliminate the pathogen in our body. Let me introduce briefly the health and biotech group as biological biotechnology company or institution formed by myself. We are now located in Beijing, Shanghai, Tianjin, several big cities in China, and the HB Group is also the main body of National Engineering Center of Cell Products in China. Our group started from the construction of several stem cell technology platform in 2007, extended to research and development of cell drugs, and to translation medicine in 2019, and finally to industry, the health industry in last year. Here is industry base of stem cell technology and translation medicine. We call it Cell Valley. In fact, it is a special scientific park combining with stem cell bank, R&D center, CDM platforms, industry cluster, translation medical center, and a tourist attraction platform. Well, here show you our objective of research is to develop the cell drug fighting the refractory disease. We working on cell therapy and tissue engineering. We study the. We want to develop the drug of stem cell, immunocell, and exosome products, and we're also working on tissue engineering by mixed cell with biomaterial or exosome extracellular vesicle mixed with biomaterial, and to produce different type of cell products for the application. Last year, Professor Chen of Beijing University published a paper describing in stem cell translation the Tianjin model revisit. This model is, in fact, is established by our group 20 years ago, and now she found our Tianjin model could be served as a reference for other institutions in the field of stem cell translation medicine. Let me briefly introduce one of our technical platforms. We develop several technical systems to collect the stem cell from tissue of placenta and then actually expanded to set up a master cell bank, and after that, if we want to produce the stem cell in larger scale manufacturing, we can continue to this system permit to do many study on the R&D and the cell of cell drug. As you know, placenta, in fact, contains several types of stem cell. One is hematopoietic stem cell, everybody knows, and the other is mesenchymal stem cell. This stem cell can be isolated and culture expanded and then stored in the bank. And if we need to use follicular, we just take one and short and then inject directly into the patients. And we also can use the master cell to produce, to do the tissue engineering products. In comparison with MSC, mesenchymal stem cell from bone marrow or adipose tissues, the stem cell came from perinatal tissue has many advantages. They has no ethical problem. The cells with possess high proliferative ability, and they have low immunogenicity and easier to collect and isolation, especially easier for larger scale manufacturing. You can say that UCMC just means MSC from umbilical cord. They when we culture them in long-term culture, they can proliferate for a long time. We can then achieve many cells, to treat the disease, to perform a complete clinical trial. We also identified a population of MSC with high activity of immunoregulation and pro-angiogenesis. This population was labeled by WCAM1 positive. So we using the cells produced in our factory. We have performed a number of animal studies in different disease models, including blood blood disease, critical lung ischemia, and myocardial infarction, and pulmonary disease. And then we also evaluate the cell, the safety of this cell, and we perform the beauty administration in monkey, and also culture them in long time to observe if they have some changes or genomic mutations. Hopefully, soon we have done sure the mesenchymal stem cells from perinatal tissue are very safe, no problem for clinical use. Actually, in China, we have two ways to apply for clinical study or clinical trial. One way is like FDA, EMA, just we apply direct to the SFDR for the products application. Another way is just apply for a clinical pilot clinical study through the Minister of Public Health. And so if we want to study stem cell can use for which disease, which indication, we can study it just for pilot study to look if work or no. And if it work, we submitted the application to SFDR for clinical trial of new drug. This is a paper we described how to apply for NID in China. In fact, the system is quite similar to FDA and EMA, sometimes more stricter. And we also, this is our results for just for pilot study. We look if MSC are effective or not in different more than 10 disease, pain disease, and every study gave certified results. That's allow me to continue to do the complete clinical trials. We now have 3 types of stem cell products. One is placenta MSC hydrogen, just for topical use. And the other is UC MSC injection. And the third is highly active MSC injection. And we get the permission for clinical trial from CFDR several indications, diabetes 4 use and GVHD, pneumonia, LDS, acute or chronic liver failure, and also critical lung ischemia. This one is approved in France by NMC, where the study will be performed in France. Here, I just give you some example concerning the clinical application of perinatal MSC. As you know, the MSC now can be injected by several ways into the body. We can inject intravenous injection or intramuscular injection or just topical use. Our 3 types of stem cell products can perform through different route of administration. here just show you the use of MSC to treat the critical lung ischemia. The left leg of this patient is suffer from severe critical, and the doctor recommended to cut the jump, and after the patient asked to perform such clinical trial, we just inject this stem cell intramuscular injection 3 times. After that, this diseased leg was completed recovery. We developed the products of MSC mixed with biomaterial in the form of MSC mixed, and we put the products into the shingles before, and the shingles can be stored in the minus 80 degrees. And then if we want to use, we just sew it and put direct into the wounds. You can see the example of the patient received the injection for MC. The left is injected intramuscularly plus topical use. The right just topical use. Every patient receive 3 injections of MSC in different load, by slowing different load, and the wound can be recovered completely. This is the primary result of our clinical trial. We are now in phase 2, and we were into the phase 3. After clinical trial, we may get a new drug, so in the form of stem cell. The perinatal MSC has been used widely globally, and you can find from clinical trial governance survey that there are more than 300 clinical studies using UC-MSC, and they has been used to treat more than 20 types of disease. Here, just to show you the publication. Publication by Chinese scholar concerning clinical stuff of UC-MSC in different disease. You can see total more than 600 papers published related to the use of UC-MSC, and 11 clinical indications. Many patient has been under clinical investigation. And here, just to show you the UC-MSC can be used for liver failure or cirrhosis in China. I just searching for the website. Found a total of more than 750 patients has been used in China to treat the liver disease. And MSA are also some way effective to treat COVID-19 pneumonia. When the COVID happened in Wuhan, the first one of the best treatment is stem cell. Our Chinese scholar has published this data to use UCMC to treat the COVID pneumonia. So in summary, the market of cell therapy is coming of age today. MSC therapy is a foundational technology of stem cell-based therapy. Perinatal MSC are the most promising MSC therapy due to their easy collection, low immunogenicity, high proliferation ability, and biological activity, cost-effective manufacturing. If we culture the MSC from bone marrow, we need at least 3 months to do it. But if we use the UCMC, we just need 1 month. So 1 month, so the MSC has a high drugability. We have to strengthen the cooperation between industry, university, and research, and to accelerate the transformation of cell-based therapy products and clinical applications. Just as what we have done actually in China, we hope our experience can give some reference for other institutions. Thank you very much.
Thank you very much. Good afternoon, everybody. It's a pleasure to be here. Thank you very much for the invitation. And what I would like to do in the next 20 minutes or so is to share some of the work we've been doing at EPFL for the last about decade in bringing technological innovation to its clinical reality. And I'll show you some of the challenges that we've been facing, some of the successes, and some of the ongoing work. So I will talk about neuroprosthetic medicine, which as a whole can be labeled as a bioelectronic medicine. And as such, I mean, I talk about implantable technology that rely on electronic devices that are designed to deliver electrical function or electrical modulation to the body or record information from the body. And in the context of neuroprosthetics, so the interface we're developing is designed really to interact with the nervous system. So most of the techniques that are currently used at the clinic in terms of neuroprosthetic medicine are modulation technologies. So we try to write information into the nervous system. And one of the big challenges now is actually how do we do implantable techniques that allow to record information on a long-term basis. But first, I'll show you some example and detail a little bit what two of the sort of key techniques, two key devices that are currently present in the clinical context worldwide. So the first application is the cochlear implant. It's a device that is designed to restore hearing or to provide hearing perception to patients who are completely deaf. So who you see here on the photograph, this is a child. And so cochlear implant have this specificity. It's one of the very few devices we can implant from the birth of the infant. So we can insert the devices at a very, very early age, and the patient then, through his or her lifetime, wears the device and allows him or her to actually hear sound. And the device has multiple components. You see on the slide that here we only see the exterior, the wearable part of the cochlear implant. There's a matching part that is implanted into one part at the surface of the skull, and then the actual electrode, which are designed to deliver electrical pulses within the cochlea of the patient, is then surgically inserted into the cochlea. So here, the reason I'm showing this is to also give you a feel for what are the components that are needed for this type of implantable neurotechnology. So in the case of the cochlear implant, you first need this wearable system that is composed of a microphone that captures sound. Then you have a speech processor, and then you have an antenna or a coil that you see here that is inserted at the back of the head of the person. And this coil is then sending the information to a receiving coil that is fully implanted, which in turn will actually convert these signals through a small electronics that are implanted into a stream of electrical pulses that will then reach the cochlea of the person. So it's really a highly sophisticated technological structure that have multiple electrical components. And so today, patients who wear cochlear implant can have phone conversation, can appreciate some level of music. So it's actually a very successful technique. Now, a second very well-known example is the deep brain stimulation. So the components are exactly the same or very similar to the one I've presented for the cochlear implant. Or you may also be familiar with the pacemaker, the heart pacemaker, and this is also a very similar technique. And so in the context of the deep brain stimulation, we have an electrode that is surgically inserted in a deep structure of the brain. So you see on the cartoon, on the cartoon here, you see the electrode is inserted in this particular application to reach the subthalamic nucleus. So this is, you have a small craniotomy that exposes the surface of the brain, and then surgically the shaft here is inserted into the brain so that the electrodes, or the little tips here, are actually there to deliver this stream of electrical pulses. And then the second part of the system is the wiring, the cable that is then subcutaneously inserted and threaded towards the pulse generator, which is a macroscopic box that is usually inserted at the chest level of the patient, and that hosts a battery and the electrical stimulator. And this is what then the person is using to control the type of electrical modulation that is applied to the brain. And the outcome of such a technology is to actually stop or regulate some of the tremor that are associated with Parkinson's disease. So these are examples of what neuroprosthetic medicine is today. And so where are we going now, and what are the challenges that we need to face and overcome to build the next generation of bioelectronic medicine? And the key here, if you have one message to remember from my presentation, is this idea of interdisciplinarity. Because you have to, neuroprosthetic is really one key example of how you need to bring competencies, knowledge, and know-how together in order to actually bring advances for the benefit of the patient. And so when you start a group, when you develop a neuroprosthetic device, you actually need, of course, to have very strong information on the neuroscience part. So whether it's at the macroscopic level or the neuron level, or even at lower, more nanoscopic structures, molecules, and etc. So you have to know exactly what you want to interface, which part of the nervous system you want to interface, and how you want to interface it. Then you need to call on to engineers who will help and design the actual tools that will communicate, that will deliver this information that you need to write into the nervous system, or that will help collect the information from the nervous system. And there we have, it's really engineering at large. We need material scientists who develop new materials, new chemicals, chemical engineers also who can deliver, help us conceive new drugs. You have also electrical engineers who will then build the electrodes, build the electronics that actually will control or process the information collected from the nervous system. And now more recently we have the third players who are the computer scientists. With more and more we've seen the devices and the system we're building is actually the introduction of artificial intelligence to help process process the information that we're also collecting or need to deliver to the nervous system. And so those three disciplines are quite standard in university or academic environment. And what we need to actually move forward this innovation, that already putting these three groups of people together is one challenge, but then we need to interact with the clinicians. And so it's very important that early on we bring in the clinician at the table in the discussion so that they tell us what are their needs, and then we can design and engineer a system that is actually most appropriate for the clinical need. And of course, it's a bi-directional conversation. So we learn a lot from the clinician. They can also, through the techniques and the technology we're developing, they can actually also conceive new therapies and new treatment. So that's for the overall structure of bioelectronic or neuroprosthetic medicine, and I want to share with you some of the steps we've conducted at EPFL in the context of spinal cord injury to show you how we've put together some of these elements. And so this work really comes from the expertise and the drive of Grégoire Courtine, who is a professor of neuroscience and neurotechnology at EPFL, who is really an expert in how do we help a patient who suffers from spinal cord injury to walk again. And so he came with this idea of finding ways to treat paralysis. And so here what you have on the left-hand side is sort of a cartoon of the spinal cord, just to give you an idea of all of the region where along the spine from the neck all the way to the lower back, you have multiple positions where you can have an injury. And so what Grégoire decided to explore is, well, how can we deliver electrical modulation in the portion of the spinal cord that is sitting below the site of the injury? So when you have a spinal cord injury, what in essence is, in a simplistic manner I'm explaining now, but what happens during a spinal cord injury is that you cut the bridge between the brain and the rest of the limbs. And so depending on how high or how low is the injury, you are either tetra or paraplegic. And so the observation that Grégoire had made was, well, the part of the spinal cord that sits below the site of the injury remains connected to the rest of the body, to the peripheral nervous system in particular, and to the limb. So the idea was, well, if we could actually substitute the information that the brain usually sends to the spinal cord and do this artificially, maybe we can actually help the body restore its own function. And so in order to do this, we had to develop quite a range of innovative neurotechnology, one of them being an implant that you need to position at the surface of the spinal cord, understand where and how often we need to deliver this electrical stimulation, and also design a rehabilitation therapy that would go together with these techniques. And this is where a third player came into the equation, and this is Jocelyne Bloch. She's a lead neurosurgeon at the University Hospital in Lausanne, and together we've actually really put efforts and teams to actually try and bring forward these ideas. And so I will now focus on the end of that I was the most involved in, which is the design of these novel implantable electrodes. And this now goes back, so when you want to introduce new implantable technology, you don't immediately start in humans, so you have to sort of start from preclinical model, and in particular working with the rodent model. And what we have designed is actually an electrode system that would allow us to deliver electrical pulses, but also chemical drugs directly at the very surface of the spinal cord. And one challenge that we were facing is miniaturization. That is really specific to the case here, because we were working on rodent and in particular rat model. And we also had to actually have a technology that could conform the curvilinear structure of the spinal cord, because the spinal cord is not a flat plane or a rigid structure, and therefore you need to have an implant that would really cover the curvature of the device. So we came up with a technology that we now called eDura, for electronic dura mater, where we've managed to integrate electrodes, so metallic track and electrical contact, within very soft elastomeric material. And you see here on this video, there's an implant that is at scale here for the spinal cord of the rat, so it's a few millimeters in width, within which we have multiple electrodes that have been patterned onto this silicone, and we have similar elasticity between the soft polymer and the actual electrical tract that will deliver the electrical pulses to the spine. So how do we do this? So there we actually borrowed technology that exists currently in the microelectronics area. So I'm actually an electrical engineer by training. So we've explored how we could use thin film technology to build these devices, but instead of building these devices onto silicon wafer, which is what you would do in microelectronics, or glass plates if you want to make displays, we decided to apply this technology to soft polymer substrate. And so we operate in a clean room, which is an environment that is controlled in terms of dust and temperature and humidity in the room, because we use a lot of polymers and photosensitive polymers in particular. And then we've produced these devices just like you would do a standard integrated circuit, except that we work with polymers, so we do a lot of spin coating, and then we can deposit thin films. So the electronic material, we typically use sub-micron thick materials to actually form our electronic structure, and at the end we can relieve the sample from the carrier structure and have a soft and compliant electronic system. So this was for the technology. So then we went together with the team of Grégoire to actually validate this technology into first an animal model. So the video you'll see now is on one of the rats that we have implanted with this technology. The rat here has suffered a spinal cord injury, so he cannot walk. Here on the left, you have a cartoon of where we apply the stimulation. The green and the red corresponds to the two legs of the animal, and you see that when we alternate the stimulation left to right on the spinal cord, so the precise location where we deliver the electrical stimulation, the animal can recover use of its hind limbs. So the animal does not decide when it wants to walk. We decide when we apply the stimulation, but that's how that demonstrates that the system can function. So the next step was, well, how do we actually move forward? So this is one proof of concept into a rodent model. Now we need to bring the technology to a higher level of maturity in order to translate it ultimately to its clinical trial. And so the next, in neuroscience, one of the next animal model is the use of a non-human primate to actually explore really therapeutical potential of the technology. And so there we actually came up with this translational framework that is represented here, where we decided to really test as much as possible before going in vivo the stability and the robustness of the technology that we were developing. So we leveraged advances in advanced manufacturing, like 3D printing, in order to actually have in-house models of the spinal cord. So what you see here in the center, you have an artificial portion of the spinal cord. So these are the vertebrae. We can also print a mock-up of the spinal tissue, so the soft tissue, and insert our implant at the surface of this mock-up. And then we've also engineered a biomimetic environment where we could actually test the overall system at 37, so in similar condition to the human body or the body in general. In PBS is a saline solution to mimic the ionic environment within which we would use the device. And in the final modulation that we've applied is actually mechanics, because in the cervical area or even on the spinal cord, this is also a very mobile structure. So we had to actually implement this type of deformation in order to see whether our technology would sustain this type of deformation. And then the final element is to actually integrate with the electronic stimulator so we can ensure that the electrodes deliver what they're supposed to deliver to the tissue. And then, of course, as you progress in the fabrication of your devices, you do systematic failure analysis, and if it doesn't work, you re-optimize, reprocess, etc., until you have sufficient level of maturity of the technology, and only then you can move to the higher animal models. And so just to give you, again, a more concrete idea of how we did this, so we did this very systematic technology testing in the context of the cervical area of non-human primates. So we did MRI images of the animals in two positions for the head, extended and turned. So we had the min and max deformation of the cervical. Then we printed our model, we inserted our implant into the model, and then we inserted the whole system in our multimodal biomimetic platform. And this is how it looks. This is in an incubator, so we maintain everything at 37. And here what you're looking at is really the typical movement that the head, or actually the neck level, would withstand. And with this, we were able to really collect information from our implant. What you're seeing on this graph is the impedance of our electrodes for multiple generations of the electrode. And the take-home message here is to see that after 500K, technology 1, actually half of the electrodes were failing, so we had to revisit and improve some of the technology so we would have a better stability of the device. So this is where we are currently with this soft technology for stimulation of the spinal cord. We've now implemented this throughout the spinal cord, and we've validated their functionality in all of these models. But now I want to conclude the presentation on showing how Grégoire and Jocelyne have now taken the concept to the clinic. And with this, they had to actually change the electrode technology, because one big challenge that we have, and I think many of you here understand this, we've had these comments already throughout the last 24 hours, is that translation takes time. And here what I've shown you with the introduction of not only new materials, but also new ways of producing this implantable technology, it's actually very long to bring this technology towards clinical reality. So we are currently doing this. We've spun a company who is now going through all of the regulatory steps in terms of biocompatibility, sterilization, etc., to bring the soft electrode to the clinic. But Grégoire and Jocelyne wanted to go faster and test their therapy at in patients. And what they've done is actually repurpose existing clinical neurotechnologies. So there is currently a device that exists for the spinal cord that is used for pain treatment. So it's an electrode paddle that resembles the one I showed you before. And so they decided to use this technique, and they collaborated with a company and then spun a company as well in order to actually really program carefully where to apply the neuromodulation. So here is very similar to what I showed you before. For a given patient, they do a lot of imaging to reconstruct and compute the model where we would need to position the electrode in space. So this is a spinal cord with the rootlets. This is now the corresponding electrode paddle that is positioned at the surface of the spinal cord. And then they applied the technique and they implanted the patient with this technology. And here's one of the very first videos that they did. Here you have David, is a patient who has a spinal cord injury. When the stimulation is off, so no stimulation is applied onto the spinal cord, he cannot move his leg. When the stimulation is on, and what you see is the type of patterns that are applied on the left-hand side, you see that he's able to actually use and drive the locomotion of his legs. So now he has, they've done terrific progress since this initial demonstration. They now have a system where the patient can actually use the technology outside, and now he's really only using this small support to help him walk freely here now by the lake in Geneva. And this is really a very exciting and encouraging story around how far you can push technological innovation in the lab. So with this, I'd like to conclude and give you an outlook that I hope that I've convinced you that you can do innovation and interdisciplinary research in the academic environment, and this is really at the heart of neuroprosthetic medicine. I've shown you that preclinical research is essential. This is how we can understand all of the mechanism and the rules and the parameters that are needed to deliver a therapy. Translation towards clinical realities is possible from an academic starting point, but it's very long, so you need a lot of collaboration, you need a lot of people to support also your effort, and you need long-term commitment from various parties. And we, at the PFL, we've had the chance to have such type of support. And here I gave you one example, or actually 3 examples of what neuroprosthetic medicine is, and there's actually a very wide palette of application that go from sensory function to other more brain interfaces that would benefit from advances in neurotechnology. So with this, I'd like to thank my group. We are a very multidisciplinary group. This is, I think now you got the message that we need to conduct research where we go from neuroscientists all the way to computer scientists via the traditional engineers. I'd like to thank my colleague at the Center for Neuroprosthetic at TPFL, Grégoire and Jocelyn at NeuroRestore, and Onward Medical, which is the company that they've launched. We've actually co-founded and is now helping on bringing this technology as a therapy, various sources of funding, and thank you very much for your attention.
Thank you very much. It's really a great pleasure to be here, and I'm really very glad for this invitation to talk today. So what I'm going to present to you is maybe some kind of new brain-machine interface, because we were quite scared by the contact between the tissue and the electronics, and we were not able to design such intelligent electrodes. So we tried to use light and sound in order to stimulate at distance the brain. So in our case, what we do is we're trying to restore vision, because in the last 400 centuries, we have not really progressed to restore vision in the blind patients. So this is done at the Paris Vision Institute, and as you can see here, the idea is that losing sight is the most handicapping, and it's one of the worst handicaps, because you not only lose vision, but you lose also your autonomy, and it's what is the most scaring handicap for people. And what we see is that in the next 30 years, the number of blind patients will double, and the number of low vision people will triple. And this is due to the fact that we have disease due to aging, like age-related macular degeneration or glaucoma. So the idea is that these diseases, they either lead to the loss of photoreceptors, like in age-related macular degeneration or some retinal dystrophies, or they are due to the loss of retinal ganglion cells, the cells that send the information to the brain, as in glaucoma and diabetic retinopathy. So I just introduced here the macula, because I think it's a very important area, because it's where we see with high visual acuity, and it's where we always put the image. So, and you, for instance, see when you're watching a face, you scan with your fovea all the area, the important area, like the eyes, the mouth, and everything. So the idea is that we want to restore vision in this very specific area. So at the Paris Vision Institute, we are trying to address all the steps for ocular disease, from the early stage with, for instance, gene therapy, which is for Leber optic neuropathy, now close to be to get the authorization to put it on the market. But what I want to do is try to convince you that we can now even address blindness in the patient. So the idea is that we could, when patients are losing photoreceptors, try to, introduce some visual information in the retina with a subretinal implant or an epiretinal implant on the surface of the retina. It's more complex when patients are losing retinal ganglion cell because they lose the optic nerve, the contact between the eye and the brain. So then you need to stimulate directly at the visual cortex. So we have first started to work on restoring vision in patients losing photoreceptors. So the state of the art in the last 10 years was the American prosthesis second sight from the company second sight, the Argus 2. And what you can see, it's similar to what Stephanie was presenting for cochlear implant, where you have a system connected to an external device, and then you could stimulate some electrodes that were on the epiretinal side of the retina. But in fact, not all of these 60 electrodes were really generating a pixel. And so if you watch an image, already you can immediately see that with 60 electrodes, you will not recognize a face, and you can use it, but it will be for very limited kind of vision. The objective is to go to more than 600 electrodes because you can already detect or recognize a face with this type of device. So we have been working, and especially we worked with a device developed first by Daniel Palanque at Stanford University, and he agreed to produce this implant here in France in our startup Pixum Vision. So this implant is, you see, completely wireless, so it's just a piece of silicone, and each unit here, oops, sorry, I made a mistake, is composed of photodiodes surrounding an electrodes, a central electrode, and you have a ground grid where the current is returning. So the idea is that you can stimulate with light each of these units so that it will not only provide the information of which of these units have to be activated, but also the energy to activate the electrodes. In fact, the photodiodes are infrared, so you can really stimulate with quite high energy this implant. And so at first, Daniel had shown that it's possible to use this device and activate in an isolated system like this, an isolated retina, blind retina from a rat, and even in vivo here by recording the visual cortex. So at this stage, we wanted to demonstrate that this could be useful for patients. So we decided to go to non-human primate. So we first did similar experiment as Daniel did in this kind of system on a non-human primate retina, where we had removed the photoreceptors with a razor blade. But then we moved to the in vivo condition, and you see that we could implant this device in the non-human primate. What we see is that the photoreceptors are degenerating above the implant simply because you separate the retina here from the choroid below, and so the photoreceptor no longer receives the oxygen and all the metabolites. It's not that the implant is toxic, it's just that they need the contact to the choroid. So in fact, we have a blind spot just at this position, and when we did some kind of behavioral studies, you see that the animal can generate visual saccade everywhere, except when we stimulate with light the implant, because the animal is blind at this position. But if we stimulate with an infrared pulse, then it can generate saccade, so it means that he's seeing with the infrared sensitive implant. And it was sufficient to stimulate only one unit of the implant in order to elicit this behavior. So this meant that really we could provide some vision with this implant, and it was then possible to move to patient. And you can see here the patient are now able to read letters. Here they can read words, and their visual acuity is close to 1/20, so 20/460. So it's very close to the level of legal blindness. So we could almost consider that these patients are seeing again. And what's also nice, in this case, the goggles were completely opaque, but now we have new goggles where you see this black rod where you have a camera in front and you have a video projector projecting an infrared image here on the implant, so that the patient with these transparent goggles, they can fuse the infrared image generated by the implant with the natural peripheral image. And I just want to remind you that the peripheral vision is very low. If you try to count your fingers, if you put them on the side of your face, you will not be able to count your fingers. So it's clear that in the periphery we have very low vision, and the idea is that we want to provide better vision in the center. So in this case, we have still a margin of progression, so to improve further the implant, but in this case we have already 370 F of the unit on these implants. So this is now in phase 3 in a multicentric clinical trial in several European countries and also in the US. So, but we were not happy enough, so we decided to move to another strategy. And this other strategy is what is named optogenetic therapy. So optogenetic therapy has been a revolution in neuroscience because you can stimulate neurons with light. And so the idea is that in some algae, you have a protein, an opsin, that is sensitive to light. And so when you stimulate with light, you open a channel in the cell membrane. So what we decided is to take the genetic code of this protein, insert it into an AAV, inject the AAV into the eye so that the AAV would diffuse to the retina, and neurons, residual neurons, after the loss of photoreceptors, will become photosensitive. So in fact, we turn these neurons into kind of pseudo-photoreceptors. And so others had shown that it's possible to do this, and this was done by Zhao in Detroit, but we moved to non-human primate because we were interested to see whether we could bring this to patients. So you see here the result from Zhao in Detroit, where he could express one of these channels in retinal ganglion cell and show that it was possible, even in this blind mouse retina, to elicit some spikes. So we moved to non-human primates, and the idea was to express the protein into the retinal ganglion cell. Here, you see that we could express this in the macular area. You see this nice macular ring here. If we take this retina and put it on a multi-electrode array like this, we can record the spikes and show that when we stimulate with light, we have a big increase in spiking activity, although we added some synaptic blockers. So this is clearly not a natural activity, it's really an optogenetic activity. And so with this, you can show also that the activated area is really the activated and the area that was transfected. So in this case, it was an opsin that is sensitive in the blue range. We know that blue light is quite toxic, so we moved to a red range opsin, which is crimson air, and you see that we selected the best AAV, we selected the best protein in order to translate this to patient. And you see here the results. You see that with this novel opsin, we have a nice relationship between the moving bar here that was slowed down 4 times just so that we see the activated electrodes, and you see in orange the electrodes that are activated by this moving bar. So it's clear that we have a nice spatial resolution. We could show that we would have a resolution that is much better than what we have with the retinal prosthesis. And what was also nice was to show that, in fact, we reached the plateau of activation of the retinal ganglion cell with a light pulse of 20 to 30 milliseconds. So it's suggesting that we could generate a video rate stimulation in the patient. So at this stage, we just needed goggles because these opsins are not very highly sensitive. So we will need to normalize the light level in a high range, but then we can move to patients. And so there are already patients that are seeing with optogenetic therapy. So I apologize, it's in French. So the patient is seeing with kind of flickering because we have an event-based camera that underlines the edge of the object. So this is the first time a patient is seeing with optogenetic. So it's a first in the world to have, in fact, a patient with neurons activated by optogenetic therapy. So the patient was blind for 40 years due to retinitis pigmentosa. So it's quite slow because he has to really learn again to see with this new kind of vision. So there's really a plasticity in the brain in order to learn. So we now are moving our interest to patients having lost their optic nerve. So you see that there are already some clinical studies where either with a surface electrode, the patient could see some letters, but you had to do a very sequential stimulation, which is not nice if you want to have locomotion or any kind of fast vision. And then there was another study with such penetrating electrodes, and where the patient could also see some letters, and it was much faster. But the difficulty with these penetrating electrode is that they generate a glial scar around the electrode, and after some time you lose completely the activity. So we wanted to do something that would be more kind of distant stimulation. And at first, of course, we thought, oh, optogenetic therapy, of course. But unfortunately, the light is not really diffusing in the brain, and also we have the dry matter above the brain, and when investigators are removing this dry matter in non-human primate, it's often that they lose the primate in a few months or a few years. So we decided to try another strategy, and this strategy was, we thought about it with Michael Tunter, where we had done some recording with ultrasound, fast ultrasound imaging. So maybe you've heard this morning from Matthias Fink about these incredible methods of using ultrasound. In this case, you can use ultrasound to see the brain, and thanks to the measurement of the blood flow, you can, for instance, do this kind of mapping of the retinotopy of the visual cortex, or here you can see also the ocular dominance column, even very deep in the visual cortex. So we thought if we can do imaging, why don't we do stimulation? But in fact, when you stimulate the brain with ultrasound, you need very high intensities. So we needed something to make neurons sensitive to ultrasound. And that's why we have selected a channel that is mechanosensitive. Again, we introduce the genetic code in an AAV, inject the AAV into the brain so that the neurons, they become sensitive to ultrasound. And the idea would be that the patient would be wearing some goggles, making images, and that then we would generate some ultrasound images, ultrasound pattern on the visual cortex. So is it completely stupid? So we started to do some tests in rodents. So you can see that we can express our ionic channel in the visual cortex of rodents. We can do stimulation, and this is the light response to a natural stimulation. And this is the response to an ultrasound stimulation on the visual cortex. You see that we have a very similar dynamic, but the only difference is that the latency is much shorter. Of course, because we have stimulated directly on the visual cortex, whereas when it's a natural stimulation, it needs to come from the photoreceptors up to the visual cortex. So it's why it's much shorter when we do the stimulation on the visual cortex. Then using some ECOG arrays at the surface of the brain, we could record some local field potential induced by the ultrasound. Of course, if we did not inject the AV, the ultrasound were not having any activity. And then we could move the ultrasound stimulator, and you can see that we are moving also the ultrasound activated area. So this means that we have a quite good spatial resolution. So we have the temporal resolution here, we have the spatial resolution, and all this is highly compatible with visual restoration. If we stimulate with ultrasound the visual cortex, is the animal having the perception of a phosphene? So we did, we generated animals, and we learned them to associate a flash of light with the delivery of water. And the animal is learning in 4 days. You see, it's learning quite nicely. And then they lick the tube as soon as we do a flash of light. And then here we started to do an ultrasound stimulation of the visual cortex. And you see that it's very similar to a flash of light. So it's very likely that the animal is perceiving light when we stimulate the visual cortex. And if we go just a little further, if we enhance the ultrasound pulse, you see that the behavioral response is increasing, and also when we record at the level of the visual cortex, we see that when we increase the pulse, we increase the activated area. So this is very like when you do electrical stimulation of the visual cortex. When you enhance the current injected, the visual phosphene is much more intense. The final thing is that when we did the behavioral stimulation, I was really amazed by this result, because what you see is that the latency for the behavioral response is much longer here with a natural stimulus than with the stimulation at the visual cortex, the ultrasound stimulation of the visual cortex. And this will remind you this image, of course, because due to the stimulation at the visual cortex, you shunt all the way from the eye to the brain. So it's why it's much shorter also for the visual response. So we hope that we can now proceed. We are already moving toward non-human primate, and the idea is to try to move to patient. So I hope I convince you that this kind of different brain-machine interface, I mean, we have the retinal prosthesis with a visual acuity which is close to 1/20. The alternative solution with optogenetic therapy, where we hope that we could get to the cellular resolution and so to a much better visual acuity. And also for patients losing the optic nerve, we will try to develop this visual restoration at the cortical level, but what we call sonogenetic therapy. So it's a kind of new era of brain-machine interface that are more kind of genetically based brain-machine interface, but they provide some kind of distant activation of the brain. So with this, I just need to thank all my collaborators, and especially José Sahel, who really started this whole project of visual restoration at a time where we were such a small team. I could not expect that we would be here today. And also some very good colleagues like Michael Tanter for all the sonogenetic stimulation, but also Burton Roska for optogenetic therapy, and Daniel Palanca for the prosthesis. And of course, GeneSight, who developed the optogenetic therapy, and Pixum Vision, who is developing the prosthesis. But thank you very much.
So, dear friends, we have just gone through some fascinating research and engineering proposals, traveling with Einstein and Heisenberg, curving space-time, exploring cutting-edge digitalization and modeling, and repairing essential functionalities of human body. I'm not a specialist in ethics, but my particular career as a medical doctor, a researcher, an astronaut, a research minister, has driven me to go beyond the scientific and engineering facets of our developments, trying to constantly look to the planet Earth and the humanity it carries in the solar system with a global, humanistic, and long-term perspective. And I think that while exploring new territories, what we did, and their limits, it's essential to take a certain distance to enlarge our perspective. And I'm really grateful to the steering committee of CAETS 2022 to have taken with enthusiasm, I would say, this possibility to integrate ethics session and raise awareness about the impact, the societal impact of all this breakthrough in the middle of this stage of the art work that has been presented. So many thanks for having this possibility to have this session today. And I would like to warmly thank Christiane WOOPEN, because she has been there for the 2 days. She did a wonderful and relevant introduction, opening speech, and she accepted to be with us on this panel, to be the facilitator, moderator of a lively discussion and interactive discussion with distinguished members that I will call to join on stage. So Patrick COUVREUR, Virginie Dignam, Alison Noble, et Thierry Magnin, please.
Thank you very much, Claudie, for the introduction, and a very warm welcome for all our panelists. This was a long day, or 2 long days, and there's just 1 session left, and we will not do the whole discussion here on the panel, but open the floor for your questions or comments as well after we have discussed some ideas and questions. So the idea of Claudie and myself was that we first have a very small round with let's say 5 minutes each, so that all the panelists can introduce themselves, apart from what is in your booklets anyway, and say how ethics and social impacts is influencing your view on technologies, or what kind of role these questions play in your perception and hopes for breakthrough technologies. May I give the floor first to Thierry?
Thank you very much. I am a physicist and a theologian, and I was first involved in solid-state physics regarding to the behavior of materials for nuclear industry. And already I was asking a lot of ethical questions in this field. And then as a theologian and particularly involved in moral philosophy and ethics, I was moving to bioethics and to the ethics of technosciences, digitized technosciences, such as nanobiotechnology, neurotechnology, and the influence of artificial intelligence and data treatment on this. One of the major experience for me was to be included to work ethics inside 2 consortia, Toulouse-White Biotechnology Consortium with universities and industrials, and the second one related to Metagenopolis, a consortium related to the work on gut macrobiota and the relation with health. And my job was to be discussing about ethical questions with biotechnologists directly in their daily life, I would say. My job was to introduce to them what is ethics, how we can try to define ethical questions and dilemma, and then to try to ask the good questions they can ask themselves from their daily work, and then to be able to elaborate some solutions and some proposals together. And that is very interesting for a philosopher to be able to make this kind of ethical questioning through the daily experience of biotechnologists. And I think that for research and for the study of ethics and for learning ethics at school or in an engineer school or at university, we need to introduce ethics inside the program about technology, science, or economics, or lawyer, and so inside, and not when everything is done, and then we take some time to reflect about, but I think it's inside. And I see, and I will finish my presentation by that, as a professor at Catholic University in Lille, where you have engineering school, business schools, medicines, and a lot of different formation. We see a more and more increasing number of students. They want to have a job inside which they will find a sense. They will find a sense to their job and to the service to the community and to the society. And the ethical behavior inside the job is one of the major components to find a sense, and I think it's now very important, not only the questioning when you have time for. I think it must be included in the research of real progress.
Thank you very much.
Thank you. My name is Virginia Dignam. I'm currently a professor of responsible artificial intelligence, whatever that may mean, and I suppose we'll discuss that in the panel for some time at Umeå University in Sweden. I have been working in artificial intelligence all of my professional career. First, since the beginning of the middle of the 80s in industry, I was developing all types of AI systems in the 80s, 90s, and beginning of the 2000s. So before all the big hype and the big developments in data-driven AI appeared. And after that, I have been working in academia. Actually, the work I do in academia and the research and the policy work I do nowadays is mostly driven by this experience that I had in academia. I just out of university. I went to work for a ministry in Lisbon, where I was working at the time. Working at the time to design an AI system which was going to determine the priority of applications for social housing in the municipality. So who should get the priority for houses that became free at the social housing program. I develop also systems for supporting insurance agents developing and determining the veracity and the importance of insurance claims. I worked for the car industry to develop systems that verified automatically the configuration of cars before they get into the production line. So I did all kinds of different AI applications. Some of them, they failed miserably. Some of them were a bit more successful. Some of them even led to cars that are still moving around in our roads. But from this experience, really showed me that whatever design, whatever research, whatever models we develop do have a very concrete impact in society. Especially those projects that failed for all kinds of reasons really showed that
the designs, the engineering systems that we build do have a societal impact, and the design of these systems is political. The choices that we make, the assumptions that we take, the ones we include in the design or not include in the design, all that will determine the system that we develop, but also the impact of that system. So that's what led me to go do a PhD later on, at the beginning of the 2000s. And since then I have been really working both in the research but also in policy, exactly to support researchers and practitioners in understanding and developing the tools to support this type of understanding. How can we, before we even start designing something, before we start developing a system for whatever reason or whatever application, ask ourselves why, ask ourselves what kind of potential consequences are in these systems, and this leads to a need for a much more multidisciplinary approach than just trying to optimize the engineering of the system. To build the best engineering possible system is not always the system which is really going to have the best beneficial impact in society. So bringing those things together and working with the policymakers and the researchers on understanding what kind of values are relevant for developing systems? How do we translate in one way or another based and understand the context in which these values are important? And how do we translate those values in context into some clear and workable and implementable functionality for the systems? Also understanding that by building the system we are changing the environment in which the system is being applied, and really helping with the design and understanding of this type of trade-offs. Because I'm not an ethicist, but I think that this issue of understanding the values that really matter for society and how to address those trade-offs between the values as an engineering discipline, it's the kind of things which has led most of my work in the last 20 years.
Thank you.
Thank you. My name is Patrick COUVREUR. I am a professor of pharmaceutical technology and biopharmacy at the University of Paris-Saclay. And as you have seen during the first session, I am specialized in the use of nanotechnologies for targeting disease tissues, cells, and also subcellular compartments to deliver drug at the right place and on the right time into the body. So I confess that I am a little bit sightseeing probably in terms of ethics, to be frank. But I can say that in my daily life in the lab, you know, I, as a director of a doctoral school also, I was always very sensitive to the education of the young people. Because I am convinced that actually there is a very big pressure in general to the scientists to be the best, to publish more and more in the best journal, etc., to attend to the International Congress. And for the young people, it can be a problem. And I am always trying to moderate, I would say, the pressure I am putting to the young people and to educate them to deliver, if I can say, fair results, fair data, which are reproducible, and not to deliver data that the boss is asking for. I think that it is an important point in the terms of ethic. Then, from the other side, apart that I am an academic, I am also a pharmacist. So I was educated to think about, to develop medicines, drugs, in a way that they are safe, of course, and they are able to treat severe disease. And so I think that from the point of the ethical point of view, it is always important to think about what I am doing. Is it useful for eventually be translated towards the clinic and to the benefit of the patients? And to do that, you have always to think about, when you are developing a new technology, what is the relationship between the risk and the benefit for the patient. And for sure, each drug, each medicine you are taking, you have always a risk. But the life is also a risk. So you have to take some risk. But I would say that the benefit has to be much higher than the risk. And this can be a conducting thing which has to conduct the research of the people in the field of the drug discovery and the drug delivery. Of course, afterwards, probably I will speak about specifically in the field of the nanotechnology, what type of problems concerning ethics we can meet, but at the moment, I will stop here.
Thank you very much.
Hello, I'm Alison Noble. My background is in medical image analysis, so that's really the intersection of computer vision and medical data. And for me, the big transformation has really been happening in a remarkably small amount of time. It's over the last decade. It was 2012 where AlexNet, which was a convolutional neural network architecture, exploded the field. And so I think sometimes we don't reflect on how far we've come in 10 years, and that's really important. We might also want to be thinking about where we'll be in another 10 years. But we've really gone from having an algorithm to now having deployed systems in healthcare. And whereas a decade ago, I would say I was an interdisciplinary researcher, I was working with radiologists, I work a lot in ultrasound, so I was working with the sonographers. Today, we have brought social science in to start to think about these questions that I think you've heard a little bit about yesterday as well. So we have to think about medical ethics. We have to think about, are things safe? Does the technology provide access? And we haven't really talked much about access and the importance of that. We'll have to think about AI ethics issues. We have to think about what is the bias, potential bias of a model. If I use my model in a new context, will it work? That's clearly very important. And another emerging area is thinking about the changing relationship between human and machine. Today, we think about AI models being used for prediction to support decision-making. But you can flip that role, and you can get AI models can teach people. So there's real questions in the future which will become ethical questions about what do we feel comfortable with as a society, as medical professionals, as patients, in terms of how we will interface with technology. And the final area I've become interested in as we've deployed solutions is data ethics. And I've been particularly working with the Royal Society in the UK on looking at privacy-enhancing technologies. And you heard a little bit about those yesterday. They are a whole class of different technologies that allow you to not copy the data into your institution and process it, but how do you think about data that might be remotely, you can then process it using, for example, federated learning. And how does that allow you to balance some of the risks of data sharing, but benefit for good in terms of how data is used? And the Royal Society published a report looking at perceptions on this, reviewing how the technologies were emerging. And for me, quite interestingly, how these types of technologies are emerging in parallel to how artificial intelligence methods are emerging as well. And they're starting to intersect. And I think as they do, there'll be some other issues that will be quite interesting, related again to the ethics of how things go forward.
Thank you very much. I think it's clear that here is sitting a whole bunch of experiences and a universe of ethical questions that we have to follow up now. I want to start with some social impacts and two concrete examples. So for the pandemic, we have seen that breakthrough technology like mRNA vaccines have brought a major tool for managing the pandemic. At the same time, this technology was not combined with a governance system around it that granted access to this technology worldwide in an mainly equally way. That led to more hunger, more poverty, trillions of lifetime losses in earnings for young generation because they lost education, even inner country migration in the global south, rise of authoritarian systems. So having a breakthrough technologies combined with a bad governance around it, if I may put so bluntly, yeah, then a breakthrough technology can even enhance inequalities. Yeah, so vaccines actually also, although being a great success, enhanced inequalities globally. So that's one issue I wanted to raise. And the other one, having gene therapies, and the most expensive one now for 2.8 million euros for one, or dollars, don't mind, for one patient, is also something that that societies have to decide whether they want this. So it's a breakthrough technology, and for this patient it's life-saving, and for the family it's beneficial. But does a society want to take up these costs for one patient? And what about all the others who need that money as well? So if I may, Alison, start with you. Do you want to comment on that? Because that was already in your presentation.
Yeah, I guess from that particular example isn't my area, but I think it is an issue that doesn't have, it's complex, and many of the ethics issues are very complex. I think you can say, why didn't you do another approach to providing access? But there are also issues in maybe doing that as well. So there's always, you have to be thinking through the scenarios. I think there are other examples in the pandemic that I think maybe highlight similar issues. As an imaging person, the radiology was another one where that was used. It was also evolving because no one had to work within a pandemic before. And people tried to model things as well to help. And that turns out to be not very helpful because the models were not stable. And there are a number of articles published as well. But I sort of dodged your question because I don't really have an answer to it. Perhaps someone who comes more from that side, but being able to think about COVID therapies might be able to comment. I'll pass it that way.
But I think ethical questions are not there for immediate solutions. Otherwise, you would get a Nobel Prize or something like that. Patrick?
Yeah, I have absolutely no answer to your question because it's a very complicated question. I would say that the problem of the gene therapy and the COVID vaccine are facing not exactly the same limitation and the same problems in terms of poverty, etc., etc. I think that with the SARS-CoV-2 COVID pandemic, there was an urgency. And it was already fantastic that a vaccine could be discovered so rapidly with really a breakthrough technology. But behind that, of course, there are, I would say, the governmental responsibilities to choose from a political point of view. What will I do first to take vaccines for my own country or area? Or I will share it with poor countries in Africa and in other developing countries. Of course, it's very difficult to decide about this. I am not a politician, so I cannot answer to this question. Now, the problem of the gene therapy is another very important problem, which is less urgent, but also from a technical point of view, complicated. Because you know that more and more we are going towards a personalized medicine. This means especially in the field of the DNA delivery, and gene therapy. This means also that the market, I am not a specialist in that, but I heard from the people from the industry that the markets are becoming smaller and smaller. And this is not only because of the technology, but also because the markets are so small that, of course, the cost is increasing in a terrible manner. And of course, there is a need to have big discussion again between the governments and the companies, the pharmaceutical companies, to see what is necessary, what is needed in terms of cost, and what is exaggerated. And once again, I cannot myself answer to that complicated question.
I also will not try to answer your question, but actually your question raised in me many other questions, so maybe that's something to go. And I think in a sense is, in one hand, a matter of dialogue between all those that have a stakeholders in the issue. Often those dialogues or those discussions or those trade-offs, it is a trade-off. You have to decide, do I give the vaccine first within my country, or I share it with the whole world? Do I solve the medical problem of one single person or of the many other one? How to divide costs? So it's always a matter of trade-off. And probably, like in many other issues of trade-off, there is not one single answer. And one issue which I always get very unease with is the idea that somehow there will be a way to engineer ethics, so that ethics is something which can be solved as an engineer problem, and that people who know about all kinds of things will come with the answer. And that gives me always a very uneasy issue. I think it's the most we can hope, the most we can demand is some openness about who has been involved, what kind of powers, what kind of stakeholders, what kind of assumptions, what kind of choices have been there in the decisions and in the solutions which have been taken. So I think that we have to take much more a question in terms of not so much what is the result, whether person A or person B gets gene therapy, whether country A or country B gets the vaccines, but demand much more an openness of exactly what were the powers, the trade-offs, the stakeholders, the ones involved in the discussion, because only from there we can learn, or we can try to understand how the results have come there. The result in itself, it's I think not so much the main importance in the whole process. It's a matter of process.
I am not going to repeat what they said, so just perhaps some other fields. The international solidarity, that's what you said, and that's quite important, very difficult to regulate, I understand that. But I think also inside the country, in ethics, one of the important points is justice for social institutions. And I understand that solidarity is very important, but there are some priorities inside the choice about solidarity, even for a country, for our Western country, for instance, which are rich enough to practice that. So what are the priorities in terms of health? We have to choose. That's a political question I'm not going to answer, but it's a political question. It's very important, in fact. The second one is, to my opinion, the pandemic with the vaccine, there are other points with social impact, and particularly a kind of suspicious reactions in front of propose technologies and vaccines. So we can be afraid about these suspicious points. It's my case, but it is in fact in the society. And we can ask why the impact of this vaccine has also a part which is anxiety or a reject of the vaccine. Even if sometimes the reason for rejection looks completely crazy, but perhaps not so crazy in a kind of way to think the world. And the authority of scientists and the authority of politicians were challenged, I think, in our countries. And that is a question also, we will speak more about that perhaps later on, but how the population is taken into account in the elaboration of the political points. Even if it was very urgent to be able to answer to this pandemic, I understand that. But we cannot decide from the top to down in our kind of democracy. It does not work. So it's also a question of justice and the co-elaboration of some project for institution. I think that point is also a kind of social impact. And I think to finish this part, I do like technologies, and I think that technologies will go on to really participate to the freedom of people, and particularly in the health system. But everything is not coming from technology. And when our promises are to focus only on technologies, then there are some problems too.
There is in Germany the saying, I don't know whether it exists in French and English as well, if someone has a hammer, every problem is a nail. Yeah. And sometimes I have the impression that's with technologies as well. So if you have a technology, you think that all of the world can be solved with technologies. I came to this problem when thinking about social freezing of oocytes for women who want to postpone their family planning because they want to do first a career. And there were even companies financing social freezing for the female employees so that they can first follow their career. And I think, well, that's probably not the best thing to do, because we could have social solutions to empower women to follow their career and have children early enough. So why intervening in the female body and collecting oocytes with the hormonal stimulation and all the risks that go with that, and postponing family planning? So if it is autonomous decision, okay. But a society that puts this pressure on professional careers and says, well, we have a technological solution for that, is a framing of a problem. And it's a technological framing of a social problem. And I think this is important as well when dealing with climate also. Yeah, we do not really act properly with our environment, and we have these huge problems with climate change. And instead of changing our economies or our way of producing something and to have competition in the free market, we use again technological solutions to deal with the climate. So I think they are important, but they are not the only access to solutions. And to think in broad socio-techno-economic ecosystems is what is the challenge for our societies. And that comes, I think with the vaccine or the pandemic as well. So vaccines were a wonderful solution for parts of pandemic management, but not for the whole pandemic management. But politicians, not the scientists themselves or technical experts, but politicians framed it in some countries at least, once we have vaccines, the pandemic will be over. So this technological storytelling can be really part of the problem, and that raises a question for the stakeholders all of you mentioned. Whom should we involve to deal with these ethical problems? How can we do that in a society and then on a global scale? Yeah? So how can you imagine to have really a citizen participation in framing our socio-technical economic ecosystems? I think, Patrick, you want to say something from the nanotechnological standpoint?
Yes. In France, at least, probably Claudie remember it was 10 years or 15 years ago, there was a sort of citizen debate. You remember about that with nanotechnology. People were fighting one to another. It was terrible. It was citizen debate, you know. The problem is that we see now that 10 years or 15 years afterwards, we have a sort of assessment step back about the toxicity of nanomedicine, the activity of nanomedicine, and we see very well that we have actually 40 nanomedicine on the market, which are working not bad against cancer, for instance, also against the pandemic with the vaccine, because if you have not the nanotechnology to protect the messenger RNA from the degradation allowing to enter into the cells, you have no vaccine. So you see, 15 years ago, we have the experience that the relationship between the benefit and once again the benefit and toxicity is largely in favor of the benefits. So what to say more about that? Of course, what could be in this field an ethical problem is that with nanomedicine you are looking to repair body, because you can do also body reconstruction by using some type of nanotechnologies, or the treatment of severe disease. And I think from an ethical point of view it is okay. I think that's my opinion. But what could be dangerous would be, of course, to go one step further, looking if you can increase the ability of the body up to the normal value, I would say. To increase, for instance, the mass of the muscle by injecting steroids or molecules like that as nanoparticles to improve the delivery of this type of molecule. This is true also probably with some anti-nociceptive peptides or erythropoietin, which is used for the people making bicycle, you know, Tour de France, etc. So this is transhumanism. And I think that from an ethical point of view, we have to have a red line, a frontier in between restoring the health and not improving the normal capability of a human, of an individual. This is my opinion on that.
Do you want to comment on
Yes, maybe not so much on this last part on the transhumanism. It's not at all my area. But going back to the idea of participation, I think that one thing you said, which is I fully agree, is that there is not a technological solution to all problems, and trying to solve everything by technology, it doesn't really lead to nowhere, let's say. But at the same time, we also have to realize that there is also not one single social solution or approach to all the different problems and all the different questions. So in terms of participation, I think we really need to take a multi-step and multifaceted approach to participation. Of course, this type of dialogues or community participation in terms of referenda or discussions is one way to go, but we can kind of see it in a more layered way. with starting as a top layer, accepting and assuming that human rights charter is kind of the leading all societal but also technological development. So we need to take the human rights as the most, at abstract level, the most leading direction. But then there are, of course, translating human rights into technology or into societal solutions is extremely complex, and it needs to be taken in a contextual way. So participation starts by taking the role and extending the role of democratic process and democratic institutions. And I know that across the world we are seeing some type of tiredness or problems with also our democratic institutions, but democratic values, I believe, need to be core to participation. And then understanding that there is not one size that fits all. So we do need to accept that the way things are implemented at different regions or different countries or different people in itself, we need to have that type of flexibility and understanding and accepting the different levels of participation in that way. Because, like I say, it's not like trying to get everybody to agree on one single solution to everything. It's also not the way. But if we go in this type of higher levels, then more concrete, contextual decisions or procedures, and then still accepting the individual determination to take things in one way or another. That's probably the best way to ensure participation. And accepting that not everybody, there is not one institution or one person who knows everything. So we do need to take different disciplines and different sectors into consideration.
I would have a question for Alison, if I may, then following up on that, because now we have tried a little bit to contextualize technologies and say they're part of the story, yeah, but they cannot make the story on their own. On the other side, participation could also mean to convince society or to make society understand what technologies can contribute, to have an innovation ecosystem, yeah, fostering really good technological contributions. And what can the technological experts, companies and so on do to close the gap between those who just don't understand anymore what is happening? And not everyone has to understand all the complicated things that were explained during the last 2 days, but at least the principles they can understand. Yeah? Everyone uses a smartphone, and I would say 98% of people using that don't know how that works. Yeah? And that can be with health technologies as well, but there has to be kind of confidence that it is not harming myself. There has to be trust around the governance systems and around the people who deliver that and to produce that. How can we bring society and technologies closer together, though, so that they can benefit from each other?
Well, I think there's a big role here for many people in the room. It's in terms of the learned societies, because they're often very good at explaining things well to the public and better than any technologist would. And that becomes one of communication. I think the other thing is to make sure that you do have links with policymakers as well, who again, you often say, if you're pitching to a policymaker, the age you'd pitch to would be more like 13, 15-year-old. And so if you can communicate the benefits at that level, people are going to understand. They'll be able to ask the questions. And that becomes then trusting. And that's again why I think the learned societies have a role here. They are deemed to be experts not representing governments, and so there's a real role there to try and explain some of the breakthroughs and maybe work with some of the other stakeholders as well to get the message out through trusted sources. And if people read about case studies, they read about success stories, they will then connect. And then they will start to understand and appreciate what new technologies will produce. And I think that's a really important part that's sometimes underestimated, the power of being able to change people's opinions, but change them in a good way, rather than trying to introduce some negative bias.
Perhaps you can take this mic and give this to Thierry, wanted to say something.
Just in the same way, I remember the debate in France about bioethics, and particularly work on embryonic cells and so and so. And I remember that citizens who are not involved in science, who don't know nothing, they ask the good questions, in fact, to oblige the scientists to develop more and to be able to say, okay, we know that, but we have some limitation on this point, whatever the position we have in bioethics. I think that the pedagogical way is not only to explain for a lot of people we can easily understand what we said, but also to be able to say today we know that, but we don't know that. And in a good communication, if you only say that you don't know that, that will be a catastrophic result. But if you say everything is okay with this, no problem, no limitation, I think that you will not have, at the end, sufficient trust for people and citizens. That's one point. And the second experience is when in France we make at a small scale kind of communication on gut microbiota and the relation with health, which is very complicated, in fact, because a lot of parameters can interact. And when we ask to people to give their own microbiota, and the Association of Patients and the citizens who were involved in these meetings said, okay, we are okay to give our, but we want to be associated not only to give our, but at each step of your research, which is very busy for for the scientists. But I think that if we want to make collaboration, that is one way.
Patrick wanted to.
May I add something? I think that education is a key point, because when you don't know a technology and you don't know what is exactly a messenger RNA, for instance, I can understand that most of the people are reluctant about the use of messenger RNA because they are confusing with DNA, with genome, they don't understand exactly what the messenger RNA will do. I think it would be very important to have more lessons concerning chemistry, physics, biology, basic sciences since the primary school. Because how can you really share the value of a technology with the public if the public don't have a minimum scientific understanding of what you are saying? And I think that during the pandemic it was one problem.
Just maybe to add one more issue, I fully agree that education is key to this, but to most issues. But in other end, I was just thinking, I don't really understand how my car works. Even I work in a car factory, but still don't completely understand it. I also don't understand how paracetamol works. But me and most of the people in the world will somehow accept and understand how to interact with this type of systems. I know how to interact with paracetamol, even if I don't really know what it does. I know how to interact with the cars in the traffic and even how to drive one. But so it's not so much about understanding all the ways and the functionalities and the way that technology works, but really more about the functionality of this technology. And in that, I think that there is a big role, I agree, for the engineering societies, the national engineering societies, but also to other types of agencies which really support this trusting in technology, but also not only the trusting in itself. You cannot really trust technology. You need to trust agencies that guarantee or that take the responsibility and accountability for what happens in the correct use, but also what happens when things go wrong. So we do need to have this type of agencies around which support the use and the functionality of the systems that we interact with, without necessarily expecting that everybody has a level of understanding of how things work. And at the same time, we also need to understand that there are different roles and different capabilities to interact with technology. Like everybody can understand how to cross the road or things like that, but we demand a driver's license for those ones that use cars. We demand a doctor's license to do the ones who prescribe you the medicine. So we do need to have also for other types of technology and for the digital technology also to develop this type of institutions and this type of roles which support the trust and in the interaction in itself.
I think that raises an important issue, because it's not only about explaining the technology itself, but the policy framework around that as well. Yeah? So it is not only to understand the device of deep brain stimulation or how this works, but that there's a control, that there's an oversight, that there's a licensing. Yeah? And that applies to artificial intelligence as well. And I want to bring these ICTs up now as a topic, because that is part of almost everything we do at the moment. So artificial intelligence is all around. I wonder if we would talk so much in the public about that, or perhaps in a different way, if we would call it machine learning and not artificial intelligence, or algorithmic systems or something like that. So it's also about this wording. And I want to raise the question how we talk about artificial intelligence, and what is the story that their technical experts, engineers and so on use when talking about, for example, autonomous cars. They're not autonomous. Of course, from engineers' terminology, I understand that, but from the ethical point of view and from the awareness shaping in the public, you say cars can do the same as humans. They can decide whether they want to be a grass cutter or a car, or if they want to go to Portugal or to Africa. But that's not the kind of autonomy, of course. Yeah, they automated cars and they do automated driving, but not autonomous one. Or if they say artificial intelligence decides something. Artificial intelligence, it cannot decide anything, because there cannot be a value weighing. It cannot give reasons. It cannot contextualize or so. It can infer. It can count. It can deliver some results, but it cannot decide something. Only if we allow an AI system to automatically lead to specific solutions. But that's not a decision. Yeah, and I wonder if what your vision is on how we implement all the wonderful things AI can do in a society that is disrupted between visions and dystopias.
Well, give it a go. Artificial intelligence, as we speak about it, is not only the algorithmic system. I like your definition. If we talk about algorithmic systems, is a completely different impression or feeling than when we talk about artificial intelligence. But the way that we use the term artificial intelligence in normal language is not about the algorithmic system. It's about the social technical system, which includes the algorithmic systems, but also includes the people, the organizations, the institutions, all the ones that develop, that use, that govern, that manage, that implement, all those. We cannot separate the algorithm from the social technical context in which is being used. And indeed, words are all very unfortunate in a sense, because we try, with all these words of intelligence or autonomy or learning are all very loaded words in terms of their meaning for people. They've been used usually only for human contexts, and now we are using it as a way to refer to some technical capabilities which in some very tenuous way might be understood as intelligence or as a neural. There is no neural in a neural network. There is no intelligence in an AI system. There is no learning in a machine learning system. But those words really make things difficult, and I think that it's very important indeed to talk about and to discuss the narrative of what we are developing and how we are using these terms, because they come, like I say, with a very loaded issue. So. But when we talk about the impact, about what these systems are doing, how the effects are, I think that we really need to focus the issues of responsibility, the issues of ethics, the issues of accountability, separate that from the technology itself and the functioning of the technology, and focus that on those that are involved, those people, those organizations that are involved. So it's not about the algorithmic system itself. And if anything, these systems, they are able to give us a big mirror of what we actually are. By the data that we collect and that is interpreted by the systems, we see it's not the system which comes with the bias or the lack of fairness or the lack of inclusion, is the data that is our past which is like that. And if we, in many cases, it shows the prejudices or the ideas that we have in terms of which data needs to be collected, what kind of data is relevant for what kind of systems, and that comes back in a mirror, like I say, by the systems. And in that we can use the failures or the problems of that we are identifying on the use of AI really to tell us something about ourselves and not so much about the algorithm.
Alison?
I think I agree with you. If we replaced AI by automated systems, which is what a lot of it today is about automation, we would be probably correct in terms of what's deployed, but it sounds pretty boring as well. So I think one of the problems we have is people like the hype and the word artificial intelligence. And I know I've had the personal battle myself with calling the research I do artificial intelligence when I would rather use machine learning or deep learning. But if you communicate with people in clinical medicine, they use the word artificial intelligence. So I try and insert clinical in front of it and call it clinical AI, just to make myself feel a little bit better. But for us to, I think more maybe to address should we be concerned about this, is we do have regulators. And I think the regulators are there to ensure that, regardless of what we call it, the algorithms that are embedded in systems are tested to high standard, and that the claims that will go with what you're automating, if it's in imaging, there are some very good methods for image reconstruction, for example, that will use these sorts of techniques. If we trust the regulators, then we don't really need to worry so much, in my view, about what we call it, because deep down the technology will work.
But regulators really struggle at the moment. Thierry?
I think that artificial intelligence is a very important tool, and we have seen a lot of very good examples during these 2 days. Nevertheless, I am thinking about the example some months ago of this engineer coming from Google, and he was in the persuasion that its own chatbot, he was building this chatbot, and he was thinking that this chatbot has a real consciousness, not an awareness, but a consciousness. And he said to his chief, we have to take into account the consciousness of this chatbot, an engineer from Google. So that means even if for people who are very well documented and their job is to work in artificial intelligence, there is this kind of confusion. That means perhaps, I suggest that education is not only for citizens, but also for engineers themselves. And it's a kind of minimum in philosophy, I would say, which in anthropology, perhaps more. And I think it's necessary, really, even if I don't want to condemn this engineer, is not that. If he has the confusion, is because he is a mentality which favors this confusion. So, how to be sure that the minimum in anthropology can be also taken into account in the formation of numerical digital engineers.
Did I perceive that correctly, that you want to intervene?
Yeah, it is. Yes, I know, and it's again an issue of narrative. It is one specific anecdotal individual case. It was the person in itself was extremely troubled person. It is, I mean, he did say that, and the media, of course, ran away with the idea, and I don't know, probably all of you have been asked by the media to intervene and to tell about, indeed, is AI conscious or not. It's again, it's an issue of narrative, it's an issue of words. Once we start talking about intelligence, about learning, about, it's very easy to continue that narrative, to imagine that from intelligence it will come consciousness, from learning it will come wisdom, from, it is a kind of natural connections that we make because we are used to make those connections about people. But again, we have to understand that these words, when they are used about technology, have nothing to do with their original meaning in the human and philosophical context. So I think it's very important to realize that what these systems do is identify statistical patterns or correlations between data that is represented in a very mathematical way. So there is no steps in the whole algorithm which ever will become conscious just by looking at patterns in data. But I fully agree. It comes from the issues that you highlight, that these words that are used to describe technology are actually not the proper words to discuss what we are doing.
They are framing at least something, but Thierry wants...
No, it's just to challenge. I have no solution. Yes, perhaps it's a special case, but I met some people who think that chatbots or other kind of machines have a consciousness, But one of the question, which is very not so easy to answer, but I think many people can have a different opinion about, is this old one. Is thinking calculating? But if you ask this kind of questions to a lot of people, you will have a lot of different response. And your response is not, but I am not sure that everybody will do this same kind of response.
I agree that not everybody has the same answer. In the Middle Ages, we looked at trying to understand people in terms of fluids and fluid connections. We have tried to understand the brain of people, comparing it to watches or to clocks. So there is always this idea that there is a connection between mechanics and human consciousness or human intelligence. The latest technology that we have for that is AI, so we try to describe AI in terms of the ways that people reason, but increasingly we also describe the human intellect and human conscious in terms of what we can compute. There are mathematical limits to what can be computed, so there is a limit, and theoretical computer science study exactly those limits of what can be computed. So there are many problems, many situations that are not computable in the way that we have designed the computers now. I'm not saying that there will not be other technology later on which is based in a completely different type of paradigm which will be enabled to compute other things, but with the technology that we have at the moment, there are limits to what can be computed.
Yeah, but I think the point is at the moment. Yeah.
Yes.
So we don't know what will happen in the future. And just to misuse my role as a moderator, I want to have a scenario and just not to discuss it here, but to give it to you for discussions, if you like, later. Because I am actually not sure if it is not possible to design a humanoid with tissue that is technologically so brilliant that you cannot distinguish it from our skin, that the face and the mimics is like a little bit with Sophie from Hanson Robotics, but think it just 50 years more in the future, so that you cannot distinguish it really from mimics of a human being. The movement and all the mechanical things, the reaction, the communication, the empathetic communication, watch the film Her, is so perfectionized that from the phenomenological appearance, you cannot distinguish it from a human being. And what do we do then? Do we have to grant that entity dignity? Does it have autonomy? Can we harm it because it can convey the impression that it can suffer? Yeah, that there's hurting something or so. So what do we do then? Do we say, please open up your gut so that I can see whether you're a human being or a robot? Yeah. So I see that we will have much more problems on that. But that's, I think, not in the middle of the interest of those who sit here for breakthrough technologies for health. So I will come back down to earth again and ask this and follow up on this very important issue on open data and open science and open algorithms and this. So how can we proceed here to really make data a global or a common good, to make science something benefiting everyone and societies, and having participation not only from the big companies, especially in these ICT technologies, but having startups coming up because they have access to the data also that the big companies have, data sharing and so on. So how can we develop policies that really make this a shared endeavor for the benefit of societies?
Well, it is a very important but also a very complex question, because
we don't make it less.
Everybody agree that the open science is certainly a very positive point. Why? Because it allows the science to be available through the publications, through the data, to everybody. Each citizen who is paying taxes to pay the professor, the researchers, making the research and providing the data. So it's absolutely normal that each citizen would be able to have access to a publication, to the science in general. Also, the other advantage of the open access, of course, from a conceptual point of view, is that it will be more easy to collaborate together, because all the data, all the publications will be, you will be able to share all the data with all your colleagues. And probably also that a very important result is that you will have a science which is more reliable, because the data are public, so you can repeat the experiments and show with your fingers, this is not working, because I repeated it, sorry, but it is not a reliable result. So this means that there are a lot of advantages. The other point is that, as you know, the library in the universities, until I would say probably 5 years ago or more, they had to pay a lot to big editors to have the journals in the library, etc., etc. But now the problem is that the editors they understood that the open access is very important. And they changed their minds in pay to read to pay to publish. This means that now when you have the open access, the scientist who is willing to write a publication which will be published, he or her will have to pay for the publication to be put in open access. And of course, if you are doing that in high-level journals like the Open Journal of Science or Nature, etc., you have to pay a lot, until I would say 8, 9, 10,000 euro per article. And you can imagine that the scientists are writing the articles, they are reviewing the papers of the other scientists, and now even they are doing the mise en page, because the editor asked that the paper is written in a way that it will appear in this presentation. So this is of course a big problem to resolve. And I would say that in the field of, if you are looking to the scientists in general, in the universities, in the research organism, they are all in favor of the open access. And that's clear. But they are afraid, and this has taken into account, they are afraid about what will be the evaluation. Because until now, the evaluation was based on the quality of the papers where you were publishing. If you are publishing Nature, Science, etc., in prestigious journal, you have more chance to advance in your career than if you are publishing in the Journal de Pharmacie de Belgique, for instance. And this is a big problem, because the young people, we have once again to think to the young people who are willing to advance in their career, to have their research better known internationally. So how will be tomorrow the evaluation? Because actually, for the people who don't know, the evaluation is based in part to what is called the bibliometry, the impact factor of the journal, the H index of the author or of the scientist, etc., And this is the reason why at the Academy of Science we did 15 proposals, and it is not limitative, based on the ID, which is also the ID of the European Community, that we have to evaluate people more qualitatively, looking to the publication, even if the publication is not written in a big journal, but looking what is the message, what is really the science behind this publication, before to make an evaluation, and only afterwards to look to the bibliometric aspects. This is...
Yeah, thanks for this complex picture. Before giving the word to Alison, I want to invite you already to prepare for your questions, because then we can take up your interests and questions.
So I guess it extends to open data, is partly, yeah? So that's a big problem for health data, as we know, because you've gone through the ethics approval if you've got data from a hospital, and you can only use it for your own institution's purpose. And so I think I believe in open data, but I believe in it to serve a role to do certain research. In the UK, and perhaps elsewhere, there is a move towards trusted research environments, where if you want to work with hospital data, you go into their trusted research environment and you can work within those data. It's a very new idea in the sense it's not matured. And there, I think, there will be the notion of being not open, but accessible data. And I think that's really important and valuable, because if you're going to be working on modelling and producing solutions for real-world problems, you've got to access the real-world data. And there are all the issues in being able to access patient data. And the data controller being a hospital, they are not going to be releasing all the data to anybody that asks. And that's also where the privacy-enhancing technologies come in as a family of solutions that, depending on the scenario, will allow you to access information or be able to process data that is not open in the general sense of open, but you will be able to access. So I think we need to have that conversation, which is not just about making everything open, because sometimes you need to access data that is sensitive, and in that case, having other mechanisms to do that is really important. And this is a whole area of research where, when you deploy solutions as well, you want to look at the translational research questions. It's almost impossible then to be able to say, oh, that data we capture, we can now release for everyone else to use, and it doesn't really make sense either. And so it's the context of doing an evaluation. So I think the open has a role, but it's only part of how we evolve research. Yep.
Yeah, I agree with you, but I would like to say it depends which type of data. Because, for instance, if you are looking to once again to the COVID vaccine, it is because the genome of the virus of the SARS-CoV-2 was in open access that we were able to design so rapidly the messenger RNA, which was efficient for vaccination.
You must have had a great time. Good evening to everyone. And I'm going to be brief. First of all, as a member of the Academy, both in France and Academy of Engineering of America, I'm delighted that we could have the opportunity to help this event to happen. The second decision that we made was really to make sure, as we do every year, a global event called Science in the Age of Experience, every year, mostly in Boston, but I was pleased to follow up the request of the Academy and said, we are organizing this event and the program is going to be hosted here tomorrow with a great agenda. So you're all welcome. Anyone who wants to join this event will be welcome. At Dassault Systèmes, on listening to the great panel session about ethics, the reason why we are so much interested with this kind of topic is, of course, we are a science-based company. There are multiple things that I think we have contributed to do to improve the world, in short, using science in many, many sectors of the economy. One of them at the beginning was to make sure that complex object, 3 million parts engine could fly safely, and that the only question you would ask yourself when you go from Paris to New York would be, what's going to be the movie you're going to see, or what's going to be the lunch you're going to get, because no one asked the question, will we land safely in New York? But when you think about this amazing progress in humanity over 1 century, 1 century ago, it was very difficult to do, and there are many people here who have been contributing to make this a revolution. If you look at the statistics of the world, and you take in a developed economy, not speaking about immortality, but speaking about death causes, at the very top high points, of course, as you know, it's about oncology, and of course vascular and heart. But there is a third point that not many people talk about. Exactly at the same level, at the top of the high risk, it's medical practice error. And they are not very often mentioned. If you go, just to give you an idea about the shape of the curve, if you go middle level of the curve, you are 50 years old in good health, come back home, sit in your sofa, and die. Of course, this is statistics, but it gives you, this is the middle of the curve. At the almost bottom of the curve is taking a train, but at the very bottom of everything, if you assume that life is about moving, is taking a flight. So the point I'm trying to make here is that the life science sector can take massive benefit of a lot of technology, modeling, simulation, data science. And this is going on right now. It happened that we participated to 60–70% of the clinical trial for the COVID vaccine. And as you may know, clinical trial cost half of the total cost, half of the time, and 90% of them fail. It's a gigantic investment around the world. If it's like you were saying, I do 100 airplanes, I put all of them in the air, and this one is staying in the air, this is the one I'm going to produce. Now, this is not going to happen over one day, but we have demonstrated that with science and technology, putting this together, powerful collaborative methods, approaching it on a system view, which is really about having people understanding each other, we can change the game. And those clinical trials were operated on platforms that really reduce the cycle time from 5 years to maybe a year. So the move has started, and this is the topic we will cover tomorrow with many prestigious speakers. And I want to notice that the one from Moderna, a great partner, could testify that this transformation is going on and will not stop. Because the biologic, not only is difficult for research, for lab, for clinical trial, but the question of producing them is far more complex than just chemical-based pharma, which is a big-scale problem. So I believe that we can work together. We have many partnerships with many of the companies you represent or you have been working with around the world here, on that the possibilities to create virtual twin of human at a certain scale, certain approximation, as this beautiful, beautiful power. It helps people who are not understanding each other to finally try to observe the same phenomenon, imagine similar solutions, and invent, create new solutions we have never thought about. That's the program for tomorrow. And if you have time, don't hesitate to stop by. We will have a great audience and a great program. Thank you, and I will be joining you for the dinner. And I know that, as Professor Johnson told me, the commonality between all presentations across the board that we have seen at this event is the system approach, the connection between disciplines who are not used to work together, to understand each other, is a beautiful human journey. That's the one we want to organize. Thank you very much, and enjoy the evening.